NIH the 19 institutes of 27 total that now fund human microbiome research. I formed this group about 5 years ago because I saw all this emergent research occurring, and we felt we needed to coordinate this. We've done a lot of things over the last 4 or 5 years, but this one is probably going to top everything. We want this 3-day workshop, and it's being webcast, so there's an off-site audience as well. We want this workshop to accomplish 2 major things. It is the end of the Human Microbiome Project, so we really want to assess what have we accomplished, what are the legacy resources and so on, how are the data being used, how are the tools being used, but probably more importantly, we want to ask the question, because this is a growing field, what else do we need to do? So the overall theme for all 3 days, the way the talks are structured, the way the roundtables are structured, even the way the joint QA sessions are structured, is to have us revisit over and over again from the community, what are the gaps, needs, and challenges for this field? Okay? Because we have 19 institutes represented here, we're all going to take this information back to our own home institution and hopefully utilize it to form possibly new FOAs or simply to direct the dialogue within our own institutes. Okay? So I just want to take a couple of minutes to kind of describe in more detail what I just quickly outlined. I see that all the seats are not filled, but I think some people are filtering in. So just come in at any point in time. I want to cover the goals and purpose of the workshop and what the charge is to you as participants. Let me just take a moment, though. Do you see that watermark in the back? I mean, talk about committee engagement. This is a scientist at Genelia Farms who's married to one of the committee members, and she was inspired. She's an artist as a hobby, and she was inspired to develop this graphic of the microbiome family. Every outline of all 3 people in that graphic is made up of microbes. Isn't that cool? So I mentioned the Trans-NIH Microbiome Working Group. We've been in business for about 4 years. Now it represents 19 institute centers and offices which support human microbiome research in the extramural program. So even though we have a lot of intramural people here and calling in through the webcast, because we're program directors, we actually dedicated most of our attention to trying to help support human microbiome research with the extramural communities. We've been very busy this past year. Not only has it taken us a year and a half to organize this workshop, but at the same time, we're conducting an in-depth portfolio analysis over 5 fiscal years of human microbiome research at the NIH in the extramural program, and are working to get it published. If all goes well, we're going to submit it to Cell Host and Microbe for consideration. So let me just trace very quickly, though, kind of like the momentum, the impetus for this workshop. Certainly you know about the Human Microbiome Project. It's been a 10-year community resource activity. Resources like building an entire research committee is one example of a resource. Resources like reference data sets, computational tools, clinical protocols, and so on. It's a major investment by the NIH, the Common Fund at the NIH, to really help catalyze, kickstart this field. You can see all the institutes, medical centers, and so on that have been involved. At the same time, we saw growth of human microbiome research support across the NIH. So from about 2012 to present, we're now seeing of those 27 institute centers and offices, all of these different institutes funding the area. Now, because they each have their own mission, sometimes this work can get siloed, unintentionally perhaps. And in other cases, a gap might be identified by one institute that's clearly a gap for another, and so we want to have this workshop be a place where we can really compare notes about what we think as an agency is needed for the field, not only each institute. The other thing that kind of drove this workshop, you know this, in 2015, back in the Obama administration, which now feels like a lifetime ago, he commissioned, the White House's Office of Science Technology Policy commissioned a committee called the Fast Track Action Committee, in which these 16 agencies, look at the bottom, all the logos, this is the logo of logos here, we were commissioned to do an assessment, a portfolio analysis of microbiome research across those agencies. Now, obviously not just human microbiome research, included microbiome research writ large. So the study of microbial communities using modern 21st century approaches. Okay. We published a paper in the inaugural issue of the journal Nature Microbiology in which we summarized what we came up with. We identified $900 million across the federal government over 3 fiscal years across all of these different agencies. Yes, it's true NIH had the lion's share, but we are not the only game in town. So that was a very important realization when we did this data call, this assessment. That birthed the National Microbiome Initiative in 2016. It also birthed, for the federal side, the Microbiome Interagency Working Group, which is the committee that talks to the agencies to figure out what are we doing in different agencies around a particular topic, in this case, human microbiome research. And you'll see on day 3, the Joint Agency Panel, many of the members are actually representatives on this committee and have all agreed to participate on the panel and talk about what they perceive as the gaps, needs, and challenges for their agencies and across the federal government for human microbiome research. So now we're here in 2017. Okay, a lot of momentum from 2012 in particular, but here we are in 2017. So to repeat then, the purpose is to really take stock of NIH's investment in the Human Microbiome Project, very important activity for this agency, and to evaluate what's really needed to advance this field over the next decade. We want all of the audience members, the speakers, the poster presenters, the roundtables, the agency panel members to help us identify and develop this priority list of knowledge gaps, technical hurdles, new approaches, and research opportunities. We might see 1,000 different ideas over these next 3 days, or we might start to see emergent things coming up as we talk about gaps, needs, and challenges. Let's see how it goes at the end of 3 days. And for us as an agency, then, we want to use this information to help inform development of novel prevention and treatment strategies that are based on host microbiome interactions over the next decade. This is a Uber workshop, I'm going to call it, because if you look at the themes for each day and then the sessions within each theme, you can see that each of them could be a workshop alone, a standalone workshop. So the 3 themes are, obviously today is overview and approaches, but then theme 2 is interaction of the host microbiome system, and then day 3, or theme 3, is microbiome interventions for maintaining health and treating disease, and then the joint agency panel. Okay. Within each theme are a series of sessions. I'm not going to read them to you, but they're structured around the overall theme. Day 1, day 2, day 3. And then for the joint agency panel, these are the agencies, CDC, FDA, DOD, NIST, NSF, USD, VA, that are the agencies that are either currently funding human microbiome research activities or planning to mount them. And that's why we brought them together. I also brought together a few offices under the Office of the Director, like the National Institute for Minority Health Disparities, Office of Research on Women's Health, Office on AIDS Research, because those offices and institutes have particular interests in the microbiome. The speakers and the participants were selected from this pool of approximately 3,500 investigators that the TMWG identified when we were doing the portfolio analysis. We spent many, many months debating and discussing who's going to be a good representative for that area of the microbiome and who's going to be good about talking about what's needed for the field. We didn't want someone, to just stand up here and talk about their own research. We wanted them to be a representative of that area in the larger microbiome discipline. So a lot of effort went into picking the speakers and the participants. This thing is going to get published. We just had a core writing team meeting just now with the editor for A Cell Host and Microbe. The core writing team are Greg Buck, Curtis Huttenhauer, Mike Snyder, and Owen White. We have 16 different TMWG committee members taking notes on Google Docs and otherwise for this meeting. And all speakers will contribute to the report. So no one's going to speak for you. You'll be able to contribute and edit the final report. It'll include a summary of the presentations, maybe more importantly, it'll include an assessment of what was identified as the critical gaps, needs, and challenges. And if all goes well, it gets submitted to Cell Host and Microbe. Thank you, Lakshmi, wherever you're sitting. Okay. Now, I'm going to do a little bit of housekeeping. I don't think I'm chewing up too much time. I want to do a little bit of housekeeping because there's a lot of moving parts to this workshop. First of all, speakers, please, please, please load your talks on a flash drive and use the laptops that are on the podium. We have a Mac and a PC. That'll save us a lot of time. Please make sure, even if you don't have a slide, close your talk by addressing a one critical gap, need, and challenge. Because we're going to have a round table at the end of each day, those of you who talk today, please sit in the front so that it makes it easier for us to entertain questions from the audience and the off-site audience. Moderators, you have a big job. This is a really packed program. So please, please, please keep your speakers on time. Each of you have, except for the keynotes, each of you have 15 minutes. And each session has a joint QA session, so we're not going to entertain questions at the end of each talk. Instead, we're going to have a joint QA session at the end of each session. So moderators, you got a big job of keeping people on time. There's timers here and there's timers on the table. Poster presenters, please put up your posters. I see some up already. Up before the keynote talk, you have all day long because we have 3 different poster sessions. So put your poster up before the keynote talk and address a gap, need, or challenge at the round table if you haven't addressed it in the poster. But be sure then to take your poster down at the end of the day, because then the next round of posters have to go up. More housekeeping. You already received information about the pre-order information for box lunches. I think it's too late for today if you didn't pre-order for today, but we do have a cafeteria and a cafe up on the upper level on this end of the building. We have a tent. A 250-person tent. The idea behind that is, because I knew it was going to be overwhelm this building if we try to get everybody through the cafeteria. And there's really no good seating except in the cafeteria. So we have a tent out there on the lawn. That's for us, okay? 250 people. And your breaks, your lunches, it's up all 3 days. So even after the workshop is over in the afternoon, if you want to keep on talking, just go out there at the tent, right? Use this as a networking opportunity, too. Dinner options. I don't know if you noticed, but the registration table is on this side of the hallway. On the other side of the hallway is a table that says microbiome-related materials, but also on there is information about local restaurants and so on for those of you who are out of town. Oh, I should say this. Restrooms, another housekeeping, are at either end, right? Either end of this hallway. There's also a set of restrooms at the top. And we're webcasting this entire thing live. Each day has its own URL. We also have a Twitter hashtag, hashtag eTeamMicrobiome, where people can live tweet this entire workshop. We also have a Gmail account, eTeamMicrobiome@gmail.com. Please let me thank Jen Fetweiss from VCU and Aaron, who's sitting around here somewhere, I thought he was, Aaron Markets, from the Common Fund, who did a great job of organizing and coordinating the Twitter account and the Gmails. And the reason we're doing this is we want to be able to allow people to live tweet the workshops, but also we want questions from the off-site audience to be sent to us so we can entertain them at the daily roundtables and the joint agency panel. The cutoff times for Wednesday and Thursday are 4:30 for questions. The cutoff times for Friday is 12 noon. We have a workshop website which has everything, including the program, everything you need to know about the workshop. But please make use of this Twitter and Gmail. However, we also want to make sure that every speaker was comfortable with having stuff tweeted, webcast, photographed, and so on. That's true for the speakers. That's true for the poster presenters. Okay? So please, please, we're all trying to ask every speaker what their preference is. But please ask the presenter, whether the poster presenter or a speaker in the session, if they do or don't want their talk poster live tweeted. And presenters, please also share that responsibility. Let the audience know what your preference is. Okay? Photographing, especially with posters, but also with slides. Please ask the presenter if they mind if they have their talk or poster photographed. Presenters, share the responsibility. Let the audience know what you do or don't want. Okay? And especially because we're webcasting, please let your moderator know if you don't want your talk webcast. Okay? It could be simply a headshot, but if you don't want your slides shown, if you don't want a headshot, maybe the audio is okay, but whatever it is, please let your moderator know so we can accommodate it. We have a whole crew of IT and videocast people in the back, and they'll accommodate us. So let's get started. I'd like to first introduce Howard Auckman. I've known Howard for a very long time. I thought it was so important to provide a evolutionary perspective to the microbiome. We don't often talk about the evolutionary perspective of the microbiome here at NIH, but clearly the microbiome and host is a co-evolved system. So let's hear from Howard. Howard is our... Walter, can you help Howard? Here you go. Let me give you this. And the title of Howard's talk is What the Great Ape Microbiome Can Tell Us About the Human Microbiome. Thank you, Howard. Howard is one who chooses not to have his talk webcast. The audio will be across the webcast, but no headshots, no data. Thanks, Howard.
Thanks so much. So thanks so much for the invitation to speak with you. I'd like to talk a little bit. The first half of my talk will be sort of an overview of where we've come from in the last 10 years, and then I'll basically shift to tell you where we think we're going, at least in the future of our work. And so we've been working on the great ape microbiome, and the reason for that was actually really simple, is that we, at the time in 2007, when the Human Microbiome Project had started, we had a lot of information about what was going on within humans, but we decided as evolutionary biologists to look what was going on in great apes, our closest relatives. So what I show here is a figure of the relationships of great apes. We have humans here. Our most close relatives are the chimpanzees. The types of chimpanzees are the bonobos, that is Pan paniscus, and Pan troglodytes, the common chimpanzee, of which there's at least 3 subspecies. And we're more closely related to one another than we are to gorillas. And then the other great ape species are the Asian great apes, that is the orangutans. And a lot of sequencing, a lot of work has gone into the fact that show that humans and chimpanzees at the DNA level are about almost 99% identical to one another. So our questions were really simple when we started out about the gut microbiome. We wanted to know whether the gut microbiome of chimpanzees was similar to that of humans because of their genetic relatedness, the recent ancestry. Are they different due to their diet, geography, since these are African apes and most of us are here in the States, that we've been testing, our different social structure. And then, most importantly, is what might the ape microbiome inform us about human microbiomes? So when we did this work, the only work that had been done on, and the samples that were available were principally from animals that were from zoos. So we wanted to find samples that were from their natural habitats of apes. And from that, I worked with a colleague of mine, Mike Worraby. And Mike Worraby goes to Africa very often. He's looking for the origins of HIV. And so he's taking chimpanzee fecal samples. So I called Mike. We got many samples. He got me in touch with Beatrice Hahn, who has a large repository, who was then at the University of Alabama. She has a very large repository of fecal samples of African apes. And we took a very small sample looking at 2 species of gorilla, or the 2 subspecies of gorilla, the eastern and western lowland gorilla, the species of chimpanzees, my pointer's not, shown on the upper left, that's Pan troglodytes, and there are several subspecies. These are by the blue dots, and the green dots on this are Pan paniscus, that is the bonobos, and you can see where their territories are. And we had humans in this from Africa and from the United States. Now, when we produced these data, of course, we used the technique, I'm sure all of you know about this, unless you've sort of been asleep for the last 10 years, okay, because this is the technique that most people have been using. It's actually based on work that was done by Norm Pace, culture-independent techniques for looking at microbes, just by PCRing up genes in that. And then it was made a high-throughput approach, sort of based on the work of Mitch Sogan in 2005. And it's a very simple methodology, which is why it's so widely applied. You collect samples, you extract DNA, you amplify the 16S ribosomal RNA, and it's very nice. 16S ribosomal RNA is ubiquitously present in all organisms, and it has highly conserved regions and somewhat variable regions, so you can make primers to the conserved region, amplify up a variable region. Then you sequence it up, you get many, many sequenced amplicons. You bin these into OTUs. OTUs are operational taxonomic units. This is a terminology that was developed in the 1960s by Bob Sokol, and it's the group of whatever organisms you're looking at. It's not assigning any taxonomic rank whatsoever. It's intentionally vague term for that. So if you're looking at some group, in this case, we're looking at the OTUs, and we're just binning the sequences based on their overall similarity. So we get several OTUs. You taxonomically assign these, you can buy several databases that are available, even though many studies stop at this point. Actually, it's after this point that all the fun begins, once you have everything taxonomically assigned in that. So when we first developed these data, there were very few tools in which to analyze them. And we produced these sort of heat maps, and this is just showing you a heat map of what's present, and it's the relative abundance of these things, of these OTUs among these A species. The humans are at the top, the chimpanzees, again, the blue dots, the bonobos and the gorillas. And if you squint your eyes, you can sort of see that there might be some differences in that. And we can see that in all, this is just a heat map of the approximately 9,000 OTUs that we got out of this. And they were OTUs that were present in 2 or more individuals. And the reason they're in 2 or more individuals is because we thought if something was unique to an individual, it could have been an artifact or something, so we would just throw it out for the time being. And we find out that the 3 dominant sample of phyla, all the samples were Bacteroides, Firmicutes, and Proteobacteria, and also there were a couple other phyla that were present in the samples. So when we first started to look at that, we wanted to know what the relationships of the microbiomes were to one another. And we already knew the relationships of the host. And here's an unrooted phylogeny, unlike the one that I showed you first, that shows you the relationships. In this case, the bonobos and the chimpanzees, of course, are the most closely related. And of course, the bonobos are fairly closely related. That shows you the different subspecies of chimpanzees. Then the humans are most closely related to them. And outside of that are the gorillas. So this is the tree based on mitochondrial DNA. Humans are fairly closely related based on mitochondrial DNA. Our closest relatives are the chimpanzees, then the gorillas. So when we looked at the community composition based on the abundance of these OTUs, we got a tree that looked like this. You can see the terminal branches, that is the colored branches, this were really long, showing that there's a lot of variation between individuals in the species. But if you actually looked at this closely, the interspecies branching order, and that's what's coming up in the numbers here, happened to match the tree. So this showed us that host phylogeny actually was a driving force in the divergence of microbiomes in that. And so you can see the same species here are clustered together based on their microbiome. So if you were to give me a sample from any of these species, I could tell you what species it was based on this microbiome. But also, the microbiomes of humans were more closely related to those of chimpanzees and the 2 chimpanzee species were, of course, most closely related, just like their genetic relatedness. So we looked at that, but of course, then it started to bother us. Why are those terminal branches so long in that? What are the forces that are driving the individual variation within a species? So we looked to a very large data set that was available at the time. The data sets that were available for humans were not as extensive when we first did this analysis. And we turned to looking at chimpanzee microbiomes. And because there were lots of samples that were available for Gombe chimpanzees collected over the past decades. These are the same chimpanzees that Jane Goodall has been looking at since the 1960s. And we took chimpanzees that were from 2 colonies, the Kasakala colony and the Matumba colony, so we had 2 geographic regions, so we could see whether geography played an important role in the separation of the microbiome. We also, as you can see here, we know the genetic relatedness of the individuals, that is, the parents and the offspring. They've all been watched, and so we could tell by the fecal samples which individuals they came from. So we've had that. We have cases where we had the parents and the offspring. We have cases where the parents and the offspring went into 2 different colonies. There are cases also, if you can, most of the samples came from 2008, the same season we were trying to correct for diet. But also there are samples from 7 individuals who were collected in the year 2000, 2001, and 2008, and those are the ones that are cross-hatched on here. So we had in the total approximately 35 or 40 samples. We did the same exact procedure on that, and we saw how we could assort the microbiome. First thing we had to do is find out, does Gombe chimpanzees, do they look sort of chimpanzee or Gombe-esque? And you can see here in a phylogenetic analysis the same sort of thing that I showed you before, that all the Gombe chimpanzees were more closely related to one another based on their microbiomes and on their genetics than they were to other species of pan-troglodytes and that. So they form one sort of cohesive group together. They're based on their microbiomes. Their gut microbial communities look like they're from chimpanzees, and they're all more closely related to each other. Here's just doing a phylogenetic analysis. Here's a principal coordinates plot showing that all the Gombe chimpanzees cluster together. Separating out the factors, we saw that there was some geographic structure in the population, the microbiome. So looking at the different microbiomes and plotting where they are, you can see the communities can be separated most of the time. There are some individuals that are intermediate, and actually those are the individuals who were born in one community and then moved to another one. So it told us that there was a lot of sharing between members of microbiomes among members of the community. There was also some sort of temporal structuring to the data. Those samples that were collected in 2008, just on this plot, all started to cluster together. So there's temporal structuring. Surprisingly, there was almost no genetic structuring to the populations. If we look at the overall relatedness of hosts, that is sort of looking at Hamiltonian coefficients of relatedness, you're related to your children by 1/2, to your nieces and nephews by 1/4. That's shown by the blue dots here. The darker the blue tells you the more closely related. You can see more closely related hosts here don't really have more closely related microbiomes. So just to put this in order, to show you the statistics, the gut microbiota can differentiate chimpanzees by communities and by community affiliation and by their gender, but they couldn't differentiate by overall genetic relatedness. Now, while we were doing this work, a lot of information had come out about the human microbiome, and then we had many, many samples. So we were able to have a sort of a direct look at the contents of the microbiome of humans versus chimpanzees and gorillas, and looking at the great ape communities. And if you look at the order, look at the taxonomic ranks, not just what the OTUs are, but how they're being assigned, you can see that at the order of phylum, if you took all the humans and all that were available at the time versus the chimpanzees, we can show here, without even labeling what the phyla are, that the profiles look about the same. This is the same thing we found before. There were these 3 major phyla in the microbiome and a few other ones. Looking at the level of bacterial class, they look pretty similar. At the level of order, they look pretty similar. But once you get down to the generic level, that is for the bacterial genus, you can see there's some sharing, and the colors just tell you specific taxa, denote specific taxa, but there are some differences. And at the time, a paper came out that looked at what they called enterotypes in the human microbiome. And this was from Per Bork's group. And what they did, looking at 30-some individuals, mainly from Europe and the United States, they found out that they showed that the microbiota assorted into 3 groups, which they called enterotypes, that were not nation, continent specific. They weren't related to age, sex, or body mass index. And what you have to realize about enterotypes, these are not discrete groups, but they're based on the relative abundance of different taxa. So we have the enterotype 1 here, which is called the Bacteroides one. That means there's a prevalence of Bacteroides in the group and less of other groups. There's the Prevotella enterotype, that has a high proportion of Prevotella, and that there's a Ruminococcus one. And so they said that there were 3 enterotypes based on the overall, the relative abundances of different genera within the microbiomes. Trying to figure out what the purpose of these enterotypes are, or even if they really existed, there are a few really nice papers that were published. I particularly like this one by Gary Wu, where they looked at the effects of diet, linking long-term dietary patterns, as you can see, to microbial enterotypes. In this paper, some authors on this are in the audience. Here they found 2 enterotypes, the Bacteroides and the Prevotella, which that is different abundance of each one in that. Of course, they didn't favor 3 enterotypes. They only favored 2 in this. It seemed like long-term diet was associated with enterotype. If you had a high-fat, low-fiber diet, like most of us have, we had the Bacteroides enterotype. But if you had a low-fat, high-fiber diet, you had the Prevotella. And they did a nice gene experiment to see whether you could shift from one to another. So when we started to look at that, we were wondering, are these things real, these enterotypes? So we started to think about this, and how could we compare this to chimpanzees at the time? Are these human-specific features, or are they present also in great apes? Now, if it arose from its switch in diet to a high-fat diet, we shouldn't see it in non-human primates because they don't really eat very high-fat diets. But alternatively, if the polymorphism is present and has been maintained in other species, then maybe it's a real adaptation. So what did we do? We looked in these Gombe chimpanzees, looked in the same studies, and we focused on these because they're about the same age as human populations. The Gombe chimpanzee population probably arose about 100,000 years ago based on genetic data, and human populations diversified, came out of Africa approximately 100,000 years ago. So we did the same sort of analysis, and we found that the data partitioned into 3 groups, just as we saw in human populations. And in fact, we had the same species driving the enterotypes in chimpanzees and in humans. So we can see here, there's the Bacteroides one here, Bacteroides in the chimpanzee as well. So you have about the same enterotypes having the same compositional features in chimpanzees and in humans. Also, as I mentioned, we also had samples that we could follow longitudinally. So we looked at this, and we found that over at least year-long periods, every chimpanzee we saw switched its enterotype. And so here are the 7 samples for which we had multiple samples for multiple years. And you can see in every case of this, there was a switch from enterotype 2 to 1. There was no consistent pattern. Even among relatives, we have two chimpanzees that happen to be siblings. They started with different enterotypes, and they ended with different enterotypes in that. Now, you might recall that, as I mentioned, that if you give me a sample from chimpanzees and you give me one from humans, I can always differentiate these samples and tell you whether it's a chimpanzee, the fecal sample, or human, just based on the content of its microbiome. Okay, so I can separate them. So if you think about this, what does this tell you about the origin of enterotypes? Well, that means they were probably ancestral, that is, to both chimpanzees and humans. And actually, we have found some enterotype features in gorillas as well. But it shows that they're ancestral, they were present in the common ancestor, and they've been maintained for millions of years and did not arise as a result of the modern diet. So if you think about this in evolutionary terms, you can say there's ancestral strains here of some genus here, and it's been following the lineages of chimpanzees and humans being separated for millions of years, just as the genetics of these organisms. And it's very reminiscent of things that are sort of symbionts that sort of co-diversify with their hosts. So the next thing we did was to look at the pattern of co-diversification of microbes, because it gave us the idea that maybe microbes are somehow co-diversifying with their host. And so we asked the question, did the bacteria co-diversify with their host during grade 8 evolution? And so this is a cartoon that's taken from a paper by Nancy Moran and Dan Sloan, but it just shows how you could have community assortment of different microbes and different hosts. And there's one way where you can have, and these are just the extremes, you can have intermediate cases as well. You can have an environment with lots of microbes in it. Let's see if I have a better pointer here. No. You could have an environment with many microbes in it, and then the communities again assort, that is, they select from the environment so that you get different ones in it. Or you could have pure co-diversification. That is, you have certain bacteria that are ancestral, and they've been co-diversifying with the lineages over evolutionary time. So our prediction, of course, is that because humans and other organisms uptake their gut microbiota each generation anew, and there are changes in the individual lifetime that they're taking in from the environment, it suggests that this figure here on the left is correct, that somehow the hosts select from the environment for that. So we wanted to test for co-diversification between gut bacteria, but unfortunately, the type of data that we have makes it extremely difficult to do this. So far, I've only been telling you about community compositions, that is, what's the relative composition of one taxon and another and the relative abundance of these. And to find evidence of co-diversification or co-speciation, most ways people do it is to look for the concordance of phylogenies between the bacteria and the host. And here's the classic example shown from Nancy Moran's data, where you have aphids and their endosymbiont Buchnera, and you can see the two trees, the one on the left of Buchnera and the one on the right of the aphid host, are almost completely concordant. That is, they match. And that gives you fairly good information that these things have co-diversified, in this case, over millions of years. But you can see to get this Buchnera tree, they had to take an individual gene, that is ATPase, and follow its phylogeny, build a phylogenetic tree of that. But unfortunately, the 16S sequences that we've all been using in order to look at microbial community composition evolved way too slowly in order to do this. One OTU may contain sequences that diverged probably on the order of 10 to 20 million years ago, which is the entire time of the great ape diversification. So we have to start looking at features that looked a lot faster. So, and this just tells you it just explains in a cartoon what I've just told you. We have an OTU, but within each OTU, we might have many, many bacterial species or even genera, and each one of those could have co-diversified with the host. So due to its slow rate of evolution, 16S OTUs cannot tell you very much about co-diversification, because it could have very, very divergent. So we needed to develop a method that had much higher levels of resolution to that. And so to do that, what we did was the same thing that everyone's done with 16S genes, but we do it with protein coding genes. And you don't have to read any of this because it just tells you everything that I've already said. All you have to know is from this is that we've termed this technique called phylotags. So if we get 16S tags, sometimes they're called I tags, or pyro tags, depending on whether it's made on an Illumina or an old 454 machine. But we just call these phylotags, so we can follow the phylogeny of an individual gene that's in the microbiome. So we did this, and we focused on a gene called gyrase B, which is an Enilobacteria, and we focused on individual bacterial families. So these are just from my gorilla samples here. We looked at a couple families, the Lactospiraceae. If you look within a sample of gorillas, in that, we just take all the samples together. We have approximately 100,000 reads. If you look in that, we have a total of 425 of those reads were assigned to the Lactospiraceae, and overall you get 73 OTUs at the 99% level. If we look at the gyrase B gene, of course, all of them are targeted to the families, so of course there are 8,000 reads, all of that, and we get 3,000 tags now, or sequences, or OTUs that denote the species. More impressively, if we looked at the Bacteroideaceae, again, we have the same number of reads. There were no reads assigned to this family when you look at 16S genes, but if you target the gyrase B gene, you can see we have up to 300 of that. So we have much within each of these OTUs, and sometimes things that are not even detected, we have lots of variation in that. So then we started to do the same sort of study that we did before, taking the same samples. We have chimpanzees, 2 species of chimpanzees, the gorillas and humans, which all came from New Haven, Connecticut. I was at Yale at the time. And we sequenced them, and we had this number of OTUs for each of these cases. So we had the Bacteroides, we also tested Bifidobacteriosae, and these things are sorted into many species. And what you can see here is that the strain phylogeny of these different Bacteroides species in there matched, in many cases, the phylogeny of the strains. So this told us that these were strains or species of Bacteroides and Bifidobacteria that matched, that is co-diversified or co-speciation with their great ape host. Now, people always ask, well, okay, the one at the top here, Bacteroideaceae species 1, what is it? You don't even have a name for this, okay? Even though here's a species that's been co-diversifying with great ape hosts for millions of years. So just for this meeting, I've given them names. We have Bacteroides letoproctoriella and Bacteroides curtis-huttenhaueri, okay? So now if you want to know what species are co-speciating, co-diversifying, we have names for it here. Now, this isn't always the case with all the bacteria that we've looked at. We look at the Lactospiraceae, we find many OTUs, and this thing has been transferred many times within all these lineages. So as a result of this, and this is, by testing for co-diversification, we see humans and African ape microbiomes are composites of strains that have been co-diversifying, and those that have been transferred on between lineage for millions of years. If we look at the divergence times, they're collinear with those of the human hosts. So this indicates that nuclear DNA, your mitochondrial DNA, and many of your gut bacterial genomes have diversified in concert during evolution. Okay, so now that is by way of background information. What about microbiomes of most current concern? So one thing that we're looking at is what are the phylogenetic depths of this co-diversification? Do different human subpopulations have their own strains, much like we've seen with Helicobacter, for example? That is, different human subpopulations will have their own strains of that. So has the co-diversification also occurred within human population, or is there just transfer of these strains among humans? Also, how far back does it go? Do mammals have these bacterial lineages co-diversify with all mammals? We want to know whether this co-diversification actually denotes coevolution. Evolutionary biologists basically are looking for function in this. Okay, we know these things have co-diversified, but are they important? Is there functional relevant? If you have chimpanzees have a strain of a species and you have one of the species, is it better for you to have the one that's normally in humans, or doesn't it matter at all? It's just by chance that these things have co-diversified. Also, we've developed this phylotag method, but we're now starting to look at metagenomic data, since this is what everyone is producing. We know, could we get these co-diversifying strains from metagenomic data instead of taking a completely targeted approach and looking through a lot of data? Another issue that we've looked at and related to another project are absolute abundances of bacteria more relevant than relative abundances. The techniques that we've been doing so far, using the 16S tagging method and even the phylotag, just gives you the relative abundance of the different taxa. But it could be that some organisms have very depauperate microbiota, and a couple interesting papers and hopefully we'll hear about this later, on insects have come out and saying, even though people have been doing a lot of tagging and looking at microbiota, they actually have very, very low, and there's a paucity of microbes in the gut microbiota. How do we classify microbes? I just made up some names of microbes, of course, but how do we really classify microbes to species or any higher taxonomic levels? And also, what we've also been wondering about is what are the unclassifiable bacteria in all these complex communities? So to look at one of these issues, we've looked at how much strain co-diversification, because we looked at metagenomic data sets. And what we did originally is to look at metagenomic data sets that were already available, in this case for the Malawian microbiome data sets, to see whether we could find any of these co-diversifying strains. That is, we searched these data sets for gyrase B sequences that were part of the right species. And you can see here for bifidobacteria, we find cases where there are Malawians who we found a few sequences of by dredging through this. And in fact, the Malawians are outgroups to other human populations, which is exactly what you expect for genetic data, because Africans are the most ancient human populations. We've done this again with looking at Bacteroides. We found a Malawian sequence. It's an outgroup to sequences that we found in chimpanzees and bonobos. So I'm calling this Bacteroides knocking gorillas. Okay, there's my species name. So here's a co-diversifying strain that's not found in gorillas, but it's found in humans, at least Africans of African origin, and in chimpanzees. So we're finding that at the depths of most metagenomic sequences, we really don't find many of these co-diversifying lineages because we're searching for specific homologs. But things are starting to look better when we get a metagenomic sequencing depth of somewhere on the order of 100 million reads. We should be able to detect them. Another problem that we've had is how do we classify microbes to species or higher taxonomic orders? You may have seen this paper by Laura Hugh and Jill Banfield that was published last year in Nature Microbiology. And here they claim that there's a new candidate phylum radiation where there are all these new phyla found in some metagenomic surveying. And they just gave these names. There's Curtis bacteria, Roy Curtis, Julian David, Stuart Levy, Sue Gottesman, John Beckwith, Norm Pace. They just gave them all their own phylum, different phylum name. Okay? So are these new phyla? Really? How do we tell whether something is a new phylum or a new species? So we want to know how to classify that. And then also, every time I look at a microbiome data set, it has a lot of unclassifiable bacteria. How do we know whether it's a new phylum or not? So one very simple thing that a student was doing in the lab is actually trying to figure out, when we have these 16S sequences, are they real or not? So we've been taking these small sequences, we've been taking novel primers to that, and just making full-length 16S sequences out of that, and then trying to classify these later on. And most of the classified ones already do get assigned to known groups once we do this. So a lot of things that people call are unclassifiable are actually known bacteria. They just have to be strange variants. A few of them are artifacts that we didn't pick up by our Chimera checker or other types of things, but some of them seem to represent new phyla. And I've noticed just recently, I saw a paper in bioRxiv by Steve Quake's group, where other people are starting to look at all these unidentified bacteria that are in the microbiome. These are from blood samples, but you can see by the title, humans are colonized by many uncharacterized and highly divergent microbes in that. Okay, so we have all these things that cannot be classified. How do we know what taxonomic rank they are? How can we classify microbes to a new phylum or species? So for the last few minutes, I just wanted to become an evolutionary biologist for you and tell you about what constitutes a species. Okay? Now, there's this thing developed by Ernst Mayr in 1942, which is called the biological species concept. And he defines species as actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. And this is actually the most common definition of species. It's used for the Endangered Species Act. And the thing about this is it's not arbitrary at all. I don't look at sequence cutoffs. I don't look at a type specimen and say something's a species. It's not based on attributes of individuals, but of a population. That is, they can interbreed with one another. And the biological species concept, that is, the species itself, is the only taxonomic category that's made based on a biological process. That is gene exchange, or abilities to have sex. So we can see here, it has interbreeding, reproductive in it, gene exchange, gene flow set. So how do you apply something like this to bacteria, or any asexual life form for that matter? Bacteria are asexual, they reproduce by binary fission, and therefore they're clonal. They don't reproduce, they reproduce as clones like that. So each cell is reproductively isolated from other cells. So what constitutes a bacterial species? Maybe it's just a single cell, since it doesn't reproduce. So, in light of the biological species concept, how can you classify bacteria and any asexual bacteria into species? And the answer is you can't do it. And lots of people have tried. So you'll find paper after paper trying to define prokaryotic species or asexual species. And here's a slide from a talk that Erkka Stakkebrand gave in 2004 at an ASM meeting, where he basically throws up his arms. It's a love-hate relationship of prokaryotic species. He says they're arbitrary, they're artificial because it's a man-made concept, and it's pragmatic, that people define species how they want to do it. Now, we know, though, that even though bacteria are asexual, there is recombination among bacteria. And if there's sex and recombination, maybe we can start to apply the biological species concept. So how do I detect bacterial recombination? Well, this has been done since the 1970s and 80s, work that was pioneered by Roger Milkman. And here's a picture from an old paper of his. And what he's done, 5 minutes? Oh, no problem. And what he's done is he basically aligns some sequences, and he says how much recombination is there. So he looks for tracks where there are a lot of changes versus individual changes. If there are a lot of changes, what's the chance that a lot of mutations are going to happen next to each other? He thinks it's a low occurrence, so he says it's a recombinant region and that. And he just counts up the number of individual point mutations versus these blocks. And in this case, if you were to count this up, you would find out that there are 10 blocks that are dark colored and 50 individual point mutations. So he has an estimate for E. coli, which he calls the polymorphism introduced by recombination R, and that introduced by mutation M. And he says there are 10 blocks here and 50 here, so R over M equals 0.2. So recombination does affect E. coli in producing variation. So this has been done with many, many species now that there's been a lot of sequencing in place. And here's a quote from Brian Spratt in which he says, populations in which recombination is either absent or extremely rare probably represent the minority of situations. This is a backwards way of saying that almost all bacteria recombine at some level. And if we show you this, here's a compendium table taken by a paper by Voss and Didelot, shows you the for all the species that were surveyed at the time. Here are the clonal species, that is, they have very little recombination. And here at the top are panmictic, that means freely recombining population. And you can see the R over M values. Well, there are none that are zero. Sometimes there's very low levels of recombination, sometimes there's very high levels of recombination. But since there's recombination in all of these, perhaps it's possible to apply the biological species concept. So this is what we try to do, and to define species as groups that exchange genes, just like we do with animals. So how did we do this? We just tried to detect recombination, and we did this by looking at what's called homoplasy. And so homoplasies, anyone who's drawn phylogenetic trees has heard people use these terms. These are characters that are not shared by common ancestry. So I just showed this on the tree here with this base T. If you were to line some sequences, you can see there are very different parts of the tree have the same base. So basically, this could be transferred from here to here, and it looks like a recombination event, as opposed to it being ancestral and then lost, or all these individual mutations. So we go through all these phylogenetic trees, and we count up homoplasies in these trees, and we see these as a pretty robust signal of combination, and you can look at individual polymorphic sites over the entire genome. So our technique was really simple in doing this stuff. Our approach is we downloaded all the sequences from a species from a GenBank of that. We took away closely related genomes. We defined the core set of genes. Those are genes that are shared by all the taxa in there. We compute genomic distances. We identified the number of homoplasic sites there are versus new mutational sites in that. And we have a definition of how to apply homoplasy. And we define this one statistic called H over M, which is homoplasies versus mutations for every phylogenetic tree. And so this is the only confusing slide that I'll show today, but this is just basically how we tabulate this. And so here's a cartoon showing that we had a lot of strains, genome sequences. We first sample 4 strains of it, taken at random. We make the phylogenetic tree. We count the homoplasies over mutations. We do that 100 times. We do that for 5 strains. We do that all up to n-2 subsample strains. And you would see a curve that looks something like this. It's finally asymptotes and has an H over M value. It's usually less than 1, but there's a lot of homoplasy going on versus mutations. If there happens to be a second species in there, for example, I'm just showing by the red dot, when you sample 4 strains, you're not going to get it. So the curves are going to sort of be identical for the two. But once you start getting that one strain that doesn't recombine, you're going to see this precipitous drop in the distribution. So we look for these discontinuities, which tells us there's probably more than one species in there. And so we did this for the 93 strains. I hope everyone memorizes this. I won't show it too long. And our results is that the species fall into 3 main categories. We have true biological species, which follow what I've shown you. We have cases that are clonal. That is, things don't recombine at all, so H over M is really low, so we can't classify that as a biological species. And then there's cases like Burkholderia here, where the named species that's in GenBank has sort of this discontinuity in it. We just figure out which strains are from a different species. We take them out, we rerun the analysis, and we're a happy family again. It's now one species, and we have some strains of another group. By doing this, we're able to classify 85% of all named bacteria into species based on the biological species concept. So even though bacteria are asexual, you can apply a biological species concept and apply real species of that. We wanted to see whether this test was robust, so we did it with known sexual organisms. We did it with Drosophila melanogaster, throwing some simulant sequences in there. Again, we get the discontinuity. We take out the simulant and we get the same thing. We've done the same thing with humans and chimpanzees in chromosome 21. When you put the sequences of troglodytes and Homo sapiens together, you find, again, this nice discontinuity. You take out the chimpanzee sequence, you get a nice smooth curve of that. which is nice because we know that humans don't interbreed with chimpanzees and we're separate species. So when we look at that, then we can find out what the limits of diversity there is within a biological species. Most people who look at 16S sequences say that 97% identity constitutes a species. However, we see cases where there's a biological species, so that's where the strains in there differ by 30% in their genome. So using that one strict cutoff is not useful at all and doesn't really apply. Here's Drosophila and Homo sapiens just showing you that we're all very closely related to one another. You differ from the person next to you by about one base pair out of every few thousand. So now that we can define biological species of bacteria, we're combining this with work that's been done by Phil Hugenholtz group, which is the Genome Taxonomy Database. And what they're doing is they're trying to classify things at every other taxonomic level. So are those new phyla? When you find a rare sequence, is it a new phylum, order, class? And they're just normalizing the distance within a taxa. And now we'll be able to define and classify microbes at every taxonomic level. So when you find an unclassifiable sequence, if you have a genomic sequence of that, we'll be able to figure out what it is, whether it's a new species, new phylum, order. So just some general conclusions from what I've told you today and some unresolved issues since we want to think about the future is host phylogeny, that's differences that occurred during the diversification of hominids, is a primary factor by which ape microbiomes are sorted over evolutionary timescales. We know lots of other factors are going to be introduced in the diet, geography, and so on, but there is a large phylogenetic signal to microbiomes. And so we can ask, will this limit our ability to alter microbiomes, knowing that there has to be a particular set of microbes that are in there? Chimpanzees harbor these enterotypes that are analogous to those in humans, and hosts shift among enterotypes over their lifetimes. Are enterotypes just stable microbial complexes, or do they affect the host? Do they have some function at all? Bacteria in several groups have co-diversified with great apes over the past 15 million years. I'd like to know, did bacteria co-evolve with the human populations that I mentioned, and how long and which strains persist? What taxa are co-diversifying, and are they co-evolving? Is there a function to them? Bacteria reproduce clonally. They differ in their propensity for recombination. Therefore, we can define true biological species based on barriers to gene flow. And so, is there a uniform and rational approach to delineate organisms across all domains of life? And finally, don't toss out, ignore, or underreport those unclassifiable sequences. I tend to think that they're some of the most interesting bacteria that are sitting in microbiome samples, and people usually don't report them. And I'm asking, what are the sources, the origins, and the roles of these cryptic lineages? And so finally, I'd just like to give a few acknowledgments in that. The main shout out in the first study was to Mike Worraby. Of course, I'm very, very grateful to Beatrice Hahn, who's been a collaborator throughout most of these studies, and a couple terrific postdocs and graduate students, particularly Patrick Degman, who worked on Gombe chimpanzees, and Andrew Muller, who's now moving to Cornell, has done work on the later chimp microbiome and the co-diversification. And finally, I'd like to thank the generous support from the National Institutes of Health. Thank you very much for your attention. Time for a question or not?
We have
1 minute? Owen.
So we have, at best, kind of an operational definition of what species is. Could I just ask you to speculate or amplify on what are the consequences of that for human microbiome research? Just negative or positive?
Well, the main thing in knowing whether something is a species is whether it can recombine and take new genes in a set. We're finding all of these taxa, at least as far as we're concerned, that are co-diversifying, co-evolved, and we don't even know what they are, whether they are new species or just part of a genus or whatever. Some of them are probably functionally important. So we, of course, we always talk about different taxonomic groups are there and whether they are important. Now we can actually, it's more than just giving a human construct and identification purposes. It's telling you whether genes have been exchanged between a group or not.
Hi,
Dodge,
great
Dodge,
presentation. I think it's really interesting. Can you hear me okay?
Now I can, yeah.
Okay. I think it's really interesting to observe these differences in the composition of the microbiome by different species. Have you given some consideration about how this might affect the translational relevance of mouse model research? Look,
the honest answer to that is I've never given it a thought. But if you give me a second, I haven't thought about it at all, I'm sorry to say. Okay? Yeah, Mike.
Do you think some of the differences, I like what you're doing about the species classification, and the recombination concept is a good one to me, but some of it could also be driven because geographically they're not in the same area. That is, they're capable of recombining, although they're not. You know, you could envision some things being isolated just because they're physically isolated, but if they wind up getting put together, they might start recombining again. And what are your thoughts about that?
Right, so basically, this is the same question you always have. We have squirrels that are on both sides of the Grand Canyon. We have no idea whether they'll ever reproduce. So of course, this is just our definition of how we would define species. We can't set up every potential mating in that. So there are geographic factors in that. We tend to think that in these cases that the things accumulate enough genetic differences through time that eventually these things which are sibling species or sister species will become separate species in that. But it's always a problem in classifying species if you have geographically separated groups, whether they are part of the same or different species in that. We're hoping that bacteria are widespread enough that actually, you know, if they can get worldwide, we can actually see whether there's recombination in each group. And these, we just take all the sequences that are named of that group, so it's based on metabolic character, that are named in GenBank. So some of them are from large geographic areas, but some, again, are from very restricted places. But that will always be a problem in classification of species.
We better cut it off there.
Oh,
Sorry,
I have to cut it off. I'll talk to you later.
So, Howard, will you be around all day?
Of course.
Can you join us in the round table in the afternoon?
Sure.
Thank you, Howard.
Thanks a lot.
I'm going to ask all the speakers for session 1 to join us up here to save a little time. Could you sit here? Because I'll introduce you all at once. Yeah, come on up here. And I'm having a problem, Walter. No one taught us about the timer on the podium or here at the table. If Walter or anybody on the IT team is listening to this. Yeah, I don't think we weren't shown about the timer on here, so I was using my cell phone. Thanks.
No way. I'm going to. Oh, sorry. You didn't hear me. Thanks, Al.
Not responding. I tried to plug it in. Screw with it. Is it even plugged in? Or is that driving this? I see. Thanks for doing this. On. So our first formal session is the Human Microbiome Project, Catalyst for an Emerging Field. And just to save a little time, I asked all the speakers to come up to the table. The session purpose here, and each session has a specific purpose, is to really reflect on the resources, outcomes, and legacy of this 10-year investment in this field. We have the current investigators for phase 2 of the program. We also have Mary Perry, who's in the Common Fund, the office that funded this activity. So I'm going to read all the titles and just call them up one at a time, okay? The first title is the NIH Human Microbiome Project Overview of Human Microbiome Project Goals and Accomplishments, and that talk will be, are you okay? That talk will be delivered by Dr. Mary Ellen Perry, who's in the Office of the Director of the Common Fund Office at NIH. The second talk will be delivered by Greg Buck, who's at Virginia Commonwealth University, and his talk is going to be entitled Impact of the Microbiome of the Female Reproductive Tract on Health and Pregnancy. That'll be followed by a talk by Mike Snyder, I believe this is the correct order, who's at Stanford University, and he'll discuss his project, Integrative Personal Omics, Profiling During Periods of Environmental Stress. And finally, we'll close this session with Curtis Huttenhauer from the Broad Institute, who will talk about characterizing the gut microbial ecosystem for diagnosis and therapy in inflammatory bowel disease. I'll also reintroduce you as you come up. And then remember, the way these sessions are structured, there's a joint Q&A session. Each speaker will call out one critical gap, need, and challenge, and we'll try to address that during the joint Q&A. So Mary, would you? Oh, you know what? I'm sorry, because I didn't turn the blessed page. I'm sorry. The last person, forgive me, is Owen White at the University of Maryland, who's going to talk about management and integration of the IHMP data. Thank you.
Thank you, Lita. It's really a great privilege to be here today to tell you about the goals and accomplishments of this wonderful microbiome program. This was a 10-year investment by the Common Fund, and with the goals to characterize and catalog the microbes that are on and in our healthy human bodies, and also to enable others to investigate the role of the microbiome in health and disease. And as one very indirect indication of the success and the impact of this program, NIH funding from the institutes has gone up about 40-fold since the beginning of the program. This is part of that portfolio analysis that Lita introduced from the TMWG, so thanks for that. Common Fund programs are intended to be short-term catalytic programs with broad and transformative impact. So what was it about the microbiome that convinced the NIH leadership that there should be a microbiome program in 2007? Well, we knew that there were very many human pathogens, and we also knew that they were outnumbered on Earth by more beneficial bacteria. We knew that on our own bodies there are more microbial cells than human cells, and we knew that those microbial cells contained about 100 times as many genes as our bodies do. And we knew that there were indications that these microbes influence our health somehow. But we didn't know what the microbes were that made up a healthy microbiome, and we didn't know how changes in the microbiome influenced our health, or whether they directly influenced our health. But by 2007, we had advances in technologies that would help us answer these questions. Namely, there were faster and cheaper DNA sequencing techniques, allowing very high-depth sequencing of many different samples that could be done, and there were faster and cheaper bioinformatics tools to analyze the complex mixtures of genomes or metagenomes from the body sites. So NIH approved through the Common Fund the HMP to go forward for initial phase funded from FY 2007 to 2012. And the first phase of the program is now known as HMP1. Its goals were to catalog those microbes that inhabit the healthy human body, examine whether changes in that microbiome can be related to changes in health and disease, and to provide a standardized data resource that would enable the community to study the human microbiome. It also, along the way, developed new technical approaches, including computational tools, to help generate the data and analyze the data. The data were released as soon as they were quality controlled to the community for analysis. And the total budget for the first phase was around $180 million, with co-funding from the institutes and centers and other sources of about $30 million, with the Common Fund contributing about $150 million. The program was run as a large consortium of hundreds of members who worked on 6 interactive initiatives, including technology development and computational tool development. And they were coordinated a lot by the Data Analysis Coordinating Center. There's a URL here. You might want to check it out. Oh, as Lita said, Owen White will talk about this later today. And the 2 major investments of time, energy, and budget for this first phase were on a healthy cohort study, which was to answer that question about, is there a core human microbiome that we all share? And the second emphasis was the demonstration projects. There were about 15 of these, whose job it was to investigate changes in the microbiome and that relationship to changes in health status. And you can see that there was a broad array of different types of conditions that were addressed. In 2012, the consortium published a paper collection in Nature and PLOS. And at the time, this was the world's largest collection of metagenomes from the human. And this record will be maintained, as you'll hear. This series of papers had a large impact. One of the indications of that is over 2,000 citations for the collection since 2012, and it has become a community resource for discovery by others. The observation from the group at that time was there are about 360 microbial genes per human gene, so a little bit more than 100 times as many, and for a total of about 800 million unique microbial genes, some of which have unknown functions. Together, if we pooled all our microbes, we'd have about 10,000 species. We don't each carry these, but together we have this collection. These data were deposited in the DAC. This is a screenshot from 2012, where you can see even at that time you could get the data and you could get the tools. This portal is still very popular. In last year, it had over 82,000 visitors from over 175 countries. And those people are using the HMP data to do two different things, two big categories of investigations. One is represented here, where they take the data and use it to develop new analytical tools to mine the data for information. In this case, they assigned some of those unknown genes to specific enzyme types. And in this case, there was a hypothesis-driven research that was aided by the HMP data, where this group discovered new antibiotics. The HMP papers themselves demonstrated a number of important findings, and the one I'm showing here is important to you today because it helps introduce the next phase of the program, which is the one you're going to hear about in the talks in this session. So if you focus on the first panel, this is a representation of the taxonomic assignment, or the phyla, found in several different body sites: the nose, the skin, 3 sites within the mouth, the GI tract represented by data from the feces, and the urogenital tract, which here is the vagina. And you can see that the phyla, represented in the colors, are very different from one site to another. So on our own bodies, we don't have one microbiome all over. It differs from place to place. Also, if you look across the x-axis of individuals, we differ even within our body sites. Our noses aren't all the same in the microbiome that live there. So there isn't a particular poor microbiome, healthy microbiome that we all share. But if we assign the metabolic pathways to those genes encoded by those bacteria, we see much more conservation. The metabolic pathways look quite similar across sites, except for the very specialized vagina, and they look much more consistent across individuals. So this indicated that perhaps it's a function of the microbes that we need to be thinking about more than just an inventory or cataloging of who's there. And that was part of the impetus for the second phase of the program, known as HMP2, or IHMP for Integrative HMP, because they're integrating these data, and we'll tell you about this a little bit. Owen will tell you about it a lot. The goal of this part of the program was to characterize the microbes' functions through doing multi-omic analyses of both host and microbe functions. So genomics, metabolomics, transcriptomics, proteomics, cytokines, phenotypic data, and you'll hear that each group has some specialized data that they've been accumulating. Importantly, each of the studies for the second phase is a longitudinal study to help us make links between changes that could be causal in the microbiome, causal to changes in the disease. Again, the data sets are being disseminated for analysis by the community as soon as they're quality controlled. The Common Fund budget for this phase was about $26 million, and again, aided very much by co-funding from the other institutes, centers, and offices at the NIH. In 2014, this HMP2, or IHMP consortium, published what we call a marker paper, indicating all the different types of data that they're generating and where those data are being deposited. They are also depositing their data at the DAC, so that you can now go and find the HMP1 data or the HMP2 or IHMP data. And Owen will give you an overview of that, as we said. And as Lita introduced, the IHMP2 projects or IHMP projects will be presented by Greg Buck Pregnancy in the Microbiome, Curtis Huntenauer, the Inflammatory Bowel Disease Project, and Mike Stanford will tell you about stress and prediabetes in the microbiome. Throughout the program, the consortium has been very, very interactive with the community, especially the research community. Very early on, one of their charges was to provide a catalog of reference genomes, and so they put it out on a most wanted poster, asking the community, which microbes do you want us to sequence and put in this reference catalog so you can refer back to it later when you do an analysis. They also realized pretty early on that they were generating too much data even for the hundreds of them to analyze, so they reached out to the community and they said, who wants to be part of a data analysis working group called the DOG? And that group not only helped analyze the data, but generate a lot of computational tools that helped to reveal some of the findings that were later published in the paper collection in 2012. This blue line shows Google searches for the microbiome over time, and we'd like to take credit for this bump maybe from the Nature papers. In 2013 and 14, hi guys. In 2013 and 14, Owen White at the DAC collaborated with folks at the NIHGRI and the NIAID here at the NIH to move the microbiome data onto the Amazon clouds to make it available for people without having to download the vast amounts of data. They could analyze it on the cloud. And that is still present for people. In fact, recently, Owen's group at the University of Maryland carried out a training workshop to help people learn how to analyze these data in the cloud. And also, as I had mentioned, the consortium published a marker paper in 2014 to let the community know what they were doing and where to find the data. I don't know, this is kind of weird. I'm going to look at my slides. If you ever want to come up here and walk the slides, maybe we can turn that thing around. Okay, so another indication of the impact and popularity of this program is the page views on the NIH Common Fund website, where we have about 30 programs at any one time, and in 2016 we had 30. But you can see that the HMP got about a quarter of the hits, even though it's only 1 out of 30 programs, and that was about 190,000 page views, so very high interest for this program. Another indication of the impact is the relative citation ratio of the program, which is, the median would be 1, right? So it's all normalized to 1. That 1 would be the relative citation ratio for a microbiome paper in whatever fiscal year you're talking about. So overall, the program has a mean RCR of 7.5. But what's really interesting and very powerful about this analysis is that it shows that there are many papers, maybe about 50 or 60, that have an RCR greater than 14. I mean, they're very highly cited. And these include, of course, the Nature papers that I talked about, which have an RCR of 136 in this analysis, but also several papers on computational tools and technology development. These were very highly impactful from this program, including this one about CHIME that has an RCR of 285. The HMP publications themselves address many, many different disease areas, including some of the ones that you'll hear more about after this talk. And there's yet more to come. This fall and winter, there'll be an update to the HMP1 Healthy cohort studies. There'll be more and more iHMP or HMP2 data for download from the DAC. And there'll be a flagship paper collection coming out from the HMP2 consortium. So my critical gap would be to integrate the HMP data sets, especially the phenotypic data that you'll see there's a lot of coming out of, especially the second phase of the program, with other human phenotypic data. And I want to say thank you to this very productive and happy group. This is a shot from yesterday's, there was the final steering committee meeting for the HMP program, which is kind of sad. I don't know why everybody looked so happy and relieved, but not all of us were happy. I mentioned earlier the co-funding from the ICs. You can see that very many institutes and centers and offices did contribute to this program. I also want to acknowledge the IHMP External Scientific consulting, especially Francis, who's now acting as the chair, and Eugene Chang, Rick Stevens, and Stan Falco. And of course the PIs and their teams who've been really, really interactive and positive and working hard to really carry out all the assignments that we keep piling on to them. And Lita Proctor, the HMP coordinator, right here, very happy in the middle, whose energy and positive attitude are really essential to this program. Without Lita's push and drive, really the second phase of this program, I can say with confidence, would not have happened. Thank you.
Because our timer isn't working, apparently, it's not hooked up, I'm going to use my cell phone. So what I'll do is I'll throw up 5, my hand is 5, if you have 5 minutes left. Okay, thanks, Mary. Up next is Greg Buck, who's going to talk about the impact of the female reproductive tract on health and pregnancy.
Okay, not projecting. I want to add my thanks to Mary and the Common Fund for funding our research over the past couple of years, and NIAID, NICHD, NHGRI, ORWH, and the other institutes that provided funding for our research. I also want to give a shout out to Lita Proctor, who is really honcho this whole HMP1 and HMP2 project, and has helped us get our whole project organized and working together. So what I'm going to do is talk a little bit today about what we've been doing in the HMP2 study. This is a continuation at some level of our HMP1 study on the vaginal microbiome, but now we're focusing on taking a multi-omic approach to look at the impact of the microbiome on pregnancy. The project is called the Multi-Omic Microbiome Study Pregnancy Initiative, or the MOMS PI. Our objective is as defined by the HMP2 RFA, we're taking longitudinal multi-omic strategies to try to elucidate the mechanisms by which the microbiome of the female reproductive tract impact outcomes in pregnancy, with kind of a focus on preterm birth. Why preterm birth? Well, approximately 1 in 10 births in the world are preterm. Preterm is less than 30 weeks, 37 weeks of gestation. This can be higher in other populations, up to 1 in 6. African Americans have higher rates of preterm birth. Annually, over 12 million births in the world are preterm. The mortality rate can be fairly high in some cases. The survivors, as you're probably all aware, have long and very short-term health issues that can be very devastating to the families. Over $26 billion per year is spent in this in the United States for prematurity-associated health care. And so at some level, we know that this is caused by poorly defined bacterial agents. We really don't know exactly how the microbiome impacts on preterm birth and adverse outcomes in pregnancy. We do know that we actually know that complex microbiomes have a negative impact on risk for preterm birth and adverse outcomes in pregnancy, and we're getting more and more data about that every day. So this defines our study, or describes our study to some level. It's a longitudinal study. We eventually recruited 1,594 mothers and their babies, so we had over 3,000 total subjects. It's a longitudinal study, and we are following people from first trimester through third trimester, and then labor and delivery. We're taking a very comprehensive set of samples: cervical, vaginal, rectal, nares, buccal, blood, and we have urine as well. At L&D, we get birth products. We also follow the babies at birth and at discharge, and if they stay in the NICU, we follow them as well. In our project, we've had 10 plus clinical coordinators for over a 3-year period, collecting in all of the clinics at our medical center, as well as in the Global Alliance to Prevent Preterm Birth and Prematurity and Preterm Birth in Seattle. We're collecting samples from University of Washington and Yakima and a couple of other hospitals in Seattle area. We're covering, it was a fairly comprehensive collection program. We were collecting samples 24/7 in L&D for over 3.5 years. This is the result of that process. We collected almost 250,000 samples over this 3.5-year period. The samples that we collected were for different multi-omic approaches that are kind of described here briefly. A total of 231,000. I think this is a little bit of an underestimate of the number of samples we've collected. These samples are stored in freezers at VCU for future use, and we look at it as a gold mine of opportunity for the future. These are the samples that we've analyzed, something on the order of 15,000 for the different multi-omic approaches that we've used. These samples, the data from these samples are going to be stored, as Mary pointed out, at the Data Coordinating Center in University of Maryland, the DCC. Some of our data will go in open access, which means that anybody can have access to it. There's no control at all. This will be hosted at the DCC. Some of our data has to go into controlled access because of the sensitivity. This is a preterm birth study, and you can imagine the metadata that we have associated with this would be very sensitive. So our IRB requires that it goes in a controlled access database. A controlled access database that we're using is the dbGaP, and as you're probably aware, to get access to dbGaP, you have to go through a process to get to have the access. So that's kind of the difference between open access and controlled access. Most of our data will be linked at the DCC, even if it's going to controlled access. There will be pointers at the DCC that will point to where that data is, and will be coordinated in some way, shape, or form. And our other data will go in Metabolomics Workbench for the metabolomics data. These are the data sets that are available. There are 3 focused data sets that are kind of described here. They're early pregnancy, term pregnancy, and preterm birth. This was kind of by the way the project progressed. We started with a cross-sectional study in early preterm and then looked at longitudinal term births, and now we're looking at the preterm births. These are the numbers of samples in each one of the data or the sampling types that we have here, and the omics type of analysis that we're doing. We also have around 15 or 16,000 samples that have actually been processed in kind of a general approach to doing the analysis. We do have a subtext on racial disparities here. We're very interested in racial disparities, and there are racial disparities associated with preterm birth and adverse outcomes in pregnancy. So all of our studies have about 50% or more African Americans that we're looking at for this racial disparities. I probably won't be able to talk much about racial disparities today, but there are 2 posters. Poster 7 on Friday by Myrna Serrano, and I didn't mark the poster down today, but there's another poster today with Jen Fetweiss, who will have a general presentation of some of the data that we're collecting in our analysis here. So there will be some way that you can get access to what's going on in our racial disparities. What have we learned so far? First of all, this is a kind of a general slide that covers most of the multi-omic data that we've been looking at, the 16S profiles, DNA profiles, RNA profiles, and metabolic potential and progression here. I guess the take-home message from this is if you look at 16S, this is data coming from vaginal samples collected in the second trimester in our study here. So it's basically all in the second trimester. The 16S data and the metagenomic data actually very closely reflect each other. There's very little difference in that, which suggests that our 16S analysis is pretty robust. The interesting thing about the transcriptomic analysis is it doesn't exactly reflect the genetic potential that one sees in the metagenomic data. One can see that there's some differences here, particularly in this. We're very interested in this dynamic here. This light brown taxon is BVAB1, which is a bacteria that's associated with bacterial vaginosis, and bacterial vaginosis is associated with higher risk of preterm birth and adverse outcomes in pregnancy. G. vaginalis, this is the red one, Gardnerella vaginalis. This organism also is associated with adverse outcomes in preterm birth. What you can see here is that it looks like the transcription of Gardnerella vaginalis kind of overwhelms the transcription of BVAB1 here. We don't really know what's going on here, but it's suggested that either BVAB1 is relatively quiescent or that Gardnerella vaginalis is expressing its genes at a relatively high rate. This is also reflected in the metabolic potential. This metabolic potential is based on the whole metagenomic DNA sequencing, and the metabolic expression is based on the RNA sequence. And you can see that there's some pathways here that are overexpressed. These pathways are associated with the shunt, what's called the pentose phosphate shunt, that's making ATP for these organisms in an anaerobic environment. This is interesting because Gardnerella vaginalis does not perform glycolysis. It makes its ATP using the pentose phosphate shunt. There's some other differences here as well. For example, this is a couple of these metabolic pathways here associated with synthesis of peptidoglycans and capsular materials. So there are some interesting observations here associated with this is Lactobacillus crispatus here, which is overexpressing these capsular and peptidoglycan materials, and we're looking at, and we think that this is kind of reflective of what's going on in why Lactobacillus crispatus is dominating this microbiome type. We wanted to look at if we can use integrative multiomics to discriminate data that will discriminate clinical symptoms. So we looked at two clinical symptoms. A diverse vaginal microbiome is associated with adverse outcomes in pregnancy, bacterial vaginosis. Vaginal pH is associated with vaginal health. Low pH is considered healthy. High pH is unhealthy. We did a partial least squares discriminant analysis, which is shown here, looking at cytokine, lipidome, and 16S taxonomic profiles. And what you can see here is that it generally does. This is a relatively small n included here, but we can integrate multiomic data, use integrative multiomic data to discriminate some clinical parameters, which suggests that we may be able to use this at some point to predict outcomes that might be helpful for treatment or at least prevention. What happens to the vaginal microbiome during pregnancy? This came from early work in the HMP1, which showed that pregnant women had a much more expanded lactobacillus profile than non-pregnant women, and we wanted to ask the question, does multiomic data provide insight on mechanisms by which the microbiome contributes to adverse pregnancy outcomes? Can we identify early predictors of early preterm birth looking at these kinds of analyses? The first thing is, when does this decrease in diversity occur? This shows first trimester, second trimester, third trimester samples. And what you can see is that diversity decreases by second trimester. So this is happening very early in pregnancy. It involves increased dominance by lactobacillus and decreased presence of complex microbiomes. This is showing that this is focused on vaginal samples. These are diversity Shannon Diversity Index, inverse Simpson. And what it's looking at is the change in diversity in first trimester, second trimester, and third trimester in vaginal, rectal, and buccal samples. And what you can see here is there's a change in diversity in the second trimester in vaginal samples, but the same is not true for rectal samples or buccal samples. So the diversity is localized to the female, is localized to the female reproductive tract. We want to know if multiomic data permit prediction of changes in community state. This is showing different community states associated by the dominant bacteria and tracking longitudinally across pregnancy, first, second, and third trimester. And what you can see is some of these initial community states are relatively stable. Lactobacillus crispatus, inners, gasseri. It's plotted mathematically here, so these on the left-hand side are more significantly more stable or more persistent than these on the right-hand side. So we wanted to say, by looking at these, by looking at multi-omic data, can we predict the changes? So we looked at cytokine profiles. Cytokine profiles, just in brief, if you look at the less healthy community states, for example, G. vaginalis or BVAB1, these are microbiome profiles dominated by less healthy bacteria. It tends to have increased cytokine expression. There is a pattern here. I'm not going to go into that right now, but there is a pattern here that can be used in an analysis of multi-omic data. And Paul Brooks in our group used continuous Markov chain modeling of community state types using this multi-omic data to try to see if we could predict changes in the microbiome and with any kind of accuracy. So what he did, his analysis showed that you can probably predict community state transition. It's dependent on host functions. And in this case, we look at the predictions of change in low IL-5 or high IL-5, just to pull out one of the cytokines that we looked at. And you can see you can predict the probability of transition. In high IL-5, transition from a healthy state with lactobacillus to not change, it's almost 100%, and high IL-5 that will stay healthy. In low IL-5, it's decreased. G. vaginalis, for example, less healthy to another less healthy or lactobacillus inners. In low IL-5, the probability of that change is very low. In high IL-5, the probability is high. So you can use these multi-omic analysis of host factors to predict pregnancy risk, and it's possible that at some point this will have some clinical application. I'm going to skip over the conclusion slide because I think we've kind of covered that already. I wanted to mention some of the posters that we have here today. We have two general posters from Mirna Serrano and Jen Fetweiss. Mirna's is Friday, poster 7, talking about the mom's PI, and today Jen Fetweiss has a poster 24 talking about the preterm birth study, which I really wasn't able to talk too much about today. We have two posters here talking about specific species that we are thinking are smoking guns on what's going on in the vaginal microbiome and how it's impacting negatively pregnancy. And then we have two tool-based posters, one from Tom Aradz, who's using network differential expression and machine learning approaches to try to discover differences between the transcriptomes that we've been looking at. Steve Fung is using constraint-based metabolic modeling to try to define what's going on in the microbiome as well. So those posters are today. There's poster 2 at Friday. Steve's is poster 32. The critical gap that I wanted to mention, and I'll just mention it briefly, is the human genome. I think one of the things I haven't been able to talk about in the short time I've had today is the racial disparities, the racial differences we see in the impact of the microbiome on adverse outcomes in pregnancy. There are very significant differences there. African Americans have a higher probability or higher risk of adverse outcomes, and we've shown that there are clear racial differences. We have not been able to determine whether this is all genetic or environmental. It's obviously both environmental and genetic. We would like to look in more depth at the human genome and in particular epigenomics to determine what those differences are. So I think that's the critical gap that I'd like to mention among others. And this is the team that's worked on this, the Vaginal Microbiome Consortium at VCU, our NIH partners, and our GAPS team in Seattle. So I'll stop there. Thank you.
We now have Mike Snyder from Stanford to talk about integrative personal omics profiling during periods of environmental stress.
Thanks. Okay, well, I'd also like to extend my thanks to Lita. It's really been wonderful being part of this program. It's very exciting. We're now at the very best time. So our project is to analyze a set of pre-diabetics for their profiles over time and through various perturbations. So just by way of background, I think most of you appreciate that diabetes is a growing problem in the world. In the U.S. alone, there are 29 million individuals who are currently diagnosed as diabetic, and there's 86 million that are pre-diabetic. And this has an annual cost of $245 billion each year. It's estimated to affect the economy. What's also equally powerful, if you think about it, is that 90% of individuals who are pre-diabetic don't even know it. So that means that in looking at this room, there's a lot of you out there who are actually spiking your glucose who, in fact, are pre-diabetic and have no idea. I guarantee that's true. We've been profiling lots of people who don't realize that. And why is this significant? Well, a lot of these pre-diabetics will go on to become diabetic in their lifetime. So obviously then there's a strong need for understanding the etiology, the management, and the treatment of both diabetes and certainly to prevent people from becoming diabetic. So again, the objective of our project is to do longitudinal profiling of both microbial and host components for over the now 4-year period that we've been funded. And what we're particularly interested in is profiling people while they're healthy, and then also during periods of time of stress. So there's been quite a few studies, I won't have time to go through this, showing the relationship between diabetes and the microbial composition. And there's some evidence suggesting that stress is associated with type 2 diabetes, and some work from our group saying perhaps viral infections would be contributing to this. So basically, the scheme of our study is we've been profiling, collecting samples from 116 individuals. 66 of these are either pre-diabetic or insulin resistant. And so resistance is a form of glucose misregulation that is often associated with ultimately becoming diabetic. And so we profile these folks over time. We sample them roughly every 3 months while they're healthy. And then when an adverse event comes along, like a respiratory viral infection, we'll typically take 5 more samples through this period. A number of folks have undergone various dietary perturbations, such as weight gain, weight loss, I'll mention that in a minute, and other sorts of dietary studies. And also at other times of stress, for example, 7 individuals, I believe, have undergone a colonoscopy, so we'll do many more samples then. So the bottom line is if there's adverse things going on, well, we'll try and get data from that. So basically, these are the assays we've been doing on the HMP samples. So 105 of these have made it into the folks have made it into the HMP, if you will, analysis pipeline. We've been analyzing 8 different ohms from these. We have very extensive medical testing as well, both of traditional sorts as well as those that are very glucose related. We also have collected other kinds of data I'll mention a little bit later. So the ones in blue are actually host data that we're collecting, and the first number is the number of analytes measured in each of these samples. These are the various samples we're collecting, and the total number of measurements made then is right behind that. And so we're generating, if you will, as you can see, lots of data from each of these sort of data types. So blue is host, green is from both host and microbe, and then the red ones over here are actually microbial assays we've been running. So this is a summary of the sampling. Over this 4-year period, we've collected roughly 1,800 time points with multiple samples at each time point. So over 8,000 samples have been collected and stored in the freezer. We've profiled under the HMP about 990, so close to 1,000 of these time points, and generated roughly 7,000 data sets from 9 different ohms now, and over 13 million measurements have been made. What's very special about this project and why I think it'll be a powerful reference is that nearly all of the individuals, not all, but nearly all have agreed to make their data completely open access in the public database, including their genome sequences. So that means you don't need restricted access to get access to these data, and so all of you can actually look at it. What's special about the microbiome data, or I should say here's a summary of the microbiome data. We've mostly been doing 16S sequencing for budgetary reasons, but we do have some metagenomic shotgun data from some of the critical samples, as well as some metatranscriptomic data as well. So what have we learned? All right, I'm going to go through 5 different things that we've learned. First, everyone's unique, and as I like to say, we're all special and not just to our mothers. This is the example from the microbiome. So Mary's already told you that we all have individual and personal microbiomes, but if you think about it, we've been profiling people now for 3 or 4 years, so we can actually see how their microbiome, is it stable, is it bouncing around? And it turns out our personal microbiomes, for the most part, are quite stable, even over these long periods. So in blue is actually the diversity of samples within an individual, and in red is their comparison to other individuals in the cohort. So for the most part, our personal microbiomes are personal and stable. Although it is interesting, as you get towards this end of the curve, there are people who are as different amongst their sample as they are with others. And so that'll be a very interesting cohort set of folks to look at in more detail. It's not just true for the microbiome. We think this is true for virtually every elm we look at. When we look at healthy time points, what we see is, in general, people's profiles will cluster by them according to themselves, not according to time or things like that. So, for example, this is looking at one participant. This is, if you will, the space of the metabolome. And this individual's healthy samples all cluster together down here. This other individual's samples will cluster over here, and so on and so forth. And we'll see this for other things like cytokines and such as well. So again, we all have personal molecular compositions, and they are fairly stable over time. One thing important from a clinical standpoint is that the insulin-resistant folks are very different from the insulin-sensitive folks, no matter which omic assay we use. So to run you through a little of this is the microbiome. It turns out the sensitive folks will have different strains and subspecies that are different from the resistant folks. This is the metabolomics profile. The same thing's true here. This is the insulin-sensitive folks tend to have these metabolites. Insulin-resistant have those. And we can actually get individual regression analysis, if you will, and individual metabolites that will distinguish these. This one's quite interesting because it actually comes, this is known to already differ in diabetics. What we found is that it's different in insulin-resistant people, and this metabolite actually comes from the microbiome. So a lot of these metabolites are from the microbiome, some are from the host, and some may be from the food. But the point is we can actually distinguish this. And I should say, to do these insulin-resistant assays the way we do it, they're 6-hour tests that cost thousands of dollars, and we can now, we believe, start setting up what should be more predictive tests for probably something on the order of $50 that will only take a blood draw as opposed to an all-day visit from folks once we integrate these sorts of data. One of the interesting things is by integrating different kinds of data, we can see how systems, if you will, biology and chemistry will change amongst different classes of individuals. So that is to say, here's comparing metabolites and the microbiome in both the sensitive and resistant folks, and they behave differently in those two contexts. So this metabolite, for example, will actually show a different correlation with this microbe in insulin-sensitive versus insulin-resistant. So again, depending which environment you're in, what that means is your biochemistry is operating differently between your microbe and your metabolome. So it shows the power of matching clinical phenotypes, we think, with what's going on at a more complex systems level. Likewise, we know that insulin-sensitive and insulin-resistant folks now respond differently to infections. This is our transcriptome RNA-seq data. We believe this is true for some of the other omics assays as well. It's a complicated slide, but in green here are the insulin-sensitive folks. Here are the resistant ones. These first 3 time points, if you will, this is early, middle, late in the viral infection times. This is the resistant ones, early, middle, late. And these are, if you will, molecular pathways. And what you're supposed to appreciate, just look at the fact that the green patterns are very different from the ones in the purple columns, meaning the pathways that are changing in the sensitive folks are different from the resistant folks. So they're not actually responding to infections exactly the same way. There may be technical reasons for some of this. I will also point out that together, neither of these look at all the same as immunization. So a real viral infection is not surprising. A real viral infection has a very different response, we would argue, than simple immunizations, which is what most people study. So we would argue it's very powerful to be studying disease as it's actually occurring. A subset of our folks have done a weight gain, weight loss study, meaning they've actually overeaten for 30 days and then they go on a diet for 60 days. We sample the beginning, middle, and late. About half are insulin resistant, another half are BMI matched, healthy controls. And what we found by doing multiomics analysis is we can actually see the different patterns of molecules and how they'll change as people gain and lose weight. We'll also be able to drill into some of these and see very specifically some of the patterns. And as an example, just if you look down here at this one, this is a case where molecules go up. Again, it's multiomics, it's different kinds of molecules that increase during weight gain and return during weight loss. One of the top pathways here is the inflammation pathway. And the second highest pathway that changes, or probably equally high, is the hypertrophic cardiomyopathy pathway. And it's known, obviously, as you gain weight, you're more sensitive to heart attacks. And we would argue this is a possible molecular explanation for that sort of observation, that these pathways are misregulated. We do know that people will shift their microbiome. This is work from George's lab, looking at microbiome shifts as people go through a viral infection. Your microbiome will shift. But one of the things we found from both our weight gain and we believe the microbiome case as well is that your microbiome will shift during these adverse times, but it actually turns out you still look more like yourself than the person sitting next to you. These personal microbiomes are robust, we would argue, to short-term perturbations. And this shows us here, these are the different microbial compositions. Each color is a different person that's done the weight gain, weight loss. And you'll notice they cluster by person, not by whether they've gained weight or lost weight. So again, your personal microbiome, we would argue, is fairly robust to short-term perturbations. I doubt that that's true for long-term from some of the things that have been mentioned already, but we do believe it's true for short-term. One nice thing we did during this project is that we actually introduced the idea of bringing wearables, these common wearables, for research. And this has been done for tracking activities and such. Okay, we get to see our illustrious panel here. They look very beautiful. If we can somehow bring them back to reality, this will be as much as I enjoy seeing them. Okay, well, let me tell you a little about this. Did I do something wrong here? Okay. Well, anyway, we've started bringing wearables. If you want to see, I'm wearing about 8 of these things right now. These we think are very powerful for this kind of research, longitudinal research. Okay, we're almost there. Because they collect measurements continuously, 250,000 measurements a day. We've done many things with them, but I can tell you one thing that stood out from our study is that we can pick up, when people are picking up infectious diseases, very early in the process. So, for example, this is showing the acquisition of Lyme disease from one person you're staring at right now, which is you can actually pick this up by outlying heart rate measurements as well as outlying skin temperature measurements. They will show up very early in this process. This is a daily measurement scheme, and it turns out every time for this particular individual he's gotten sick, he gets these outlying heart rate and skin temperature measurements. We've gone on and looked at other subjects because they're incredibly well medically phenotyped, and sure enough, we can actually pick this delta heart rate, if you will, very early. We think before it's showing strong symptoms, if any symptoms. We think we can pick this at the earliest times of infection. This came out of the course of our study from this project. We also think there are physiological differences between sensitive and insulin-resistant folks as well. I won't have time to get into this. This is published, so you can take a look at it. We also think that this type of project is a great precursor, if you will, for the precision medicine initiative, as well as other wellness initiatives that have now started. Because as you do these deep profiles on people, you will learn things that are very, very important for their health. And I'll give you one example. There are actually many examples, but from the first 70 folks we've analyzed, we've gone over their detail in incredible detail, their genome in incredible detail, and 12 of them have information in their genome they should know about. It affects them when they go to a doctor's office. There will be a test. related to this. And one good example that many of you are familiar with, one individual has a BRCA mutation, putting them at high risk for breast cancer. Another individual turns out has a mutation in a heart functioning gene, and sure enough, this person actually has a heart issue that got exposed in follow-up tests, and he's now on drugs because of this. So this is the sort of thing you can learn. It's also fun to reflect on the fact of where we have gone through the Human Microbiome Project over the years. Started out even before it existed with Sanger sequencing, then moved to 454 and other types of sequence, and now is getting more sophisticated. I know Curtis will mention this, but the algorithms that have been developed over this period have gotten better and better for being able to do all sorts of things. And obviously in this last phase, we've been able to bring in multi-omics analyses as well as long-read sequencing. This is just a summary of some of the algorithms that have been generated from our groups and approaches. Again, I won't have time to do this, but you can certainly visit some of the folks at Posters and learn about the algorithms they've done. And I would argue these long-read sequences, we've been involved in this as well as others, have been very fruitful for being able to pull out subspecies information and even assembling some level of metagenome sequences right from whole genome sequences from these complex metagenomes as well. You can get this if you get long enough long reads. What are some of the microbiome challenges? Well, I think a big challenge out there is what's cause and effect. We see all these associations with biological phenotypes, but we don't know what's causing what's coming along for the ride. And I think we need methods for being able to unravel that. And I would argue a related challenge is setting up predictive models for using omic information for predicting health states and such. And I think that can easily be generated from this. These are some of the posters that come from our group. Again, this is a joint project with George Weinstock's group and our group. And so there's one poster today and then 4 posters on Friday in this area. Again, these are the folks who actually did the work. When You Shall, Reza Solani, and others are listed here. Because I'm over time, I won't go through everyone, but we have wonderful collaborations with Tracy McLaughlin, and again, George Weinstock, who's an equal collaborator in this whole project, has been an important part of this, and again, thanks to NIH for supporting this. Thanks.
Next up, we have Curtis Huttenhauer from the Broad Institute and the Harvard School of Public Health, characterizing the gut and microbial ecosystem for diagnosis and therapy in inflammatory bowel disease. So the yellow flashing, Curtis, will be 5 minutes.
That's when I get electrocuted?
Yeah, 5 minutes left.
That's when it turns red. Okay. Huh? I get electrocuted when it turns red, not yellow. Okay. Excellent. Many thanks, Lita, for organizing the rest of that doesn't sound good. Try again. I'll step forward very carefully. Thanks again for organizing, everyone for attending, and I especially want to thank my COPIA and the HMP2, Rom Nick Xavier, and the entire team, many of whom are here, and we'll see at the end we'll have posters on parts of the work. So our Human Microbiome Project is on inflammatory bowel disease, which is, of course, as many people here know, both a critical health condition in and of itself and has become a really interesting model for the complex involvement of the gut microbiome specifically in chronic disease. And especially for about the past 5 to 10 years now, some better studies of the microbiome have helped to very quickly refine our understanding of this involvement from a high-level observation of ecological disruptions, reductions in diversity in the gut community during inflammatory bowel disease, down to a much better understanding of specific taxa and their common metabolic and other molecular functions that are disrupted in subsets of inflammatory bowel disease, including the 2 main subsets, Crohn's disease and ulcerative colitis, and additional heterogeneity within those 2 subtypes. So going into the HMP2 a few years ago now, we were interested in better understanding the mechanisms driving this disruption, what's causal versus responsive in the microbiome during inflammatory bowel disease, changes in disease activity, and specifically helping to translate some of the emerging ecological understanding of the microbiome's involvement into activities that would help in the clinic. And some of this might involve, for example, understanding the heterogeneity of the disease. Again, even within the two main subtypes, there's a great deal of clinical differences in disease progression and in phenotypes like response to treatment. So we can take a look at linking those two changes in the microbiome, helping to, say, predict response to treatment over time, predicting either the onset or remission of flares in disease activity. Both Crohn's and colitis can proceed in bouts of greater or lesser disease activity, and then identifying either on the host or the microbial side pathways, molecular targets that might be either causal or points of intervention for new treatments. So over the course of the past 3 to 4 years now, we've followed a cohort that ended up being just over 100 individuals drawn from Crohn's, colitis, and control populations over, in total, a set of 5 cohorts at 4 different physical locations. And this represents a mixture of adult and pediatric IBD patients and controls, recent onset and established disease patients. And then we've ultimately collected about 4,000 total specimens from which we've generated data for just over 2,000, drawn mainly from stool samples that were contributed by participants biweekly for the course of 1 year each. These mostly give us data about the gut microbiome, targeting microbial information primarily through sequencing, but then in addition to more functionally targeted data types such as metabolomics, proteomics, and then as we go into the biopsies and blood draws that were taken at baseline, these help to tell us about host molecular information during IBD, including host transcriptomic sequencing, genome sequencing, epigenetics, and then serological profiles from a small number of longitudinal blood draws. We have these roughly quarterly in addition to baseline for the biopsies. So you can see a profile for these samples of roughly what we targeted over the course of 1 year each per individual as far as time points and data types. And when we compare this targeted longitudinal profile to the data that we have ultimately generated for these sample set, we're pretty close. We have a collection across the 3 targeted phenotypes of a combination of subjects with long, dense time courses over 1 year each, in addition to most data types drawn from most subjects at these series of 6 or 7 targeted anchor time points from across the year, particularly dense during baseline when we're getting the bulk of the information about each patient. So we've been making both pilot data sets and now the whole data set available as it becomes generated through the IBD Multiomics database or ibdmdb.org site, in addition to the DCC for the HMP2 and public repositories. So the IBD-MDB now includes a series of pilot data sets that were generated over the course of the whole project. There's several, not quite all yet, but several of these large final data sets for the whole cohort, and then other documents that we've been able to provide as we go along, including the sample collection protocol, the kits that we've used to collect these samples for multi-omics analysis, and then some of the data generation protocols as well that help to derive these different types of data from several different sample types. Particularly taking a look at several of the pilot data sets that we've generated over the course of the project, we've had a little bit of time to start looking into the biology of different types of multi-omics as they affect the microbiome and inflammatory bowel disease. So I'll highlight a couple of examples from these studies. And anytime you see a face on these slides, such as Melanie Schirmer, a postdoc with the group who worked on this particular project, starting with a pilot data set that specifically compared the gut metatranscriptome to the gut metagenome over time in inflammatory bowel disease. So one of the things that Melanie looked at was identifying cases in which the metatranscriptome was particularly informative with respect to disease progression. So I pulled one example here that's a mucus degradation pathway that across the cohort, this pathway is expressed, so these are metatranscriptome samples, particularly by one species of Bacteroides, B. vulgatus. This is mostly cross-sectional data at baseline, and when we pull out time courses for individual patients, sometimes you can see that B. vulgatus is the unique contributor of this pathway in this particular individual's gut microbiome over time, both in the metagenome and the metatranscriptome. So this bug is really carrying most of this particular subset of mucus degradation for this individual. But this is in contrast to other individuals where even when at the metagenomic level, B. vulgatus is the main DNA contributor, there are very low levels of other organisms that over time can sweep into transcribing this particular pathway. And often, although not always, this corresponds to changes in disease activity as well. So here you can see over our roughly every 2-week time points, mostly different Bacteroides in addition to some unclassified organisms coming in and out of transcribing this pathway, even when DNA levels don't necessarily change. And this represents the behavior of a subset of pathways in the gut metatranscriptome. Some seem to be more stably transcribed over time, but others can switch between microbes, particularly during disease activity. In general, when we zoom out and ask about some general principles of the gut metatranscriptome, one of the things that I thought was interesting was a trade-off between how many organisms can contribute a pathway versus how hard they seem to work to do it or how over-transcribed processes are. So in general, if we summarize the diversity of organisms that can contribute a pathway metagenomically versus those that do transcribe it metatranscriptomically, you'll see first, fortunately, more organisms tend to carry pathways metagenomically than are actually transcribing them, which is what you'd expect. But then also, for pathways that are particularly unique to just one or a few organisms, these tend to be overexpressed. So if you, as an organism in the gut microbiome, occupy some particular transcriptional or metabolic niche, you tend to be working uniquely at that process. And this is one example of that. So one of our other pilot data sets that was mostly worked on by Hera Vlamikas and Eric Francosa compared, instead of metatranscriptomes, the gut metabolome to the metagenome. And of course, this allows us to pick out, for example, specific small molecule compounds that are enriched or depleted during inflammatory bowel disease, mainly paralleling each other in Crohn's and ulcerative colitis, but then also to start relating these to specific microbes that are, in this case, co-varying with those metabolites. So here you can see a list of the individual organisms that are enriched or depleted during IBD in the gut, individual small molecule compounds, and then significant covariation between those that starts to suggest potential mechanisms. So these are covariations above and beyond what you'd expect just from the disease itself, such that each of these microbes might be generating a compound that they covary with, might be dependent on that compound, or might interact ecologically with another organism that generates or depends on that compound. And you can see for specific organisms and small molecules, many of these correlations are strong enough that they've driven us to dive into the metagenomes and then genomes of these bugs in a little more detail to do some genome gazing and figure out what might be mechanistically causing this. And I'll have an example for that for Rheumacoccus navis in a few slides from now. I want to spend a few slides between now and then, though, on what I think of as the other HMP2, or what I'll refer to as the HMP1/2, a second wave of specifically metagenomic data that was recently generated and analyzed from the healthy cohort in the HMP1. And I don't need to describe the population too much. Mary introduced the background of the HMP1 data. What we've done in HMP1/2 is focus on an additional several thousand metagenomes that were generated from samples collected not just in the gut, but across the body for a subset of 100 individuals from the original HMP1 healthy population over all 3 time points each. So along with Owen and Anoop and others at the DCC, Jason Lloyd Price and a group in my lab have been working on analyzing these additional metagenomes to get additional insights into microbial function across the body in the absence of perturbations from disease. And this has led to, of course, not just more data, but a series of additional, more recent tools, analysis techniques, strain profiling, fungal and viral profiling. Owen will describe some of these later, and Mary already mentioned that the DCC has specifically loaded these data onto the cloud for easier analysis. I can personally say that was a huge boon during both the HMP1/2 and analysis of the HMP2 data as well. The couple of things that I want to call out from the HMP1/2 revolve around our ability to now look a little more closely at what Mary introduced as the potential functional core of the human microbiome. So by providing more specific, taxon-specific functional profiling across the body, we've been able to pull out a series of pathways that are human enriched, or I think of them as host-associated microbial community-enriched pathways. So these are examples such as vitamin B metabolism that are present essentially across the body, not just in the gut, not just in one body area, but in several body areas, typically carried by many different organisms, both across body sites and within body sites between individuals, that are enriched in the HMP1/2 metagenomes and specifically not present in environmental isolate genomes. So these are pathways that seem to be contributing functionality to a microbial ecology that's specifically necessary to survive in association with a host, regardless of what your exact biochemical environment is. This is in contrast to a more refined set of site-specific microbial processes that we've generated during the HMP1/2 as well, such as this example of nitrate reduction in the oral community. So again, this occurs throughout the 3 different oral body sites that were metagenomically sequenced here, different microbes providing this process across specific oral sites and across individuals within the site. But this is a pathway that seems to be universally present among microbial communities in the oral cavity. We've found it useful to think of this type of functional diversity in ecological terms specifically for contributional alpha and beta diversity. That is, which microbes can provide at the metagenomic level or do provide at the metatranscriptomic level a particular process in a particular environment. Sometimes you'll find processes, and these are metagenomic examples, that are provided across a particular body site uniquely by an individual organism, demonstrating both low contributional alpha diversity and low beta diversity. This is the same organism providing this particular pathway across many different gut environments. We can find pathways with low contributional alpha diversity but high contributional beta diversity. Only a few bugs providing that pathway per individual, but different bugs between individuals. And then, of course, both of the other 2 combinations are common as well. Pathways with high contributional alpha diversity but low contributional beta diversity. A group of different bugs can do this, and it's roughly the same group of bugs between individuals. And then perhaps the most common case for metagenomes in particular is high contributional alpha and beta diversity. A bunch of different bugs can carry out a particular process, and different bugs show up in different people. So I'm about to get electrocuted. I can see the red light getting close here. So I'll wrap up with the example that I promised earlier, linking together a few of the strain level analyses, functional analyses, and metabolic analyses that I call out over the course of the talk, specifically in a study that was done in a subset of particularly long, dense longitudinal time courses from another pilot data set from the HMP2 in this case. So this is a small subset of IBD, Crohn's and colitis patients and control individuals in which 2 postdocs with the group, Brantley and Moran, started to look at grouping together taxonomic enrichments by functional traits, so combining the microbial species and microbial functional enrichments that we've seen in inflammatory bowel disease before. And something that they noticed is that one of the most enriched functional traits among microbes, so pathways that are broadly distributed, during IBD turned out to be facultative anaerobicity. And so you can see this in our pilot data set by looking for an enrichment of any microbe that can be labeled as facultative anaerobic. And this is true across comparison data sets, either using controls from the HMP1 population or using another IBD study published from Penn a couple of years ago now. This represents a group of specific organisms, most of which are not individually statistically significant, and one of which is Ruminococcus navis that I called out earlier. So Ruminococcus navis tends to be somewhat enriched individually within IBD, and we can pin this down to a group of genes associated with a group of specific Ruminococcus navis strains that are only ever present within subsets of our inflammatory bowel disease patients. And this is based on one of a couple different types of metagenomic strain profiling tools we've developed that specifically calls out which genes are unique to this subset of strains. You'll see most of these are not characterized, but we can identify a few that are involved in processes like iron acquisition, or particularly oxidative stress, or mucus utilization like I had mentioned earlier. this leads me to my one last slide, which is, I think, one of the biggest gaps in some of what we're interested in analyzing in the microbiome is this lack of functional characterization in underlying microbial genes. So there are many cases in which, and I assume this is true for most of the audience here, we've traced down a phenotype of interest to a particular set of microbes or microbial genes. We don't know anything about what they do. So there's a big opportunity in terms of providing both biochemical and computational support to better characterize these subsets of interesting-looking microbiome-associated genes. The other gap I'll call out is technical. Again, as many folks here know, we recently completed the first step in the Microbiome Quality Control Project, or MVQC, to help pin down at the opposite end of microbiome scale, technical variation in large population studies of the microbiome. And of course, I definitely don't have time to get into that today, but one of the things that the MVQC found is that there really is a surprising amount of protocol variation among labs that can contribute to batch effects and differences in data generation at the very large scale end of microbiome epidemiology. And if we think of this as an opportunity, this provides an opportunity to generate a better characterized menu of experimental design choices that investigators can use to minimize these unwanted technical differences. So I'll wrap up with that. I again want to thank Pramnik and the rest of the HMP2 team, Owen and the DCC, along with everyone from the HMP1, and call out the collection of posters that folks from the group have here, both from the HMP projects and from other projects in the lab. Thanks again.
And our last speaker for this session, Owen White, University of Maryland School of Medicine, Management and Integration of the iIntegrative HMP data. And it'll flash yellow at me.
Okay. All right, there's my timer. And I want a laser pointer, I think. Okay. So I'm Owen White, and I'm from the University of Maryland in Baltimore. And if you don't know about Baltimore, we have a mayor who hired a work crew last night and took down all our statues. And as somebody who lives really close to the statues, I'm delighted. All right, so Curtis just introduced the Healthy Human Subject Study, and I won't talk too much about the results of the study as much as to try to just impress people that it's a lot of data. In this figure here, there's shaded in different levels of gray, an indicator for all these histograms. If it's darker, it means that we have that many samples in terms of time points. And if it's this color, bluish or reddish, that's an indicator of what was published in 2012 in the first wave for the healthy human subjects and what will be published very soon in Nature. So what I'm trying to show you here is that basically we have a lot more samples as well as a whole lot more technical replicates of the data that essentially represents a tripling of that information. Just as a sort of quality control point of view, we performed a lot of different types of analysis. Curtis introduced that very well. But if there were any concerns about how that data behaved during the course of the project, there shouldn't be. You can't really see it, but these are color-coded by body site, and their shape is associated with a circular or square, depending on what part of the project it came from, and you can see a very nice intermingling of the circles and squares, so essentially all the data across the entire project is behaving very nicely. Do want to call your attention to Jason's poster that will present a lot of the results that Curtis showed, as well as some others, which is a very exciting project that was a collaboration with Curtis and my group, as well as Rob Knight, and represented an enormous amount of computing time in terms of the amount of assemblies and annotation that we had to do, so we're quite proud of that data. You've already seen these presentations from the 3 IHMP studies, and what I want to do is just motivate you to think a little bit about the very rich clinical data that was associated with this, everything, including the subjects that we had, the time points. There's lots of primary data, there's lots of derived data, and there's a problem with this that I just want to make people aware of, and I think it's important to highlight. So the best way to do that is to show you an old spreadsheet that was generated by Steve Sherry at dbGaP. And this is, each one of these columns is representative of an early HMP demonstration project where there were different investigators that were funded to demonstrate that there may be some causal relationship between microbiome and disease. And so for each of these studies, they collected lots of different types of clinical information, and I know it's hard to read, but things like drug, things like blood pressure, things like the weight of the patient. And what Steve Sherry did was mark using different colors if information like weight had been collected, and also looked at if the variables for those different studies were really comparable across the studies. And it was kind of mind-blowing to discover that typically in clinical studies, I mean, my background is much more in genomics, not with projects such as this, that things as simple as blood pressure or medication or smoking history are all collected in different ways. And that makes finding all data sets that are associated with, let's say, a blood pressure range for patients that are male nearly impossible unless you kind of sit somebody down to slog through the data and harmonize it across it to pull that information together. And so what I'm going to identify as a very significant gap in the field as you start to think about all the presentations that you're going to have and that you've already had is that we're collecting clinical data, and it's very hard to sort of reproduce what that information is, and also, more importantly, kind of retrieve robustly across all those projects to get information that you could then sort of reuse for your own research. So I don't have a slide for this, but right now there's over 10,000 samples at SRA for human stool, microbiome stool samples, and there's virtually no way to robustly pull all that data down, and there's virtually no way to identify which of those samples came from, for example, diseased patients that were part of Curtis's study. That's not a defect of SRA. They're doing the best job they can to be collecting that information, but there's just not one uniform way that data is deposited into a database. So let me just show you how we approached that for this study, and let's hope or imagine that as microbiome research kind of metastasizes into all the different NIH institutes, that they start thinking about issues like this. So the way that we did this was to stand up a data management working group from all the IHMP projects, where this kind of represented some people who were data managers that were embedded at the labs, as well as partnering with the Data Coordinating Center. And we did something as sort of silly or administratively simple as this. This is a Google spreadsheet that represents lots of different broad data types, like something like a project or a study or a subject. And you can imagine for a subject, there's lots of phenotypic information that you would collect. And each one of these projects, or these columns here, represent the 3 different projects that you just had presented. And I'll call each one of those data types a node. And what you could do for our purposes was to just sort of click on a node and generate a document that lists all the specific fields that were associated with something like a subject. And what we did was we identified the common data elements. An example could be blood pressure, or it could be gender, or these things of that type, and identify either by creating our own control vocabularies to describe that information, or identify internationally recognized control vocabularies to describe that information that would be used across the project. And this is a pretty simple administrative tool for being able to do that, but it's essentially kind of a breakthrough idea with respect to being able to interoperate and share data robustly across projects. And The landscape for NIH data is going to kind of explode even larger than it is now, and I hope people engage in projects such as this. This is not a novel idea, but it's a very important one. So then what we ended up with were these different submitters, and we have one central database that can receive documents known as JSON objects, and once you've got a reasonable way of describing data and attaching keywords to that information, it's pretty straightforward to write software that handles submitting that information into a common database. And if you're interested in that, come take a look at poster 28, which I believe is on Friday, for our system called the Open Science Data Framework. And you can deposit in what are basically machine-readable documents to be able to capture that information. And we had validators that would review, electronic validators that would review that information to make sure that it was being marked up correctly. And it was kind of an iterative process. There was no day when we kind of pulled the trigger and said, okay, this is the way that we're going to do it. It was sort of highly iterative over the course of the project. So that's the way that the data was deposited into the system. Then Curtis's group was responsible for making something called Anadama, which is a data analysis management system. And this has lots of different features to it in addition to what I'm going to describe. But it allows you to make reproducible workflows and handle different dependencies. So if a data set changes in terms of the data that you're searching against, it sort of updates all the information as part of that chain. You can also parallelize information. But we adopted it mostly for its ability to use as a submission tool. So what I just described to you is the data harmonization process that happened across the 3 IHMP projects. And you can just sort of imagine expanding the number of people that are participating in this and having a group of people that are sitting in a smoke-filled room and kind of handling agreement of finding the common data elements. It goes into a common database. And then what we're using Anadama to do is that each group has access to the software that handles the submission of all that regularized data that's in the database into the external archives. And the advantage of this is that the archives, I love them, this is not a criticism of them, but they're overly flexible in terms of the way that they could receive information as simple as, let's describe the forward and reverse primers that were used for 16S experiment. And so at least across our project, there was a lot of uniformity in terms of the way that data was being submitted, which is a good thing. So again, think about all the omic data that we have. How would you go about finding that you've got proteomic data, metabolomic data, and 16S data that came from the same project? Well, you would only be able to do that if they all contain the same project ID. So what we try to do is, make sure that same project ID traveled with all the information. Okay, we have created a new website, and if you're interested in the details about it, please go to poster 30. It contains all the information for the healthy human subjects as well as the IHMP. One of the things that we're very proud of is that we've been capturing lots of protocol information, and we'll continue to have all the protocols from the healthy human subjects as well as the IHMP. We're also very proud of an interface which is a very responsive portal that allows you to click on lots of elements, just as I described, things like the gender of the patient, et cetera, and see really rapidly displayed pie charts for all that information. And you could select on the pie charts themselves, and it would pass you to a listing of each of the files, and then you could pass those files to a manifest. And that manifest, we have tools that allow you to then download that data directly to your own computer or use it in other contexts. And if you're interested in some of that workflow about how you could kind of do that, have a look at Jonathan's poster 31. All right, let me just identify one last gap that I think is going to be a very serious concern, is our ability to just analyze the data in reasonable time. When you start thinking about the volume of this data, something's got to happen in terms of enabling people to do this. And we had a pilot study, and I'm very grateful to the Common Fund and other NIH institutes for helping to support this, where we recently held a training workshop to analyze all the data that I just showed you on Amazon. We invited a number of luminaries in the field to present their analysis tools, and we bundled all the analysis tools into one common system that could be launched on Amazon, and then for a day and a half, we walked users through the process of doing this analysis. This was really exciting for a number of different reasons. The developers got to have kind of direct interactions with the users. The users got a chance to have the developer right over their shoulder, helping them with how to perform analysis. All the tools were placed into one common executable image, referred to now as a Docker, which is available to people. Here's all the analysis tools. And this was just a very exciting way of sort of demonstrating this sort of bold new future for how you could do analysis on the cloud. So that was really very nice. I'm going to wrap it up. I really very strongly want to thank two senior personnel, Michelle Giglio and Anoop Mahurkar, who were basically the people that really led tactical and strategical development of all aspects of our project. Here's the people who actually did the work and their posters are here and represent different dimensions of the whole overall project. And I'm very grateful to all of them. We're really kind of wrapping up almost 9 years of the HMP project overall, so this is kind of an exciting time for us, but you know, these people really made it happen in very exciting ways. And I also want to thank the Data Management Working Group for really making a robust set of data, and I strongly encourage people to think about some of the stuff that I'm thinking about with respect, or presented with respect to metadata management, because I think it's going to be a big deal in the future. And here's my sort of summary slide. Rob is here, and I'm really very grateful to him, as well as the original sequencing centers that generated the HMP data and our sources of funding. Thank you.
So now what we're going to try is a joint QA session. Just as a point of interest here, Walter and others who are running in the background, I guess the webcast is either not working and only working on certain browsers. So if you folks, whoever's in the ether here trying to run the webcast, if you could somehow double check that over our lunch break, it'd be very good. Thanks. I'm going to ask a softball question. And one of you or any of you can answer. The real motivation behind phase 2, as Mary said, was to ask the question, what other omic properties, what other biological properties of the microbiome do we need to collect to really get insight into the microbiome in the human system? And so even though we worked on preterm birth, IBD, and type 2 diabetes, those three studies were meant to be more generalizable, that there could be lessons for the larger community. So could some of you speak to what you thought was the biggest insight you got when you started to collect these other omic properties and how that might be important to this audience? Go ahead. And here, let me pass the mic so all you have mics.
Well, my first impression in the answer to that question is that looking at the host properties, starting to look, one of the big questions, as somebody's alluded to, several people have alluded to today, is taking the associations that were kind of identified in the initial microbiome studies and trying to look at what could be operationally understood from them. What is the mechanism? What are the causes? What are the etiologies of the diseases or the health conditions that we're looking at? And I think by starting to look at the host, for example, the metabolome, the cytokine profile or immunoproteome profile, and how that correlates with the microbiome community state or what bacteria are present, I think starts to give you some kind of an idea what's actually going on in the interaction between the host and the microbiome, and start to understand how that microbiome is affecting the host and possibly affecting diseases. That leads to hypotheses about what that one can then test by looking at individual bacterial strains or species to ask the question, are those impacts really being caused by that bacterial strain or group of bacteria? So I think the host-parasite interaction or host-bacterial intersection is really important, and by getting a multi-omic approach, we can start asking those questions about host.
There we go. I mean, I would concur. You really do need the host information to get the complete picture. And many of the observations we observe are contextual. And I think all of us had examples of this. So, for example, in our case, how the metabolites and the microbiome behave depends whether you're insulin sensitive or resistant in certain cases. So you would completely lack that understanding if you weren't collecting that information. So, and I think that's going to be absolutely critical for understanding how disease comes to be and also how to manage disease. I think we're going to need that kind of information to be able to implement it ultimately in the clinic.
So I'll definitely echo both what Mike and Greg said with respect to host properties. That's a place that we haven't gotten an opportunity to look closely at yet. And what Owen said, thank you, during his talk regarding the metadata. It's easy to think of host phenotype and environment as an afterthought, but having that in the project up front in an organized way helps you understand disease. From the HMP2 in particular, I think one of the biggest lessons that we've gotten has been that there's no one omics silver bullet. The HMP2 has been an opportunity to take IBD and the microbiome and really slice it in many different ways. And what we're seeing here, and I think in other phenotypes, is very much like what you'd expect in a single organism, that you're never going to see the mechanism for every phenotype using just one tool, not 16S, not shotgun sequencing. And now we've had an opportunity to use many additional molecular tools, both for the microbiome and the host in IBD. And they've all shown interesting properties under different conditions or for different questions.
I'm going to keep on asking questions, but if there are questions from the audience, yes, please go ahead.
Hi, the question is about metabolite data.
You know what? It might be valuable for you to go ahead and announce, I think, because people are listening, your name and
announce my name?
Yeah.
Hi, I'm Katie Pollard.
Yeah.
And my question is about metabolite data. It's for Curtis and Mike, I guess, but anyone's welcome to chip in. It's really cool to see metabolites sort of going up and down and to see taxonomic groups and genes going up and down in association. But this is a question about the future. Beyond the data you've already collected, what are the prospects for actually resolving metabolite changes to specific microbes, hosts, et cetera?
Before you leave the mic, what do you mean by resolving?
If I want to understand the mechanisms and just to know, for example, if a metabolite, I mean, just to be really specific, if there's a metabolite that is healthy or unhealthy, can I figure out what microbe is contributing it? Is that one microbe or a collection of microbes? Is it a microbe and a host enzyme that you need? I'm sort of trying to understand the pathways behind that and the mechanisms.
So I'm going to selfishly start with a response tied back to one of my gaps. I think one of the biggest challenges there is just lacking a lot of molecular level annotations and information for typical bugs that we see in the gut or in the human microbiome. Even someplace well studied like the gut, the majority of gene families associated with metagenomes there, as you know, are not biochemically or functionally characterized. So we can look at things like covariation and find a lot of really nice, strong covariation specifically between small molecule compounds and either taxa or gene families. And right now, that's about as far as it goes until there's more infrastructure for isolating microbes, doing the tough biochemistry to characterize what specific gene families are doing, and then cataloging that in a systematic way. And of course, contributing to that computationally with predictions and computational infrastructure for function characterization as well.
I think I would amplify on that is that even if you take a simpler system, which is just the human genome, there's not a close relationship between discovery that a metabolite is going up or down and knowing specifically what pathway that may be associated with. And even that is sort of a non-simple problem, and then map it to bacteria where there's an enormous number of uncharacterized genes or duplicated genes or whatever. It makes it a very complex problem.
Yeah, I totally agree. I was wondering maybe if we could think beyond sequencing to other technologies like imaging or other things you might be able to do with mass spec besides the classic things. I don't know, Mike, I know you use a lot of technology. Do you have any thoughts about other approaches?
Add to what they're saying. I mean, I think we'll be not far from making these associations. I think the power of the HMP, too, is that we will have these data. It's going to be pretty massive, and actually to be able to see some of these co-associations across different data types, in my mind, will be very powerful. But that is only step one. It's the co-association. And to me, what will be extremely powerful will be the perturbations you do on top of that. Probiotics people take to see how that affects things, as well as drugs people take that will perturb one particular pathway or one aspect of that, and then you'll see how those co-associations change. I think that will be very, these are humans. We can look at natural genetic variants to see how that things, but we're not mice. We're not going to go in and CRISPR ourselves, except maybe in iPS cells. But point is, will be, I think those perturbations will be very powerful for getting at some of the mechanistic aspects you raised. As far as bringing in other technologies, I mean, I do think they are powerful. I haven't done much imaging with microbes, a little bit with scanning EM, so I'm not as sure. I do believe that disease tissue imaging is very, very powerful, and we're doing a lot of this now with cancer and other diseases, to take image information and co-associate it with different omics patterns, and that's been quite interesting and revealing, both for some mechanistic understanding as well as for prognostic value. So I do think imaging will be extremely powerful. But I do think the longitudinal and, again, perturbation experiments will be the most compelling in my mind for unraveling these associations.
Just a quick follow-up comment. I think one of the goals of the HMP2 was to develop tools to address these exact questions that you're asking. It's not simply new kinds of biological tools, but some of the efforts that's going into is developing computational tools, and I know that Mike and Curtis and Owen's groups have done that. We've done a little bit of that too. There's a couple of posters here that I mentioned today where some computational biologists in our team are trying to develop new ways to use machine learning and network analysis to look at these associations, and still you maybe only get into a hypothesis, but it brings us try to bring us closer to getting a hypothesis that we can test biologically more efficiently rather than trying to do wild goose chases.
Hi, Amir Zarampar, UC San Diego. So the thing that I struggle with when I'm investigating or thinking about human trials or investigating microbiome in humans is that we're trying to understand what's going on in different parts of the gut by just looking at the stool or looking at metabolites in the stool. How can we really understand that physiology when, for example, when we look at mice, we know that the stool is quite different than what we see in the terminal ileum or in the cecum or any other part of the lumen? And can we really understand physiology by looking at the stool, or should we just use it as a biomarker as opposed to something else?
So I guess I'll start, since we've looked at that a little bit, mostly in non-human systems, both comparing stool in the human populations we've worked with to biopsy 16S profiles from different locations. That's typically cross-sectional, and then in primate models doing a more longitudinal within subject biopsies across the large and small bowel and in stool. I don't know as much about what the biogeography might be like in rodents. In the primate model, the large differences were mostly due, between stool and other sites, were mostly due to a small number of taxa. So that for the majority of taxa, there was a direct linear quantitative relationship between what you see in stool and what you see at the mucosal surface, with a small number of exceptions that were very different, thus leading to an apparently large overall difference. That being said, there are clearly biological differences, such that I think the answer to your biomarker versus mechanism question is both. that there are some exceptions where you can't necessarily think about detailed mechanism at the mucosal surface from the stool because you're losing bugs, you're gaining bugs. But there's a bulk of the community is reflected there, and stool represents a really useful biomarker, such that in something like predicting colorectal cancer, Per Bork's done good work on that. That's where the stool microbiome is literally a biomarker, regardless of what the underlying mechanism might be.
Thanks.
Hi, my name is Brandon Eicher. I work for Nutrilite, and I have a personal concern that many of our Western populations have significant dietary deficiencies. So we're running clinical trials and things, especially in the West, and I imagine this probably goes to most developed nations. The consumption of dietary fiber is around maybe 15, 20 grams tops. When we look at Eastern populations and maybe, forgive me, wild humans, their consumption of dietary fibers are significantly higher, maybe 80, 90, 100 grams are some reports. I fear that many of the inferences we're making about a healthy control might be a little off because we're going to do a blood panel and say, well, that modern medicine says that individual's healthy. I think I significantly disagree with that point of view. And I was wondering what it is you guys might be doing in the future or future studies to try to find better controls so that when we control a diseased individual versus a Westerner, these are probably subtle differences between those versus when you go find somebody who lives in a more rural environment and consumes much more higher levels of dietary fiber. Their guts might be significantly different and much more easier to prove diseased versus healthy individuals. Thank you.
Yeah, maybe I'll take a crack at that. I mean, your point's well taken. First of all, what is the definition of healthy? It's always relative, and it's possible none of us are truly healthy at some level of definition. I think one thing we are doing, your point's well taken, that Western diets are generally, and Justin Sonnenberg, I'm sure we'll talk about this. This is one of his big mantras. The Western diet is very poor in fiber compared to other groups. And one thing we are doing with Justin as part of our H&P project, we actually have people doing a number of fiber perturbations. Those data came late in the study, so they're sitting there, and we hope to analyze them. They're getting analyzed in a more superficial fashion now, but hope to dig in deeper. So I do think we need to tear that apart like you would do analytical chemists. Even when people do these things now, they still throw in, complex mixtures. I would argue it'd be nice to know exactly what each fiber is doing to each individual so we can personalize this, and that is not being done as far as I can tell, at least in a systematic, broad systematic fashion. And if you think about it, the combinatorics are quite large, right? You have lots of people with lots of different individual microbiomes, and then you want to have lots of different fiber types. So we have short chain, long chain, hydrophobic, hydrophilic, et cetera. It really is positive, negative fibers. The combinations get very large. But I do think those studies need to be done so we can know exactly what an individual fiber does or an individual other supplement to each person and customize diets that way. That's my own belief.
Yes, Liping Zhao from Rutgers. So it's exciting to see that we start to have longitudinal multi-omics data set. And so there's going to be a huge opportunity for knowledge discovery. But if you look at the data sets, they are still the so-called big small data. So you have a huge number of variables, but a limited number of samples. And also the data set has a high sparsity. So a lot of zeros because of the individual difference. So it's very important to have a biologically meaningful way to reduce the dimension. So the first attempt would be taxon-based analysis. But we know it's actually not right to analyze data at a different taxon level because we are lumping all different things in one, in a new data set. So what would be a biologically or ecologically meaningful way to reduce the dimensionality of the data so that we can really go after such kind of data set?
Well, I think we all have an opinion on this one, so we'll probably get lots of responses here. But I think don't underestimate the power of longitudinal data. Yes, it's a small number of individuals, but in most cases we're looking for deltas. We're looking for changes across situations with IBD, whether it's pregnancy or in our case, whether it's what's happening in viral infections for pre-diabetics versus not. And that really empowers you in a way that you may not appreciate, and you can be very statistically robust for individuals, and you don't necessarily need large N if you're following perturbations of similar sorts across similar individuals. So you can easily tease out significant responses. So I think that is the power of the studies we're doing. It's the fact they are longitudinal. It really does give you an empowerment that's very different. You have to shake this whole GWAS, 100,000 case, 100,000 control mentality to appreciate the power of what we're doing, I believe.
Yeah, I can't add much to that except to think on the continuum from large-scale epidemiology-style studies where you're targeting many, many individuals, usually cross-sectionally, usually relatively shallow data, down to very small-scale, more like model system studies in great depth. The HMP2, and Lita, Mary, correct me if I'm wrong, is by design somewhere in the middle so that it's targeting quite a bit of depth in a medium-sized population with the hope of getting the best of both worlds as we move the microbiome more towards detailed mechanistic understanding.
Curtis, I was just hoping that you'd at least comment on, that even if the data is sparse, which we would all agree on, it's still, there's lots of opportunity for it to be statistically robust, right? I mean, that's fair, right?
And you also get a lot of information at the pathway level, even if you can't get it down to the individual molecule level, if you're underpowered that way as well.
Yeah, and then to echo what Owen said, there are, of course, methods for working with the data that are of the wrong dimension and sparse and whatnot statistically as well.
Hi, Peter Turnbaugh, UCSF. I guess I'm wondering, so it seems like a lot of the resources, both financially and just in terms of time, as part of HMP 1 and 2, has been the sample collection and establishing these huge biobanks that you guys all have access to. So, I'm wondering, is there some sort of plan moving forward to make those samples available, either to people that are part of this consortium or the broader research community that might be developing, new technologies to analyze those initial samples or the nucleic acids or other downstream materials?
I can speak for phase 1 because there is a catalog of DNA extracts that are available, but they're not freely openly available. They're meant to, they're set up in such a way that you'd have to collaborate with one of the clinical centers that's collected the samples. So if you're interested in DNA extracts from the Healthy Cohort study from phase 1, and there's not a lot of them left, but there are some, I would recommend you contact me directly.
I guess I'd like to go a little meta on your question, though, which is, like, if we think about every presentation that we're going to see today or anything that's funded by NIH, one opportunity that's out there is it'd be very nice to have essentially kind of a PI registry where you could ask the question, okay, you've published this, but are the subjects available for reconsent, or are the samples available that you could pull out in the refrigerator? What are the resources that are associated with it? Because that's another thing where there's just this tremendous loss between all that those incredibly rich resources that the taxpayer paid for and may be available. And when you go to SRA, or I'm not to beat up on that database, but when you go to any public repository, that's not explicitly stated in those repositories.
So just to echo what everybody else is saying, we probably have the biggest biorepository of data from the HMP2 study in the order of a couple 100,000 samples that are stashed away in our biobank, or what's called the Research Alliance for Microbiome Science Registry. We did that with the idea that we would leverage those into future studies in our own group and in collaboration with others. And we're very willing to do that. The issue is the maintenance of this facility and the interaction, and we really don't have financial resources for curating and processing these samples so that we could distribute them or have any kind of way to do that yet. We're trying to work on something like that, but at the moment we don't have that. If somebody does want to have a collaboration with us, we're happy to talk about it.
should wrap it up. I don't want to cut into lunch hour or actually, more importantly, maybe the poster session. So the first poster, there's 3 poster sessions, 1 each day, 40 posters above in the atrium. So make sure and visit the posters. If you ordered lunch, I think you have to ask the registration desk where you pick up the lunches. I believe they're down here on the lower lobby. That tent, that big white tent out there on the natural lawn is ours. So go out there, enjoy yourself, network with people you haven't met before, and come back at 1 o'clock.
Okay, everyone. Welcome back from lunch. I like to be right on time, so we're already 3 minutes late. That's causing me stress. I wonder what that's doing to my microbiome. Okay, so this session is, oh, sorry, I should introduce myself. My name is Lisa Chadwick, and I'm a program director at the National Institute of Environmental Health Sciences, where we are interested in how the microbiome interacts with environmental chemicals. And if you would like to be interested in that too, feel free to find me at this meeting and talk to me about it. So in this session, we're going to be talking about the tools, technologies, and approaches for microbiome research that go beyond sequencing and try to identify what are the big needs, gaps, and challenges in that area. So we'll have a session just like we had the last one, where we'll have 4 talks right in a row, although we're going to have 1 talk, 1 sort of mega tag team talk, and then another talk, and then we'll have that joint Q&A session at the end. Our first talk is from Katie Pollard from University of California, San Francisco, and you can come on up here. Your slides should show up.
Is the timer working? Do we know? The timer is working, and I'm going to make it start. Great. Hi, everybody. Welcome back from lunch. I wanted to start by putting up a picture that represents what I think a lot of the work on microbiome science has been to date, especially any science related to the human microbiome and hopeful translational work, which would be you have a patient with symptoms, you gather some biomolecules, and we count up the percentage of sequences that come from each microbe or from each gene, make these stacked bar plots or pie charts, and then try to associate that with what's going on with the patient. And as we know, many interesting associations are detected. What I'm interested in is moving beyond these associations towards mechanism and trying to understand what's going on at a genetic level in the microbes that would be explaining those associations, and what associations might we be missing with these stacked bar charts and pie charts. The reason I'm worried about this is that even if we can characterize microbes at the level of individual species, we know that different examples of that species, different populations of cells, have different genes. And this touches on what Howard spoke about in the keynote about what is a species anyway. And we're interested in the same question. And we know, all of us, from looking at genome sequences, that the gene content differences is different across different strains of the same species. And my lab developed a tool to quantify that using shotgun metagenomics data. So we took 2 people from a variety of studies that were available in the public domain, and we asked for the sequences that represent a given species that is shared between the 2 of them, what percentage of genes are they sharing? And here's an overview of the results. So the horizontal axis is what percentage of genes are shared across people. So each dot is a pair of people. And these are organized by the different species. And you can see that there's a lot of variation, with some species having only maybe 10% gene content difference, but some of them having up to 40, 45, even 50% of their genes different between 2 people who harbor the exact same species. And of course, if you go up to genus or a higher taxonomic level, that's even more true. And we then looked to see whether the gene content was organized according to where people were from. And the answer is it depends on the bug. So for some bugs, there are these huge differences between gene content, and it has nothing to do with where someone's from. There's basically a big star phylogeny, and the different strains get everywhere. In Howard's talk, there was a question about, we got into talking about dispersal limitations and whether there's barriers to gene flow. For some species, there seem not to be. Whereas for others, like E. rectile that I'm showing here, there's a lot of structure with clades of based on either the SNPs, the nucleotide differences in their genome, or in their gene content, really organizing by continents. However, but with some sharing of these two clades here between North America and Europe. The other reason that I'm worried about getting down to a genetic level rather than a taxonomic or a gene level description of the microbiome is that even when you have the same species in two people and you look at genes in the core genome that are shared, so they have the same species and the species has the same gene, those genes harbor genetic mutations, some of them that make functional differences in what that gene is doing. So these are different species. This is Human Microbiome Project data. And in red are between host differences. So across these different microbes, on average, about 1 in every 100 base pairs, so 10 to the minus 2, 1 every 10 to the minus 2 base pairs, there's a genetic mutation between people. And even within hosts, we see genetic variation maybe every 1,000 or 10,000 base pairs. And it differs across the different pathways, of course, in different species, but there's frequently something like 25% of those mutations that I'm describing here change the protein coding sequence of a gene. So they have potential to be functional, high potential to be functional. So the idea that I want to put forward for sort of a future view of the microbiome is to study microbes in the human body at the level of genetic mutations, just like we do in human-based precision medicine. If we're going to sequence people and give treatments and diagnostics and prognostics based on your genetics, why not do that also for the microbes? And of course, this extends to other types of multi-omics, not just genetics, but you could say the same thing of the proteomics and the metabolomics as well. So we've developed a concept called metagenotyping and bioinformatics tools for doing it. And the idea is to identify these gene loss, gain, and mutation events that I was just showing you pictures of to confidently be able to call those within the populations of cells for each species present in a metagenome. And the reason, besides the two that I've already presented, or the reason, sort of maybe the reason I'm the one doing this and not someone else, is that I come at microbiome research from an evolutionary perspective. I've spent a lot of my career studying the evolution and genetics of the human genome, and of course, therefore, think about how form and function change through genetic modifications. Also, being at UCSF on a biomedical campus, I'm thinking a lot about what does the host care about, which is, of course, the chemicals and biomolecules that are presented by the microbiome and how the microbiome cells are interacting with host cells. So those reasons make me very interested in understanding this sort of strain level, subspecies level genetic variation and being able to do this metagenotyping. So why is this not more prevalent? Well, it's really hard. First of all, there's a lot of challenges in any kind of analysis of metagenomic data, and these would apply to trying to genotype the microbiome as well. I'm not going to go into them in the talk today, but as Curtis mentioned also this morning, there are other groups looking at this, all the different sources of error. We wrote a review last year that goes over some of them. Many of them are common in any kind of genomics or multi-omics studies, whereas other ones are things we as a field had to work out that are specific to studying pooled samples of communities. Things about how fast are the cells dividing, what are the sizes of the genome, what's the size of the target that you're even trying to sequence, and what are the unknown species and genes in a sample, all lead to systematic differences in the relative measurements of other things that are there. So the knowns. And so this is something we've spent a decade thinking about, and that I literally thought we'd maybe spend 6 months or a year on 10 years ago.
But I feel like as a field collectively, we've discovered a lot of these gotchas and in many cases have the tools either in the lab or bioinformatically to handle them now. But doing this is absolutely critical to the type of data sharing and meta-analysis that we heard about this morning. So that's just general. When you get down to doing this metagenotyping, there's some additional challenges. And one of the biggest ones is that most of the species that are present in most microbiomes have no genome. And so if you want to identify polymorphisms and say that they're occurring in a specific species, it's very helpful to have this kind of genome-level reference data. We and others are thinking about ways to do genotyping without mapping to a reference genome, but that is a big challenge, both computationally and just in terms of how you would think about approaching that problem. So as a first pass, my lab's focused on reference-based genotyping, but we know that it's incredibly limited by the availability of reference genomes. So what I'm showing here are some estimates that we made using universal single copy genes to understand how well databases of tens of thousands of genomes that have been sequenced so far cover the species-level diversity in different types of microbiomes. And you can see for the human microbiome, it is better than some of these other environments, but it depends where you're from. So our databases are very biased towards industrialized microbiomes. And it also, I'll point out the mouse microbiome is down here below 10% of what's there at a species level has been sequenced. And so that is a challenge for using the mouse as a model. Even though it's hard, I think this metagenotyping approach is very important. So I just want to give you a couple examples. And this is, I will say, totally just scratching the surface of what I think is possible with metagenotypes. But to show you a couple of the two examples of what we've been able to do so far. The first one is that we can now disentangle taxonomic signal and functional signal in metagenomics data because we know gene presence absence per species. And this is the work of Patrick Bradley. What I'm showing here is an example output. I'm not going to tell you for the sake of time about the method, but I'm happy to talk to you about it. This is a particular gene family whose presence is associated with Crohn's disease, not overall, not the overall abundance, but its presence in specific species. So on this side, we're seeing the association of this gene with Crohn's disease, and here is the presence of it in these organisms. And these trees mirror each other beautifully, and you can see, like, for example, here, there's a group of taxa that have the gene, and then in gray, one that doesn't. And here, those specific strains are found in Crohn's individuals, and this one that doesn't have the gene is not found in Crohn's individuals. And this, I'm explaining that as a binary, but it's actually a quantitative variable. So we can associate the presence of specific genes in specific taxa with diseases, with body sites, and we can also just look at the natural variation of that across people. And this is different, just to get back to what I started with, this is very different than looking at how abundant are the different taxonomic groups or even just looking at how abundant are the different genes. This would have been missed with either of those approaches. And importantly, I will say that the proteobacteria are contributing a lot to this signal and yet are a tiny piece of the stacked bar chart that might be overlooked in a taxonomic analysis. I'm sorry for that noise. I think my necklace is scratching this. I'm going to try to stop that. Sorry. We have been able also, in cases where the genes that we identify as having these interesting patterns of being variable across people or body sites and being associated with diseases, we've been able, when they are annotated, to look at how the individual components of the pathway map out. So we found that in particular across HMP and the controls from a number of publicly available case control studies, healthy individuals, that type 3 and type 6 secretion vary a lot across people. But it's not the whole pathway.
it's specific genes. So the components that sit here in the membrane and these TAT and SEC-SRP parts of secretion are fairly not variable across people, as shown by their genes being shown by these blue blocks, whereas the pieces of the machinery that are sensing signals inside the cell and then doing the secretion into either a host or another microbe cell, these injectors, are highly variable. And so it's specific components of the pathway that we can disentangle and try to move towards mechanism. This is, of course, impossible in most cases where we find a really interesting signal, but we have no idea what the genes do or what the specific mutations, even if the gene is there, what the mutations say, a non-coding mutation in a gene, what is the effect of that going to have? So this is one of my gaps and challenges for knowledge in the future, is to try to understand that. And the challenge is exemplified by this number. We have a short little paper we're putting together right now where we evaluated all available gene families that we could find in databases and found that 53%, not only do we not know what it does, they have not even a single protein domain that's annotated. So there's nothing to even go on. And for a lot of them that have a domain, of course, we don't really know what they do. So that's a huge gap. The last thing I want to say is that even without knowing what genes do, we can use this genetic variation for tracking strains, and this will be very useful in a number of settings, including clinical settings. Here we reanalyzed some publicly available data on the infant microbiome from Backhead et al. And we and they showed that the infant's taxonomic composition starts to look more and more like the mom over the course of the first year of life. However, we swapped the kids just on the computer and matched them to a mom that wasn't their mom, and they also start to look more like that person over the course of the first year of life in terms of taxonomic composition, reflecting just a general maturity of the microbiome, not what's inferred by us and many other people from this, that there was direct transfer of the microbes. So to test that, we went and we found genetic mutations in specific microbes in the mom that were not found in any other mother in this study, not in the HMP, not in any public database. So they were private mutations, and then we tracked her strains to see whether when the infant acquires the same species that the mom has, is it the same strain or not. And there are some direct transmissions, especially at birth, but the overall signal is the exact opposite, that over the course of the first year of life, the infant is mostly getting strains from somewhere other than the mom's stool. So to wrap up, we've quantified massive diversity at this genetic level. There's some vertical transmission, but not a lot. And what I wanted to go on towards, if we're going to achieve this microbiome precision medicine, is one other, just to conclude, one other technological gap. And this echoes a little bit of what was said this morning, but I want to make a specific pitch for reference data in the cloud. So not just microbiome samples in the cloud, but really good curated reference data. Not just sitting in the cloud for someone to download and not necessarily be working with the same version as someone else and not being able to necessarily handle the size of that data on their own computer, but sitting there with high-speed compute that you can spin up, and lots of users can now be looking at the same data and running analyses in the cloud and creating annotations, especially through multi-omic studies that get at function, curation of that data, and then adding curation back up into the cloud for other people to see. So this is right now a little bit of a pipe dream, but we heard some great steps in that direction from Owen and others this morning, and I think this is something that NIH could really tackle, and I want to make a pitch for it today. So thanks a lot. Here are the folks who did the work, and I will take questions at the end with everybody else.
Okay, so thank you for that talk. So the next talk is going to be a two-person show by Rob Knight and Peter Dorstein from UCSD. Use your slides.
Yeah, we'll just use the... Right in 1 second. which, great. Sorry, I got to pick you up on video. Okay, fair enough. All right,
Great. So I'd like to begin by thanking Lita for organizing this and for inviting us all, and I'll say to Lita and Bob Kapp and Vivian Benazzi, who've been very active in supporting this kind of thing over the years. And what we're going to tell you about is integrated multi-omics for insight into health and disease. And originally we were going to do these as one talk on the microbiome and one talk on the metabolome, but we decided that these topics were inseparable, and so we're going to be switching on and off during the talks. So there's very rapid advances in these fields, and they're really changing what we think it means to be human. And I'd like you to take just a moment to consider what you saw when you looked in the mirror this morning. We saw an organism that's just 43% human. Then I got here and I found there was no coffee, and I think it dropped down to about 33%. But where that 43% number comes from, when we think about what makes us up as human, we have about 30 trillion human cells and about 39 trillion microbial cells, according to the latest estimates, where the error bars have narrowed somewhat. So that's where that 43% number comes from. But then, as you heard this morning, when we think about it at the level of our DNA, we have about 20,000 human genes to 2 to 20 million microbial genes. So that 8 million figure comes from HMP1. In the phase 2 of that, we found a whole lot of additional genes, as Owen and Curtis alluded to. And so by that measure, we're at best 1% human. And there's a lot of excitement about systems biology and systems medicine, but it's really hard to do that when we're neglecting 99% of the system, as we do when we disregard the microbial side of ourselves. Now, you've already heard about the HMP and the tremendous achievement of the HMP in producing a reference data set. And I won't recount the statistics, but basically this project collected a total of 4.5 trillion A's, T's, G's, and C's, right? And so the great thing about this is that we had a lot of data, but that was also the terrible thing about it. So when I show you the first file of data from the HMP, despite the fact that what we're fundamentally doing is ecology, it's pretty hard to tell who lives where in the environment from this, right? And this is just the first file of this data. There's another 17,000 just like it. And this is the first 0.1% of the contents of that file. And so we really have to work that out for us. And this is especially problematic now that this kind of thing is in the hands of ordinary members of the public, right? Because through crowdsourced projects like American Gut and through various companies, you can now get your microbiome profile done. And so you have people coming into their doctors and saying, you know, hey doc, I have some great news for you, which is I had my microbiome done. Now I have a list of 1,000 microbes that were in my gut. Or even better, I had it done with Shotgun, and I have this list of 1 million genes that were in my gut. With all this data, you can tell me what's wrong with me, right? And I mean, what are you going to do? You can refer them to your colleagues in psychiatry for being crazy enough to bring that stuff to you and to think you could interpret it in the 15 minutes you have, right? But what our goal is as a community is to make it not crazy anymore and figure out how could you get something out of that profile, either with other data from the same person over time or with a reference data set. So you heard briefly about CHIME this morning. When we apply CHIME and Unifrac and other tools my lab developed to the HMP data, you take all those sequences and you get this much more interpretable map. And so what's going on in this map is each point represents all the complexity of a microbiome as read out by its DNA, typically with 16S, but you can use these techniques on shotgun metagenomics or metatranscriptomics, or even, as you'll see, to some extent on metabolomics as well. And when we color all that data according to the main variable that affects variation in those samples, you can see immediately that the main thing going on in the HMP samples is the different parts of the body are very different from one another. When I highlight the first person, you see their mouth and their gut are in totally different regions of this map, almost like different continents. And it was really only when we did the Earth Microbiome Project, looking at tens of thousands of samples from across the Earth's surface, that we understood how profound these differences on the human body were. Because what we could do is we could go out on the planet's surface, we could ask what 2 points on the planet's surface are just as distant from each other as the mouth and the gut of this one person. And if you think about your mouth as being maybe like a coral reef with complex mineralized structures covered with biofilms that your dentist bugs you about, the amazing fact is that your mouth is as far from your gut and its microbiome as the water in that reef is from the dirt in this prairie. So they're essentially non-overlapping communities. And that leads us to wonder, where do these highly differentiated communities that we have as adults come from in the first place? And if you have dogs or kids, you likely have dark suspicions about that, and all of which are true, by the way. So I can actually match you up to your dog with fairly high precision based on your microbiome. And even your dog's microbes affect how you develop. But in all seriousness, and as Gloria will tell you on Friday, how we're born has a tremendous impact on our first microbes and then how we subsequently develop. So I'm not going to show you that, but I will show you the value of longitudinal data. So this is work we did with Ruth Lay, looking at the development of the gut microbiome of one child over the first 2.5 years of life. And you can see that the first infant fecal sample looks nothing like a healthy adult gut sample. So it's up there in the vaginal region of the plot, which we would actually expect from this child's delivery mode. What I'm going to do is I'm just going to connect up the points one to the next, so you can see the 2.5 years of development at 1 week intervals. And in case you're wondering why 2.5 years, that's when the kid was toilet trained, and it's a lot easier to get a fecal sample out of a diaper than to fight someone who's probably learned how to flush. So I'm going to start this going, and what you can see is sometimes there's tremendous variation 1 week to the next, other times it's just a little variation. And sometimes the distance traveled in 1 week is greater than the difference between any two healthy adult samples in the HMP. So if you think your child looks like a different person 1 week to the next, that can literally be true in terms of their microbiome, where most of their genes are in the first place. But coming up here is something fascinating. So what you're going to see is he gets antibiotics for an ear infection. You see that tremendous regression of the microbiome, followed by a rapid recovery. That went by pretty fast, so I'm just going to rewind it for you and play it for you again. What you're going to see is on administration of oral amoxicillin, we see this tremendous reversion of the microbiome undoing months of normal development, followed by a relatively rapid rebound again in just a few weeks. And then by the time he's at 2.5 years, you can see that he's basically in the healthy region of the plot. So what we're trying to figure out at the moment, and again, everything I've shown you here is just taxonomy, so just the name tags. What we're trying to figure out is why are some people resilient and other people very, very fragile to this kind of microbiome change.
So we started to address this in trying to determine the chemistry associated with your microbiota. And we started to address this by answering a very fundamental question. What kind of molecules does your microbiota produce that then can control the proliferation and colonization of a variety of different pathogens? And so the first thing that we did is we made everybody work out in the lab, and then we had them isolate, basically use cotton swabs to isolate different microbes from them. And if you were to do this exercise on yourself, what you will get is you get organisms that look like this. It's just absolutely beautiful. But basically, anybody will be able to see these kind of microbes that even play football, as it looks like. Really unusual features, color red, and so on. But we then take these microbes and we would co-culture them with pathogens, and we developed something called microbial imaging mass spectrometry that allows us to actually look at the chemistry associated with these microbes directly from petri dishes. And so what you will see here is that this bacillus that came from a toe from one of the individuals that we sampled from, that it produces a red molecule. There's a gradient of a molecule. But then you start to see these blue molecules. These are delta toxins, phenol-soluble modules, and Staph aureus that are produced by Staph aureus. And these are virulence factors that allow Staph aureus to invade healthy individuals. And so based on this image, what we proposed was that if this red molecule is present on the skin, maybe it suppresses the production of these virulence factors. Maybe you get a decreased ability for Staph aureus to colonize these skin environments. And that's a testable hypothesis. So we isolated these particular molecules. We then add Staph aureus to it. And we simply asked the question, how many colony-forming units of Staph aureus can you isolate from this? And what you will see is that upon treatment with this particular molecule, you see about a 75% reduction of colony-forming units that you can observe of Staph aureus. And so that supports the hypothesis that your commensal organisms that live on us, at least, that they produce molecules that can control at least the colonization and proliferation of pathogens. But we actually pushed this a little bit further and wanted to see if these kind of organisms that produce these kind of molecules, what impact can that have on health and disease. And that ended up being published in this paper with Rich Geller and Carsten Sengler, who is here as well. But essentially, we took healthy microbes, we added to creams, add them to atopic dermatitis patients, and what you will see is you see a decrease amount in atopic dermatitis symptoms, and it's related to the suppression of Staph aureus. So there's really an important relationship there. But this is an artificial system. Here we're really literally overwhelming the system with these healthy microbes. So the question that I have is this really a normal condition that might be present on the skin? And so the reason why I ask that is because these molecules have never been directly detected from the skin. And so we wanted to come up with technologies that would allow us to see the chemistry associated with it. And that actually led to our first research paper, joint research paper with Rob Knight, where we started to map the chemistry in the microbial communities in 3 dimensions on the human surface. And so the idea was actually simple. If you spatially collect both the molecule information as well as the microbial information, you should be able to determine what molecules are associated with these microbial communities. This could be because the chemistry dictates the growth of these microbial communities, or it could be that the microbes produce these molecules, or that they modify molecules that are present on these different regions of the skin. And so, just to show you a very first data set for the metabolomic data, that's this movie right here. But what you will see in the upper right-hand corner are all the molecules that we're able to detect by mass spectrometry. And then in blue, it's low concentration, in red is high concentrations of these molecules. And so what you can see, as we're scanning the molecules, the MOZs, you can start to see it sort of flickering. What this is essentially telling us is that there are different chemical distributions that exist on the skin surface. And when we stop these movies in time, what you will get is you get molecules that are associated with the belly button, molecules with the hands, molecules in the groin region, shoulder region, molecules that are uniformly distributed throughout the entire surface, but also some molecules that are low in concentration on the head, and as you go towards your toes, it increases in concentration. And so if you're a microbe and you want to find your ecological niche, where in that gradient does it fit? And we have very little understanding of this. But what that allows us to do, now we also got 16S inventories of this, and we can also create these corresponding microbial maps. And so here we're looking at Propionibacterium. They're on the chest, back, and head area. And there's about 850 genera that we were able to detect in these particular studies. And so now what we can do is you can take advantage of the spatial information and say, okay, what microbes are co-correlated with the different molecules that are present, and can we use that to understand why those molecules are there or what the molecules are doing. And indeed, we were able to find a series of molecules that were associated with the degradation of triacylglycerides. And that you can recapitulate in vitro. When you take a Propionibacterium and add triacylglycerides to it, you can start to see the degradation products such as oleic acid or even the oxidized versions. So, but what this basically meant is that microbes don't only just contribute molecules that are present on the surface of your skin, but they also can modify existing molecules that are present there as well. But a data set like this, really, it generates has the capacity of generating many different hypotheses. And this was recognized by Francis Collins when he blogged about this particular paper, that this kind of data, essentially, he said, this kind of data can generate lots of hypotheses. But how do we continue to learn about knowledge about a data set like this? Well, for the metabolomic side, we built this crowdsourced analysis infrastructure. This is a worldwide effort, basically, with 127 authors that we published. But what this is a place where you can deposit all your data. So it's the largest metabolomic repository at the moment. It has about 3 billion spectra in it. But you can make annotations to your data, or anybody else in the world can make annotations to your data. And if those annotations match to other people's data, they will get that information relayed to them. And so you really build this community knowledge associated with metabolomic data, and it's also a way to capture that knowledge. And so the knowledge is clearly growing very fast, and what you will see is there's individual contributions, and sometimes you have large contributions, such as the one from PNNL here. Sorry, I accidentally hit the wrong button. But the skin data sets, it was clear that the knowledge about this particular skin data set was growing very fast as well, because initially when we deposited the data, we had 98 annotations, and by now we're 772 annotations. So it's really grown very quickly. But at one point, we got an email where they were saying, oh, hey, you have these degradation products right here that come from heme degradation. And because somebody in the world was studying an organism that was actually degrading heme, and they made these annotations, and we found those in our human maps. Where did we find those? Well, they were in the groin region of the female. Now, we'd already seen in the paper when we published it that heme was present, and that was consistent with the fact that she was on the late stages of her menstrual cycle. But what this shows was that there's now a possibility that there is some process there that is involved in the heme degradation. And one of the things that we can look for are microbes that sit in that particular region. And you can actually look if any of these organisms have this heme oxygenase. And I know Streptococcus has one of those heme oxygenases that is present. And so now you can build a hypothesis that these microbial communities are actually involved in a normal physiological process that affects 50% of the population. But if you really want to determine which one of these organisms is truly responsible for that, longitudinal studies would probably be ideal to do that.
Absolutely. And so we've been getting more and more into longitudinal studies of the kind you saw this morning with HMP2, which we think are critical for understanding therapeutic response. And this is not that snapshot data aren't valuable. So, for example, in American Gut, we've had over 10,000 people sign up to send in their samples and get them analyzed. Of course, not everyone wants to know what's in there. These are middle schoolers touring our lab, learning that we're literally going to use lasers and robots to study the bacteria in their poop. But you can get so much more out of a time series than this single snapshot, and especially when you integrate with data frames like the HMP data. So this, like the infant time series, shows you what you can get integrating with the HMP. In this case, we're going to look at C. diff infection, and this is a collaboration we did with Mike Sadowski and Alex Karutz at the University of Minnesota. But when we look at the fecal samples from C. diff patients, you see those spiky symbols there. They're totally different from the healthy stool down the bottom. And what's going to happen is that 4 of these patients are going to get a fecal transplant from one donor to replace their dysbiotic microbiome. And remember that C. diff infection is one of the most important hospital-acquired infections in the United States. Kills about 14,000 people a year. So anyway, the questions are very much like the questions that I showed you with the infant time series, only now, instead of taking a week, each frame is going to be just 1 day in the lives of these patients getting this fecal transplant. And you might wonder, given that C. diff is the underlying pathogen, will you even see any effect at the whole community level when you do this? But what you can see is that essentially immediately, so in just 2 or 3 days, all 4 patients move their microbiomes completely into the healthy state, and then they stay there during the months of follow-up. And this is coupled to remission of all their clinical symptoms. In fact, the last large-scale trial that was done to directly compare fecal transplant to antibiotics had to be stopped early because the fecal transplant was over 90% effective. The antibiotics at a standard of care were less than 40% effective. So it was considered unethical to continue withholding fecal transplant from those on the antibiotic arm of the trial. So they all got it and most of them recovered. But what's amazing about this is that it shows you how the microbiome over time can show you your progression through a particular therapy. Now, shortly after I got to UCSD, Larry Smarr, who's our director of CalIT2, was looking at the HMP data with me on a 64 million pixel display wall, and he said, well, you know, Rob, I have a very interesting microbiome myself because I'm an IBD patient, and how can I get myself on that kind of map? Now, those of you who've moved institutions know that when you do this, you expect to get a certain amount of crap from your colleagues, and in my case, it's literally been true. But Larry's been fascinating in that he's been studying himself for the last 17 years, beginning with 1 dimension of his weight, and then adding dozens of dimensions with stool tests, thousands of dimensions with SNP profiling, and now millions of dimensions with 16S and also shotgun metagenomics. And he'd been studying his microbiome for a few years, and he got this great taxonomy chart. If you don't see any patterns in there, don't worry, because neither did Larry. I hate to think of the amount of compute and personnel effort that went into making that graph. But when we redo it with Unifrac and Chime, what we see is this much clearer pattern with a blue region and a red region. When I animate it over time, what you're going to see is that he starts off at the edge of the blue, moves through the blue, crosses over into the red, and then basically bounces around at random in the red. And so you might be wondering, does this actually have anything to do with his health? And the answer is yes. So you can see this initial shift in his microbiome that's triggered by antibiotics. Followed by that, he has very frequent IBD symptoms. He's losing weight. His life is pretty bad. he switches his meds, moves from the blue region to the red, and then as soon as he's in the red region, his weight goes back up to a healthy set point, he's feeling a lot better, and most of his IBD symptoms are in remission. And so what's amazing about this is had we known Larry's individual configuration space, we would have been able to tell him as soon as you're in that red region, you're essentially going to be fine for a few years. And so having these personalized microbiome profiles are really important. Now, you're probably thinking, well, this is just, this is just one guy. Can you generalize this? And this is some work we did with Janet Jansen and a number of other collaborators, looking at the dynamics of many Crohn's patients with different forms of Crohn's. And when I start these animations going, what you can see is that the yellow and red traces, which are ileal Crohn's with and without surgery, there's a tremendous amount more variation than in, for example, the healthy controls in green, or the ulcerative colitis patients in blue who vary less overall. You can also see that there's this periodic visitation to the healthy region, even in the people with IBD, making it difficult to tell what's going on from a single time point. So we figured out how to mathematically model this as a healthy plane that we're looking at distances from. So the top row is a cartoon of how we fit the healthy plane and then measure the distance of each point from it. The bottom is the actual data with two different distance metrics. And what was amazing about this is that we actually got better disease classification from this microbiome-based assay than from calprotectin, which is currently the gold standard.
So looking at time changes is one of the ways that we can begin to understand the changes that happen within the microbiome. But spatially, you can also do this, and really it's an opportunity to integrate multi-omics information. And so I'm going to highlight this with this particular highly interdisciplinary study, and that is led by my former postdoc, now a faculty member at Georgia Tech, Naya Garg. But she was fundamentally interested in what are the chemical environments associated with these microbial communities that actually exist within a cystic fibrosis lung. And so one of the things that happens with cystic fibrosis is that your lung function diminishes. And so when your lung function diminishes so significantly, you get put on a transplant list. And so what Naya Garg was able to arrange was that we would get these old explanted lungs from these patients, and then she would section these in several hundred pieces and subject these to both metabolomics analysis as well as 16S inventories. And so then what she would do is she essentially mapped this data onto the CT scan of these individuals to gain spatial insight in terms of the distributions of the microbes as well as the molecules that were present. And using the co-localization strategies and really creating networks of co-localization of molecules and microbes, she was able to formulate a very interesting, she was able to observe something very interesting in that there are actually microbial interactions that take place that change the metabolism of these organisms when they're interacting. For example, here we see these quinolones, and quinolones are known to increase biofilm formation in Pseudomonas aeruginosa, and maybe that's why we get a local decrease in lung function when you get an increase in the amount of these biofilms that are created. So, but this is one observation that she had. There's a lot of other things that we're able to observe. So we, for example, can see the distribution of antibiotics. In this case, I'm just showing you 4 of them, but we found at least 20 of them. But notice some of these really interesting gradients here. It looks like either the drug is only partial penetrating, and this was a really interesting observation because that suggests that maybe there are microbial community gradients that exist within these lungs as well. And so she was indeed able to find some of these microbial gradients where you look at these gradients of antibiotics on the left here, and one of the things that we noticed is that we started to see other microbes that are typically not, you wouldn't think of a lung to have anaerobes present, but where we don't see these microbes or antibiotics penetrate, we start to see an increase in anaerobes. And so the question that we had is, well, what's really happening to these antibiotics here in these type of gradients, and how can we test this in a more in vitro model? And so Robbie Quinn in my laboratory, a project scientist, he came up with a system where you have basically a capillary tube, actually not a capillary, it's a 10 millimeter tube. The bottom of it is sealed, and he adds sputum to this. And so what you now have is you have a zone where that is oxygen exposed, and then the bottom part essentially becomes anoxic. And so we were able to use this to recapitulate the phenotype that we were able to see within these lungs, where you have oxygen-loving organism, even though Pseudomonas can grow all the way into the anoxic zone. But we clearly see some anaerobes that are present in these particular samples as well. What is nice when you have such an in vitro model that you can recapitulate some of this information is that you can alter the conditions. So, for example, you can add antibiotics to it. And one of the striking features that we saw is when we added antibiotics to, such as tobramycin, which is an inhaled antibiotic that's commonly used in cystic fibrosis, we actually could barely detect the tobramycin itself in 16 out of the 19 patients that we tested, whereas in 3 of the patients, there was no degradation or disappearance of the tobramycin antibiotic at all. And if we look at the sequencing of those particular patients, those 3 patients, we observed something very interesting. Using V4 amplification, we actually saw that there was an abundance of Aspergillus mitochondria there. And of course, the annotations using V4 amplification mitochondria is not that strong in terms of being able to annotate and confirm that this is Aspergillus. But this is where multiomics really comes into play, in the fact that we also see a lot of Aspergillus fumigatus-derived molecules in those same zones. But what this suggested was that upon antibiotic treatment, we were removing microbes in these patients, allowing Aspergillus fumigatus to thrive. And this is a common problem in cystic fibrosis, where you get these Aspergillus fumigatus fires. But one of the questions that I had was, well, what's happening to the tobramycin? Why is it disappearing in those other patients? Well, we use molecular networking, which is a unique analysis tool that we have introduced in 2012, but is now part of this community resource. It allows you to make relationships between the molecules that are very similar. And one of the things is you can very quickly see that there are a lot of degradation products associated with this tobramycin. But in this one patient, it was very interesting. It was the only patient that had a majority of this. And what we ended up finding out is that it's actually modifying this nitrogen here with a propyl group, but only in the anoxic zone and not anywhere else. And so this is a new modification that has not been demonstrated before, but it shows that if we start to look in these kind of lung environments into these gradients, we may actually find new mechanisms of antibiotic resistance.
So we were asked to speculate about the future and about where this could all ultimately lead. And what we'd like to do is we'd like to get this stuff out of the realm of the lab, where it costs, where you need millions of dollars in mass spectrometry and sequencing equipment, and into the clinic and perhaps even into your home. And the concept is that microbiome and metabolome analysis should be as easy as examining yourself in the mirror. Perhaps you breathe on that mirror and the molecules are whisked away through pores in the glass through a mass spec-based assay. And there's already breath tests for things like cystic fibrosis exacerbation and for SIBO, so that's perhaps not that implausible. And the idea is you could get an instant molecular readout, presumably with a better user interface than this, but on your mirror as you look at it. We could use the same kind of technology that's in Google Translate to turn that into a microbiome profile like you would get out of American Gut, or alternatively, perhaps it needs to connect to your smart toilet to read out your last data dump from there. And then the idea is that we can integrate it with large multi-omics resources, such as the HMP, to identify when you're at risk for a particular disease based on changes in your individual microbiome profile. And then if there's anything that you can try at home, or if instead there's something that you need to talk to a physician about. And then you could even imagine having that communicate back to your smartphone, so when you're faced with 1,000 kinds of yogurt in the supermarket, you can use augmented reality to zoom in on the one that you're trying to find, and then scan the barcode to confirm that you actually got it. And while this might sound like absurd science fiction, if you thought about, say, a Cray 2 supercomputer from 30 years ago, the idea that you would hold in your hand this thing that took the size of a room and required a team of technicians to keep it going, and that it would be networked to 1 billion others worldwide, and that you wouldn't even need any training to get started with it. That's amazing, right? And so the idea that we could come up with better user interfaces, not just for the instrumentation, but also for your microbiome itself, is really inspiring. And that's the kind of thing that we're trying to put together at UC San Diego. But just to finish this, we're going to motivate you with, if you could do this, what kind of insight can you get right now from the current technology into the disease process through these multi-omics techniques that otherwise you might miss entirely?
So that gets us into a case study. So Rob and I have implemented a program where we can give fairly efficient feedback in terms of functional understanding of what's happening to the microbiome. So this was an adult female, who's 39 years old, long history of heart problems, or 4-year-old history of heart problems. In fact, in 2012, her heart stopped beating and she had to be rescued, and fortunately they were able to. The cause was never established. And so then in 2016, she again started to have many major heart problems. In fact, she would have a heartbeat of 55. She was in very good shape, actually. And just by standing up, it jumped up to 188. So that was pretty severe. Also started to have lung problems. Started after 3 months after flu. So none of the tests could reveal the cause. And so we initiated an effort to understand this. So fortunately, in this particular case, we had a sample from 2014, so we were able to compare to the current state to a previous state. And one of the things that you will see if you look at the sequencing, you don't even have to squint. You see this bright green bar. This is chlamydia pneumonia. This is an intercellular pathogen that invades healthy tissue. It invades the lung, the brain, the heart. And when this organism activated, it actually paralyzes all the cells that it's in. And so upon subsequent treatment, in this case they decided on an antibiotic treatment, you can see that organism pretty much immediately disappeared, and you can see how this particular individual recovered. And all the heart symptoms disappeared upon treatment. We can also monitor by metabolomics the inflammatory state when she was really sick. You can see proteolysis as a marker of cell death. And then upon treatment, you can see the drugs slowly disappearing. So just to re-illustrate, here you have a picture of a semi-healthy individual. The disease state upon treatment starting to recover over the 80 days. It's starting to get closer to the healthy state. And it recapitulates in both the microbiome as well as the metabolome. What I find remarkable is that this was a person that was having heart problem, yet the treatment was antibiotic use. And so, of course, Rob and I both run large groups, and we also collaborate quite extensively. So we have actually a lot of people to acknowledge. This is the crowdsource group that started it. This is my group and collaborators. And then if you have any questions, just send us an email, and we would be happy to answer them. And these are our gaps and challenges. Thank you.
Thank you. I'm really glad we got to see my favorite slide, which is the horrified middle school photo. Okay, and so the final talk on this session is Elhanan Borstein from University of Washington, and we'll hear about some more gaps and challenges in this area.
So hi, everybody, and good afternoon. It is a real pleasure to be here, and I want to start by thanking the organizer for inviting me to speak about some of the research in my lab. And after listening to the other talk, I have to say that I realized that not only my talk will be very much in sync with the other speakers in this session, but also I think it'll be a good follow-up to some of the talks that we heard, because a lot of what I'll describe are methods that we are trying to develop to integrate all those different omics that are being generated. And if you've seen the last point on Rob's and Peter's slide of trying to create a system-level understanding, this is exactly what my lab is trying to do. And so first, by way of introduction, if you're not familiar with my lab, the main premise that we have been pushing for the last 7 or 8 years is that when you're trying to study a system that is as complex as the human microbiome, it is really not enough to make those long lists of gene and species and metabolites and compare them across different settings, but rather we have to apply a systems biology approach and a model-based approach in order to understand how those different elements of the microbiome come together to create the phenomena that we see. We're doing this primarily through 2 lines of works. The first line of work is trying to build models of the microbiome, focusing mostly on metabolism, as a way to try to predict how the microbiome would interact with the host, how different species in the microbiome would interact with one another, how the microbiome would respond to diet, and ultimately use this model as a way to design microbiome intervention. And this is a part of the lab that I will not talk about at all today. If you have a question, if you want more details, I'd be happy to talk about this offline. The second part of the lab is taking, again, a model-based approach as part of the analysis of microbiome data, and specifically as a way to integrate multi-omic microbiome data, which we've heard a lot today about. And I'll try to convince you that this approach is maybe the first step toward what some of the challenges that we're facing might be addressed with. And so just to give you a bit of motivation, as we heard numerous times this morning, nowadays when we have a microbiome sample, we are no longer just characterizing composition of species in this sample, but also characterizing the composition of gene and the composition of transcript and protein and metabolites. And I think it is also clear to all of us that the point is not to analyze each of these omics independently and being the 7 blind scientists that are trying to study an elephant from different perspectives and not talking to each other and getting into different answers, but rather to integrate those different omics. And indeed, the last few years have seen tremendous efforts to try to understand how the different omics that we assay from a given microbiome sample link to one another and associate with one another. We've seen, for example, studies that were trying to understand the association or the correlation between the set of genes that are found in the metagenomes and the set of genes that are found in the transcriptome. We've seen numerous studies, including some of the talks in this morning, that are trying to assay the species composition and the metabolite composition and finding correlation as a way to generate hypotheses and implicate specific taxa in the generation or degradation of a specific metabolite. And of course, this is an extremely powerful approach as a way to generate hypotheses. And again, I think Peter mentioned this approach exactly. But what I want to argue is that at the end of the day, it is also limited. And it is limited because to some extent, it is ignoring the knowledge that we have about the system. And what I'm trying to argue is that there are specific theories and specific theory in ecology and biology and genomics that tell us how these omics should relate to one another. For example, we might use metabolic modeling and metabolic control theory to tell us something about how the composition of gene and the composition of enzyme in a specific environment would affect the concentration of different metabolites. We could use information from ecology and from cellular behavior to tell us how the presence of one species in the environment may affect the behavior of another species. And the same is true for every other omic that you think about. So what my lab is trying to do is take a very different approach. And rather than finding those associations and then in retrospect figure out whether those associations fit in with what we know about the system, what we know about the biology, or trying to generate new hypotheses, what we're trying to do is start with this data, start with this information about genomic, ecology, biology, and depend exactly on the omic, building models on how two omics should relate to one another, and then use these models as part of the analysis process itself. And what I'll show at the rest of the talk are examples of some of those frameworks and how we can use that to generate potentially more insight from data that we already have. And most of the framework that I'll show you today are focusing on integrating the two omics that are probably the most common in microbiome data, which is species composition and gene composition. Okay, and so first of all, just to make sure that we're all on the same page, let's assume we have some microbiome sample with a mix of species. Each species have a different combination of genes. As we've seen, we can apply 16S analysis or a marker-based approach to characterize the composition of species in the microbiome and generate those pie chart or bar plots and understand the relative abundance of each taxa. We can also apply a shotgun metagenomic approach, map those reads to a gene catalog, and get some sense of the abundance of different gene families or different functional modules in the metagenome. But I think it is also very clear to all of us that these two facets of the microbiome are not independent, right? And what links the two is genomics. And we can represent it as a very simple model where the set of gene and their abundances in the metagenome is really nothing more than the aggregate genomic content of all the species in your community weighted by the relative abundance. And we can represent it as a simple mathematical mathematical set of equations. Now, what we have here is a very simplistic model that links taxonomic composition, genomic information, and metagenomic information. And this model is not new or surprising. We all think about it when we're doing metagenomics. Tools like PyCrust from the Hattenhauer lab are using this to predict the metagenome based on the species composition. What is surprising is how rarely we actually use this model when we come to analyze gene composition and species composition in parallel. And I'll show you a few things that you can do, and maybe the simplest thing that you can do is to visualize those links. So this is a screenshot from a browser that we hope to release in a couple of months. It's called Burrito, and it will basically, given taxonomic information and metagenomic functional information, will show you those traditional bar charts of all the species that you have and all the function, and you can look at that at different resolutions. But it will also highlight all the links so that if you now click over a specific taxon, it will show you using those gradients here what part of the metagenome is being contributed by this set of species that you selected, right, and vice versa. And you can use it as a way to explore taxonomic variation and functional variation combined while accounting for the way these two are linked to one another. You can, of course, do things that are much more sophisticated. For example, several years ago, a postdoc in my lab showed that in cases where you do not have genomic data, in other words, your sample includes many species for which you do not have genome, but you do have taxonomic information and functional information, you can use this model as a way to deconvolve the metagenome, basically figure out which gene in your metagenome must be associated, must originate from which species in the metagenome simply based on resolving this set of linear equations. A couple of years ago, a grad student in the lab, Sharon Greenblum, was specifically focusing on the cases where this model fails, when what we expect to find in the metagenome, based on what the taxonomy tells us and what the genome tells us, is actually not what we find in the metagenome. And the reason for that is because of strain-level variation that we already heard about several times today, and was able to use this to characterize extensive strain-level variation in present and absence of gene in the metagenome across numerous samples. Another postdoc in the lab show again that this model can be used to correct functional data and specifically to address a problem that we all face with in how do we normalize functional data. And so we all use this idea of compositional normalization. We look at the relative abundance of each function. We know this is a cause of problem, but if you take this model into account, you can actually use genomic data to renormalize your metagenome, and we've shown that by doing that, you can figure out disease-associated shifts that are much more pronounced. And finally, something that we published just earlier this year, again, if you account for this model and trying to figure out why do we see this tremendous variation on the taxon level, but relatively little variation on the functional level, you can show that at least some of this uniformity that have become one of the puzzling canonical results in microbiome research has to do with the way that we analyze functional data. And if you take into account this link between taxa and genomes and metagenomes, you can in fact reveal quite dramatic hidden variation in the functional data. The last tool I want to talk about in a few more details. By the way, all of these tools are available on our website. You can download them. Most of them have a relatively easy-to-use interface, and if you have any issue with that, definitely contact us. The last tool that I'm going to talk about is, again, using the same model of linking genome information, gene information, and species information in a comparative setting. And that came from discussion with our clinical collaborator that said that this is great that you can explain the abundance of gene in a sample based on the ecology of the sample, but at the end of the day, we are still interested in cross-sectional comparative analysis. Specifically, they had a set of cases and control samples. They characterized the taxonomic composition of each sample and found specific species that were enriched in disease. They did functional analysis, shotgun metagenomic-based analysis, and again found a set of gene families and metabolic pathways that were enriched in the disease state. And what they wanted to know is, can we link these two? In other words, can you tell me what is the contribution of each taxa, not to the abundance of a specific gene in the metagenome, but to the functional shift that I see in a specific disease? And can you use that to identify a putative intervention target that would rescue a specific balance? And it turned out this is a rather complex problem mathematically. It took us about 2 years to solve this. We thought it will take us a week, take us much more than that. And without going into too many details, this is a tool that we published earlier this year. It is called FishTaco, for a reason that I cannot discuss. And basically what FishTaco is doing is exactly that. It is taking samples with taxonomic profile, metagenomic-based functional profile. If you have genomic data, if you have the genome of the species in your sample, we can use that as input. If not, we use a metagenomic deconvolution approach that I mentioned before. We're using some machine learning. We're using some concepts from game theory. And at the end of the day, we are providing linearized taxonomic contribution to functional shift. And what I'll show you is just a flavor of how those results look like. So let's assume you analyze metagenomic gene content from patients with type 2 diabetes and you compare them to healthy controls. You can see that there are 3 different sugar transports that are all more abundant in patients with type 2 diabetes. And if you're doing metagenomic analysis, this is basically where analysis stops. If you also have taxonomic information, you might find specific species that are more enriched in type 2 diabetes patients, but you don't have any way to link the two. If you run FishTaco, what it will do is that it will basically show you that the enrichment that you find in type 2 diabetes in this specific sugar transport is about 80% because of certain Firmicutes species and about 10% because of Escherichia species. And in fact, there are a few species that are trying to push this enrichment in the other direction. They're just not contributing strong enough. What got us really excited is that when we apply fish taco to the other sugar transport, we see completely different contribution profile. In other words, if you're trying to reverse a specific functional imbalance, there is a very specific set of taxonomical target that you can address in order to reverse this specific functional shift without actually changing other functional profile of the microbiome. This idea of specificity between the contributor and the functional shift is something that we saw in every database that we analyzed. It turned out that those contributors are site, function, and disease specific. This is just another example that I thought was interesting. So if you look at the metagenome of patients both with type 2 diabetes and inflammatory bowel disease and compare them to control, you find that in both of these cohorts, you have an enrichment of genes that are involved in metametabolism. But if you run fish taco, what you'll see is that the underlying taxonomic contributors are completely different. So again, if you're trying to use that for translational application to figure out what is the intervention target, it will have to be very disease-specific, even though you're dealing with the same kind of functional imbalance. Again, fish taco is available online. There are several labs that are already working with that and published with that, and we have actually a web interface that will generate those plots that I've shown you before. The last thing I want to say is that this approach of using a model to link between different omics is not limited just to species information and gene information. This is a product by another graduate student in the lab that tried to ask, can I use the same approach to link the species information or the ecology of the microbiome all the way to the metabolite? And again, rather than trying to find association and then explain them in retrospect, she was trying to build a model that using genomic information, metabolomic information, some metabolic modeling, and the MEDIC database, and trying to build a very simplistic computational model that linked how the composition of species in the microbiome look like to the concentration of different metabolites. And the basic question that you want to ask is this model sufficient to explain the concentration or the variation in concentration of metabolite across different samples? And again, I will not go into too many details. The basic answer is that, yes, we can do that, at least for a substantial fraction of the metabolite. Just looking into which species are there and what are their metabolic potential would allow us to explain the variation that we see between the concentration of metabolite across samples. Again, this is a tool that is available online. It's called Mimosa. We're now working on a second version of that will hopefully be much more accurate, but you're welcome to talk to me if that's of interest. And so I think I'll end with that. I'll just conclude with this slide about critical gaps and challenges. I want to echo everything that was said in the morning about the knowledge gap, which as somebody that is trying to build models and hoping that this model will be as complete and as accurate as possible, the main challenge that we have is those missing functional annotation. This is something that in the lab we fondly call the microbial gray matter. So it's not the dark matter that are sequences that we don't have, but sequences that we have, we just don't know at all what their function might be. But I do want to emphasize again what I think is a bigger challenge is how do we move from knowing what's in the microbiome to understanding what the microbiome is doing. And I think that in order to do that, we have to develop those kind of framework that are taking the different omics and applying them into a model-based approach that is going beyond statistical association. And I want to emphasize that everything that I show you today is really just the tip of the iceberg in terms of what is needed. There are many, many challenges involved. Whenever you build a model, whenever you try to build a model, that question of scale and resolution and dynamic and time frame, and all of those things are not resolved, and there are a lot of challenges that I'm hoping the community would help us do. And so with that, I will end. Lisa promised that it will go over time. There will be a giant hook that will sweep us off the stage. Instead, we just get this. It's much less exciting. But I guess that's what we have now. I'll just thank the people that actually did the work, our collaborator, and some funding. Thank you.
He definitely wins the gold star because he came in 10 seconds under 15 minutes. So A+. Okay, so we heard a lot of examples in this session about how powerful it is to integrate all these multi-omic data sets. So we can take questions in this period, and seeing people are starting to rush to the microphones, perhaps. Great.
Great. I want to address this to Catherine Pollard. This is Marty Blaser from NYU. Catherine, in your terrific talk, you mentioned the comparison of sequences from the moms and the babies, and how when you didn't see the sequence in the older babies, you concluded that they didn't get it from the mom. But the alternative is that, that the moms are undersampled to some degree because organisms may bloom, let's say, in the babies that are not in the mom. So I wonder if you can comment on that.
It's working? Yeah. All right. Thanks. Great point. The moms are relatively stable over time, but your point was whether those might be minor constituents of the mom's microbiome. That's definitely possible. So let me tell you the resolution of what we can detect. So first of all, the whole analysis was only based on organisms where we had high enough coverage to call SNPs. For gene presence absence, you need to have about 1x coverage of the genome. For calling single nucleotide polymorphisms, we need about 10x coverage. So that's how deeply. So anything that wasn't any organism that either didn't have a genome or wasn't prevalent enough to have that coverage was not analyzed at all. So this was just for the organisms that we could study. Amongst those, we can, so because of those thresholds, we can see SNPs, so polymorphisms that would be in the mom down to, let's say there's 10x coverage if there's one read out of 10, so 1/10 of her cells, basically, amongst the sampled cells, having that low, less prevalent strain. So something below that wouldn't be detected if there was only 10x coverage. For some of the species I talked about, we actually have hundreds of x coverage, so then the resolution goes lower. So we can report a sort of a sensitivity for every species that we analyze, and it's great for some of them, and it's really not as great for others unless you do a lot more sequencing.
And so therefore I might modify the conclusion. But the other possibility is to try to sample the moms more deeply, and so that you can have a better denominator basis for this, based on the limitations that you mentioned.
Hi, there, absolutely. This was data mining a publicly available data set, so we were, of course, our hands were tied in terms of more sequencing, but that would be an important thing to do to test the hypothesis that we generated. Yeah, I'll just leave it there.
I have a question while I'm up here. So a lot of these omic data sets are sort of easy to think about, or easier to think about conceptually about how you integrate them. But in your talk, you guys were also integrating that with sort of imaging, spatial information. What are the biggest challenges that come with trying to integrate the multi-omics data just itself, and then with other data types that we're not necessarily used to thinking about in those contexts?
So I will answer it from the metabolomic standpoint. When you do untargeted metabolomics, we can only annotate 2% of the data. And so that would mean, and as mass spectrometers become more sensitive for every order increase of sensitivity, we actually increase the number of unannotated information by twofold. And so that's going to continue to increase. And so if you don't know, what the molecule is, it's really hard to think of pathways to integrate it, and that's one of the reasons why we started thinking about spatially. So for me, annotations and having reference data sets are really key to be able to annotate it. It would even be helpful if we get a molecular feature, an MS fragment of a fragmented molecule, and we can say, oh, that's in that phylogenetic tree. That's only observed in that phylogenetic tree. We currently don't have those capabilities.
From a statistical perspective, doing the correlations is really hard because the data sets are compositional, they're sparse, they're zero inflated. If you just use standard correlation techniques that make Euclidean space assumptions, you'll get 95% false discovery rates are higher, and it's embarrassing to make a network where almost all the edges are wrong. Not that you can't publish it, but you shouldn't. From a reference perspective, I mean, this like the model-based approach that Elhanan showed is absolutely the way to go if you have the genomes and if you have the models. And this is where one of those things where a few $1 million in investment in genome sequencing now could be amazing in terms of transforming how much we could get from relatively inexpensive recruitment procedures from the metagenomics instead of doing de novo assembly. yeah, and for genome mining, it would be spectacular. Although then the other problem is that a lot of it is unannotated. So even if you have the complete genome, if you don't know what the genes do, it's also hard to model that.
Brandon Eicher. First, I'd like to say I love mimosas and fish tacos. Like, that's really cool. And I was wondering, you guys are some awesome bioinformaticians and silico modelers. I'm interested in connecting those types of tools to tools in the laboratory where we can make microcosms, representative fecal samples from hosts, and start to get through some type of population level understanding of how each person's microbiome is different, but do that in the laboratory where maybe we don't have to do, hundreds of millions of dollars worth of clinicals and things. So could you speak to what you'd like to see the tools in the lab being developed to try to apply your in silico models tools to, so we can try to build these, you're familiar with like TIM1, TIM2, TNO systems that capture a synthetic human digestive system. I would like to understand more about how we can marry those two sciences together.
So I will not address this specific question. Being a complete politician, I'll maybe address the previous question more, but I think it is related to your question as well. And one of the problems with developing those kind of tools, and I think that's specifically true for tools that are trying to visualize the data, is that we have to make a very important decision of what we think is interesting, right? What kind of variation is interesting, what kind of links are interesting. And in many cases, those decisions are obvious, but I'm always a bit concerned about all the things that we miss because we bury them somewhere in the way that we decided to visualize the data or the way that we decided to aggregate the data. And I think that this kind of discussion between the experimental method and the computational method have to be an ongoing discussion, because otherwise, and we had this experience in the past, the experimentalist would get the tools without actually realizing a lot of those decisions have been made, and those decisions will be made without actually consulting the experimentalist. And so just a word of caution, I think that it's not about how do we have these tools be available to experimentalists, but how do we create a dialogue where we are in a very early stage trying to think about what's interesting and how do we want to analyze the data and what kind of signal is interesting to look for.
Okay. Liping Zhao. So I see the need of, analyzing data at a species level or other taxon level. But more and more studies showed that strains in the same species, they can show very different relationship with the particular phenotype of the host. Some may be relevant, some may be irrelevant, some may show opposite correlation. So if you analyze data even just at a species level, you may introduce a lot of noise already. How could you be sure that the functional pattern you identified actually biologically valid?
I think that's a great question. So there's two components in if you look at the genetic makeup of a microorganism. You have sort of the primary metabolism that most of us are familiar. I call it common metabolism because it's fairly common among all organisms. And then you have their specialized metabolism. These are the most divergent among them. And so we have an example with Michael Fischbach and some other people that are here as well, where we started to look at Propionibacterium acnes, and they make specific antibiotics. And if you don't have the capacity to make that antibiotic, you tend to be more susceptible to acne infections. And so we need to know that we need to have basically good reference data sets among as many organisms within the same sort of species class, as you will, however you want to define it, so that you can begin to capture it. The other thing we need to do is capture the knowledge in the community domain. So let's say, take Staph aureus, for example. If you have the MEK plus peptide there, that Staph aureus is going to be much more virulent than many other Staph aureus strains. And so if we can detect these kind of MEK plus type molecules in the mass spectrometry information, you need to start thinking about it. But we now don't have an easy way to capture that knowledge. It might be in a paper somewhere, but for me, as soon as you publish a paper, the actual knowledge that allow you to search your data is almost lost. And particularly for chemical information, this is readily lost, all the information about molecules. So we need to find better ways of capturing that information. And that will get us to that level. So it goes beyond species level. Yeah.
So with any of these techniques, they're going to be useful for some things, and there'll be limitations that you can't see. And so, for example, if what you care about is a single nucleotide change that separates your pathogenic strain from your non-pathogenic strain, the only way you're going to find that is with directed primers or with really deep coverage sequencing. But the way to think about it is, although there's going to be stuff that we're missing with the current tools, it's unlikely that the positive associations that we draw are going to be false. And you'd have to be tremendously unlucky for, say, a species level finding to not hold up when you went down to the strain level, in that you'd have to have, say, two strains with opposite effects that were perfectly balanced against each other. And that's just not that likely on entropy grounds. So I think the way to think about it is the current tools are going to miss some stuff. You'll get more when you get down to the strain level. And what that's going to be especially useful for is things like synthetic communities, where you're able to pull out a bunch of strains, stick them in a mouse or into a patient, and see if you get an effect. And there's no way you can do that with genera, right? Because it's too coarse a level of resolution. But being able to get down to the strain level is going to be really helpful for enabling that kind of experiment, and doing it with anything that you catch out of the wild, not just what you can get from ATCC.
Hey, Peter Turnbull, UCSF. So I want to ask about the smart toilet, smart mirror. I mean, I think, based on what we've heard already today, it's not sort of outside the realm of imagination to imagine collecting the sequence data and the metabolomics data. It seems like the bottleneck is then, convincing yourself that you can give, a specific recommendation directly to the consumer. And so, what do you guys think is the minimal amount of data, either in model systems or in humans, that we need to have before you tell a person at home, go eat Activia today?
Yeah, so that's a great question. I don't think we're limited by the technology so much as we're limited by the data sets. And that's why we're trying to do things like American Gut and like the large cohort studies. So trying to link up to Framingham, NHANES, other large-scale studies that are TEDI, of course, other large-scale studies that are collecting data on that scale. So you can ask, if you just had a tiny fraction of that data, would it still be useful, and what would you need to look at? The other thing to consider, like there's a whole lot of things like handheld mass spec systems and NMR systems. If you try to compare them directly to, say, $1 million instrument from Broca or Waters or whatever, and ask, can you recapture the same result in the same standards? They do very badly. But at the same time, like the CMOS sensor in your iPhone, it stacks up very badly to a century of development in film technology in terms of color accuracy and so on. But let's face it, which one are you going to use to take photos on a daily basis and actually use? So I think there's a lot of potential for technologies that are just outright worse but much faster, and trying to understand them on their own terms. What are they useful for, and does any of that intersect with what you might get in a consumer device? So just in the last week, we've been playing around with this $2 microscope in the lab, where it's this little thing that clips onto your iPhone and actually works, and gives you 60x magnification, so you can sort of put it down on a surface. It uses the light off the phone to illuminate. And is it going to replace your confocal in the lab? No, but you can probably do a lot of stuff that's useful, especially when you look at what's enabled by having a device that's essentially disposable, and where you can make a huge number of them, stitch the images together with on-device processing and so on. So I'm not sure if that answers your question directly, but that's the way we're thinking about your question.
I think we heard two really strong gaps identified in this one: the need for reference data sets and the need for annotation. So thank you.
long day.
Hello? Okay, here we go. Hey, if anybody out there could go grab either Peter Dornstein or Rob Knight, they ran off with the remote control for the slides. Where? Oh, I didn't see him, sorry. You guys are right.
Okay, good. Could everybody take a seat, please?
I couldn't see you guys over there. Oh, now I'm on.
Oh, good.
Here you go.
So you're good to go. Okay, this is session 3. My name is Dwayne Lunsford. I manage the micro portfolio for the National Institute of Dental and Craniofacial Research. For session 3, state-of-the-art tools, we're going to continue on with technologies and approaches. Part 2, this will be covering alternative models. Our first speaker will be Dr. Robert Britton from Baylor College of Medicine.
All right, thank you.
Thank you.
So yeah, thanks for the opportunity to speak. Appreciate that. I should thank Washington for making me feel right at home with that 98% humidity walking to the metro. That was quite brutal, but that's what we live with all the time there. I think our discussion at the end of the last session was really appropriate for, I think, the session we're going to get into. So a lot of the questions about what types of models can we build to emulate the microbiome outside of the host or in different types of model hosts where we have more control over the organisms are really what we're going to be focused on. What I'm going to do today start off with really what I think some of my key unmet needs are. And really what I think we need to do and what we've been doing in my lab is develop novel models for studying the functions of microbial communities, not just assembling them and looking at the structure, but also looking at the function. And I think that has been noted already several times and especially highlighted in the last session is that the lack of functional annotation of most of the metagenomes that we look at is really a huge hindrance for us. We always look at things that we understand and we know, and finding out the things we don't know is hard, and I think genetics can play a huge role in that. So what I'm going to do today is give you 2 examples of how we've tackled these problems in my laboratory, and certainly, hopefully, that will stimulate some ideas for the future. And if I have time at the end, I'll talk about some of the challenges with doing these. So my laboratory has actually taken a sort of a miniaturized approach to trying to cultivate microbial communities in vitro. As was mentioned during the questions there, you heard TIM1, TIM2, some of these SHIME models, which are much more elaborate. They're usually multi-vessel chambers trying to emulate different chambers of the intestinal tract. What we did is build mini bioreactor arrays, as we call them. They're these short, or these small little 15 ml reactors, which you can see here. And essentially we devised these because we thought that the only way to really be able to answer a lot of questions about in a hurry about functions of microbial communities was being able to do them in a relatively high throughput manner. So most of the existing systems now, it's very hard to do more than 2 or 3 at a time. So what we do is this is really a continuous flow system. Fresh media goes into the reactor, that comes out into the waste. We do this directly in the anaerobic chamber. Those chambers are heated to 37 degrees. We have a buffered system so that we can keep the pH relatively neutral. And we can really run these at any kind of retention time to mimic different parts of the intestinal tract if we want to. This just gives you an example of what one such run of reactors looks like. And so here's these 4 strips of 6. So right here's basically 24 microbial communities that you can be running at the same time. We right now have the capacity to do 96 at a time. So again, giving you a flavor for the amount of throughput that we can do. So how do we apply this? So the biggest question that we had when we decided to build a model like this is we wanted to be able to demonstrate a function. And so we chose Clostridium difficile, as was mentioned in the last session, because it's the most common cause of antibiotic-associated diarrhea. And as you heard, you can actually treat that disease with FMT. So this is the idea of bacterial function loss or community function loss that can then be regained. And so this has become a huge problem, as you can see from above there. So we set out to try to recreate basically C. difficile invasion of an antibiotic-treated human fecal community in our reactors. So what we do is we take either fecal sample from a fresh donor or from frozen. We basically put them into the reactors. We give them a little bit of time, a day, to let them grow and start acclimating to their new niche before we impose the flow on them. And then after we do that, we start the flow, and then we treat them with an antibiotic. And in this case, we use clindamycin, which is basically the antibiotic that was first associated with C. difficile in the clinic. We do that for 3 days, and then we add C. diff, and then we just simply monitor C. difficile abundance over that time. So what you can see and what we were able to do is basically show that if we take reactors, and so here we have an N of 10, and if you put C. difficile into that reactor at a fairly high level, you can see that it washes out without either at the rate of dilution, or sometimes they're even actively killed, so they'll be gone within a day. But you can see there's no invasion of that community. Now, if we treat with clindamycin, you can see that we get this nice stable invasion of that community. That stays out here, and we could go quite a ways out here with this graph now too. This community is now producing toxin, it's producing spores, all the things that we don't want C. diff to do in our gut. We've even pushed this so far as because when we first started this, of course, I'm hoping that you don't get 10 to the 6 CFU per mil if you're in the hospital. And you can see that if we go down to as few as even 150 cells inoculated into this chamber, you can see that you get a nice steady invasion. It goes and it finds its niche, which is 10 to the 6 CFU per mil. Even the clindamycin-treated communities, they actually recover before we add the C. diff, and they're at 10 to the 9th. And this is fairly similar to what you see in a patient. You do not see C. difficile as being the dominant member, even though it's causing disease. So we feel this is a fairly relevant model. Now, we've done a number of things with this model that I can't talk about now just because of the sake of time. I'll just mention that we've been able to use this model to look at the ecological advantages that epidemic rabbit-types of C. diff have. But in any event, it was really one way that we've now demonstrated a function of a community in vitro. But what are some of the other uses that we've used this for? We've been looking at a number of the areas here, and I won't go through them all, but certainly there are a lot of different things you can imagine using them for. A couple of things that we haven't done yet, but hopefully people will start thinking about using them for, is establishing microbial communities from other body sites. We're also using them for cultivation of hard-to-culture microbes. But as you can see, it's a fairly simple and robust tool, and I think the beauty of it, again, is that it's high throughput. If you have enough money in the right setup, you can be running 100 of these communities at a time, which allows you to then, if you want to form some of these synthetic communities that Rob was just mentioning about, you can do a lot of them at a time, because I can tell you we've done that, and it's pretty hard. And if you're doing it with an N of 2 or 3, it would take you a long time. So let me transition to genetics, and so how we try to tackle the genetic problem. And so, of course, as I grew up in Bacillus and E. coli, which has great genetics, and that's why we've used it for years as molecular biology tools. But unfortunately, most of the organisms that we're going to be studying in the microbiome project don't have any of these, and I think they're really critical. So we've been studying the probiotic strain Lactobacillus reuteri ATCC PTA 6475, which has been actually the subject of a few people in this room. It's an isolate that's interesting. It's got anti-inflammatory properties. It's been shown to improve disease in a number of settings, including autistic-like behavior, work by Mauro Castamatioli. But one of the challenges that we had when we started working on this strain is that we just didn't have any way of making mutations in a stable and high-throughput manner. And I really think, at least in the probiotic field, when I got into it about 12 years ago, most stories stopped because, in essence, they just did not have genetic tools to go in and try to assign the mechanism for whatever that probiotic was curing or making better or preventing. And so we turned to recombinering. So I'll just give you an example of the recombinering that we've done. And really what this is the ability to make unselected point mutations, deletions, and insertions in the chromosome. And this was first pioneered in E. coli. But we've now been able to adapt this to strains that are not there certainly are non-model organisms. So recombination is recombination-mediated generated engineering. At least that's where the name comes from. And really what I'm going to talk about today is briefly basically this activity of phage lambda. It's also the E. coli racprophage, and it's this protein beta or RECT. And what this is a recombinase that facilitates the annealing of homologous sequences in the chromosome. And so, of course, E. coli has all kinds of tools, all kinds of great things that you can do with it. It likes to take up DNA. So why did we even think that we could do this in a gram-positive organism that has 1 billion years of evolution in between it? And that really came from Don Kort's lab, where here you don't need to know anything about the data here, but what they were looking at was the activity of the recombinase. Beta is the E. coli recombinase, and this is basically the number of recombinants they could get. And what was interesting is that when they screened a whole bunch of other organisms from gram-positives and gram-negatives, you can see that they were able to get the exact same activity if they used an Enterococcus faecalis RECT. So that's something that's quite far away. So that suggested that this activity was probably a conserved activity from phages all throughout life, and that's a whole other interesting story. So to give you the idea of the simplicity of how we can make mutations in the Lactobacillus reuteri right now, what we do is we clone this RECT gene under the control of an inducible promoter. RECT is highly toxic to the cell, so this is absolutely essential for the process. And so once we make the RECT, we then transform an oligonucleotide into the cell that has a mismatch corresponding to the place where we want to make a mutation. Basically, RECT will protect these oligonucleotides from being degraded by the cell, and they will also facilitate the annealing of this oligonucleotide to the lagging strand of replication. That's then extended by polymerase, and you basically end up with a cell now that has a wild-type chromosome, or in the red you have a mutant chromosome. And so you just simply segregate those, and now, voila, you have a mutant and a wild-type, or vice versa, mutant and wild-type. The beauty is that we use an unstable plasmid, so one night of passage, we essentially have now created a strain that has a single base pair mutation or a few base pair mutations that's different. There's nothing else that's different about these strains. And so just to finish, I'll give you an idea then of how powerful technology like this could be, and hopefully suggest that this is something we could start thinking of doing in mass for some of the more interesting species that we have identified in the microbiome project. So this is essentially the mechanism of action of vancomycin. And so vancomycin is a glycopeptide that binds to the muramyl peptide as it's being secreted, and it prevents cell wall synthesis. So this dipeptide here is actually formed by this DDL ligase, diala. And so that's the 2 terminal amino acids. And so that's what vancomycin likes. And pathogens actually go in and change that last alanine to a lactate or a serine to prevent vancomycin from working. But it turns out that certain bacteria, including many lactic acid bacteria, their enzyme just automatically does this. They put in a lactate. So that's why a lot of these organisms, including reuteri, is resistant to vancomycin. And really what's exciting from Chris Walsh's lab at Harvard University, what they showed is that it was really due to this single active site tyrosine being changed to a phenylalanine. So the single hydroxyl group changes whether or not you put an alanine in or you put a lactate. And they predicted that would dictate if you would be vancomycin resistant or not, but they never had the ability to test that. So that's what we did here. We used our recombinering technology. We shot in an oligo. We changed that amino acid to change the activity. And you can see here's Lactobacillus reuteri. It's completely resistant to vancomycin. It doesn't care that it's on this E-test strip. If you delete the ability to make lactate, you can get sensitivity, but you can see all these suppressors arising here. But if you actually then do that mutation that we made, you can see that we get very good killing. It's now that's a clinically relevant level of vancomycin. You may be asking yourself, why do we do this? We want to use this as a delivery vehicle for human therapeutics. And although reuteri has never been suspected of causing any kind of sepsis, we want to have a way of being able to kill it off with a clinically relevant antibiotic when it's used. So really what I wanted to just end with is I wanted to show you two stories about how I think that we're approaching, looking at some of these more functional studies of these communities. And so really being able to cultivate, using these things as unmet needs, or cultivating, some of these communities, whether it be from disease or healthy individuals, we need to be getting a lot more individual members out that we can then mix together. As I said, I think that developing genetic tools for key members is really important, because I think now, hopefully, most of you in the room understand that strains matter. So knowing what a species is there doesn't really make any difference. And as I just showed you, one amino acid substitution can totally change your ability to resist an antibiotic. And I think that, I know we weren't really supposed to talk about funding, so I'm not going to say give us more money, but what I would say is that I get asked a lot, where do I send my grant where I want to study mutualists or good guys or good communities, in the absence of having to focus on applying that to a disease? And I don't know the answer, and I'm hoping maybe somebody can tell me that by the end of the conference. So thank you.
Am I live? Okay. Our next talk will be Angela Davis, and she will describe the Drosophila model for gut microbiome research.
Thank you very much indeed, Jane. Okay. Lovely. So I'd like to start off just by giving an explanation of the rationale for our work on Drosophila. For after all, I think I'm the only person speaking in this meeting without a backbone. I think anyway. So the rationale is, firstly, without doubt, the best system for studying the human microbiome is humans, and sustained research on the human microbiome will make us all healthier and will alleviate chronic metabolic, immunological, and possibly neurological disease. And by far the best way to get to that good place is by including the strategic use of model organisms. And model organisms, especially to determine the fundamental principles of microbiome science, so that we can identify the most productive avenues for working for human microbiomes. And in this respect, Drosophila, I think, really does have a very special place, because we can conduct experiments that would be economically unfeasible with mammalian systems, and also we bypass all of those very important animal welfare issues that relate to mammalian and indeed all vertebrate systems. In addition, Drosophila is just an amazing system for microbiome research, and that's because we can so easily take the partners apart and then put them back together again. So we can generate germ-free or xenic Drosophila simply by dunking the eggs into bleach, which removes the surface microorganisms, and then aseptically transfer those eggs into sterile medium, and the Drosophila will grow up, and we can maintain Drosophila, if you wanted to, under germ-free conditions for as many generations as you want. The microorganisms are generally culturable, and we can resynthesize the association with standardized microbiota simply by adding the microorganisms of choice to the food on which any developmental stage of Drosophila is feeding. The microbiome is dominated by bacteria, I'll come back to this point just at the end, but notice here, naturally, the diversity is low. Between 15 and 100 OTUs, that's at the 97% sequence cutoff for the 16S ribosomal RNA gene. And in our laboratory system, they're dominated by Acetobacter and Lactobacillus, and especially by multiple OTUs of 5 species, and we have isolated and characterized representative members of those 5 species so that we can generate notabiotic flies with maybe monoassociations with each one of these or with representatives of all 5 species. So what I'd like to do today is to describe 3 studies, 3 experiments that we've conducted that I think illustrate the utility of Drosophila and also illuminate something about the fundamentals of animal microbiome interactions. And the first set of experiments relates to the nutritional significance of these microorganisms. And here's our experimental design. We can rear Drosophila with basically 2 components to its diet: glucose for calories and yeast, which provides protein, essential lipids, micronutrients. And it's a 4x4 factorial design, as you can see. And to give you a sense for the scale of what one can readily do in Drosophila, we were working with a mere 4,000 germ-free individuals. And those 4,000 germ-free individuals cost us a teaspoon of bleach per 1,000 individuals. So that's the cost of working with germ-free facilities in Drosophila. Okay, so you're all welcome to come and join me and work on Drosophila. Here are the data, and along the y-axis is the proportion of insects that make it to adulthood, and along the x-axis is time. And notice that this experiment took us all of 3 weeks because there's a very fast generation time. Towards the right, where we have plenty of yeast in the diet, the conventionals in black and the exenics in red perform well and roughly equivalently. The median development time is indicated at the right of each figure. But as the yeast in the diet declines, so the performance of the insects decline, particularly when the diet becomes more unbalanced, and there is a separation from the conventionals and the exenics until we get to the most unbalanced diet, where all of the exenics die as larvae. We then went on to identify what was the nutritional problem by adding back in protein or B vitamins to that diet, and we found pretty good rescue of the exenic Drosophila by adding back B vitamins, and then characterized which B vitamins were important by individually omitting each of those B vitamins. Each data point is one of 3 replicate experiments. No significant variation for the conventionals at the top, but among the exenics, although there was a lot of noise, it was pretty clear that riboflavin is important, where we got virtually none of the exenic insects on diets without riboflavin, making it through larval development. In other words, these gut microorganisms are sparing the dietary requirement for B vitamins, especially riboflavin, with the implication that these bacteria are indeed providing these B vitamins, and we have gone on to establish that lactobacilli are important. On the nutrient-sufficient diets where development was unaffected in the exenics, however, when we examined the adult flies, we discovered that they were inordinately fat. And that obesity we could rescue not at all by adding back in lactobacillus, but to a very large extent by adding back Acetobacter. And even these early experiments were giving us an indication of a first indication of synergistic interactions, because when we added back in the lactobacillus, which on its own was completely ineffective, we got a further improvement in the metabolic phenotype of our flies. And we're now proceeding from here to use this system to explore what we call community-level metabolism. That is, metabolic benefits to the host from small communities of microorganisms that cannot be predicted at all from the effects and benefits from individual taxa in monoassociation. In my second experiment, we worked with a single diet, which was a nutritionally sufficient diet for the Xenix, but multiple different Drosophila lines, in this case from the Drosophila Genetic Reference Panel developed by Trudy McKay and co-workers. We worked with Xenic flies and notobiotic flies with the standardized 5-species microbiota, and you can see overall that the Xenics had elevated lipid, they were hyperglycemic, and a somewhat reduced protein. But this masks a considerable among genotype variation. For example, in relation to lipid, when we remove the microorganisms, some genotypes become inordinately fat, some a little bit flat, and some of them actually become leaner. In other words, we have a really important effect of host genotype on these microbiota-dependent nutritional effects. And because the genomes have been sequenced for these lines of Drosophila, we could apply a genome-wide association to identify the genetic determinants of this variation in these microbiota-dependent traits. We found that the great majority of these genes were expressed in the gut and or neurons rather than other tissues or organs of the animal, and the great majority of them have homologs in mammals right across the animal kingdom. And I think this is important because there are suggestions kind of in conversation and creeping into the literature that taxon-specific genes of unknown functions may well be particularly involved in interacting with the microbiome. I'm not saying that's not true, but certainly our data aren't indicating that. We went on to validate some of these genes, and the way to do that was we took mutations in the genes of interest and compared the phenotypic trait in the notobiotes and the Xenics, and validation is a different response to that mutation between the two microbiological conditions. And as illustrated by these two examples, and for most of the genes that we validated, but not all of them, the effect of the mutation was evident in the Xenic fly. In other words, the microbiota may well be masking the phenotypic consequences of host genetic variation, with the implication for flies and possibly for people that dysbiosis may yield different types of symptoms in different people with different genotypes. My last experiment is a transcriptomic and RNA-seq analysis that we conducted on Xenics and notobiotes, this again with the 5-species standardized microbiota, in 17 Drosophila lines that span the full genetic diversity of the species. And we found a number of instances of co-expression. That is, if the expression of one gene, in this case FOXO, is high in a particular line, so is the expression of a second gene, Chico, both of them involved in insulin signaling. And in this particular instance, this correlation or co-expression was evident in notobiotes, but not in Xenics. And indeed, if we looked right across the full extent of expressed genes, those pairwise correlations were much stronger for notobiotic than for Xenic flies. Moving from pairwise comparisons to considering multi-level comparisons, I demonstrated that nearly half of the expressed genes could be assigned to 11 modules of co-expressed genes. And of these, 7 of those modules, the level of co-expression differed between notobiotic and Xenic flies. And the pattern of that co-expression was that in the notobiotes, the co-expression was elevated in the differentially expressed transcriptional modules, not only relative to the Xenics for those modules, but also relative to the transcriptional modules that weren't differentially co-expressed, indicating that the microorganisms are organizing the transcripts in these, and it's nearly a fifth of the total of the expressed genes above the intrinsic baseline. And we are hypothesizing that this may well be involved in the promotion of robust health. So where are we? We've been adopting a taxon genome type approach from which we've discovered, I think unsurprisingly now, that microbiome-dependent host traits are very complex. They're influenced by among-microbe interactions, they vary with host genotype, and they include microbial structuring of subnetworks of the transcriptome, not just at the level of the individual gene. And this brings me to one very specific knowledge gap, which is that we really do have inadequate databases for the molecular taxonomy and function of eukaryotic microorganisms, because our recent work has been extending into particularly fungal associates, which, although of low abundance, appear to be really quite important in terms of metabolic effects. Because of this complexity, we're moving on much closer to where all the action is, and essentially the host microbiome interface, to explore the exchange of metabolites and infochemicals between the partners, and also to investigate the community dynamics of the microorganisms so that we understand their abundance and their distribution. And our approach is to use transcriptional and genomic data, many of which we and colleagues have been collecting over the years. And in here, I'm really echoing Hannon that there's a great advantage of modeling. We're using both ecological modeling and metabolic modeling to identify feasible solutions and to identify specific hypotheses. And we're now gearing up to test these hypotheses, adopting metabolic approaches and imaging approaches, so we have a really good understanding of the spatial relationship among our different microorganisms. And where our data don't fit to our models, where our data don't fit to our models, then we can use that to revise our models and operate iterative around that circle. I have a large number of challenges, gaps, and needs, many of which, before I came to this meeting, I interpreted to be rather fly-specific, but actually, they're actually echoing a lot of the issues that have been addressed today. But I would like to make one really general point, which is, if you look at my circle here, these are a lot of different techniques which are coming from different traditional disciplines. Microbiome science is inherently multidisciplinary, and as NIH takes its priorities forward, let us remember that we need to have procedures and ways to enhance and to sustain both our institutional and our funding structures to facilitate that multidisciplinarity. At which point I'd like to just thank NIH for funding lab members and collaborators, all of whom contributing directly to this research. Thank you very much.
Thank you, Angela. Our next talk is by John Rawls from Duke, zebrafish as a tractable model for host microbiome interactions.
Thanks. So my laboratory is fundamentally interested in trying to identify mechanisms that underlie host microbiome interactions. And I think that, as we've seen today and we'll continue to see tomorrow, one of the big problems facing this field overall is that we have a scaling problem. We've been incredibly effective at identifying microbial species, strains, genes, metabolites, and even host genotypes that contribute to these interactions. But I think that animal models and in vitro models, like you're hearing about in the session, have a really important role in helping us attack this scale problem in order to identify the most important factors. So I am going to talk about the zebrafish as a model system today. And one of the reasons that the zebrafish and other animals are relevant for human microbiome research is because of our shared ancestry. Everything that we know about host microbiome interactions in humans is a part of our heritage as animals. Even from the earliest stages of animal development, facets of our body plan that we know are important to host microbiome interactions evolved quite early, including the gut, lipoproteins, steroid bile salts, and adaptive immunity, all which were present in the common ancestor between fishes and mammals. And because of that, we're able to use model systems like the ones that you're hearing about in this session, because we can learn about how host microbiome interactions have evolved in different animal lineages, but also use evolution as a filter to identify and distill mechanisms of host microbiome interaction that are present in all animals and are therefore probably of particular importance in human health. So the zebrafish possesses many of the tissues and physiologies that we know are important in microbiome interactions, including an intestine with many of the same proximal distal specifications and cell types as occur in mammals, and we'll talk more about that in a minute. They have bile, mucus, peristalsis, microbiota, etc. There's also pancreas and liver and other important tissues that we know play a role in the overall interaction with our microbiome. But as with any model system, there are strengths and weaknesses, which I think are really important to consider when deciding when and where to deploy any model system. In particular, in fish, there are some important bits that are not present, including a stomach, Panna cells, and crypts. And so one needs to choose carefully when deciding which model system to use. So in the zebrafish, there are clear benefits. So as I've just mentioned, the anatomy and physiology in the fish is overall quite similar to what occurs in humans. But of course, there are some differences, right? The fish has limited utility as a model for lung biology, of course, but there's plenty of things that are conserved that make it useful. Importantly, the fish is transparent from the earliest stages of development into young adult stages, and this allows us to look at host microbiome interactions and host physiology in general using microscopy with, I think, really unrivaled temporal-spatial resolution, which facilitates many different types of studies. The other important thing, similar to Drosophila, is that gnotobiotic husbandry in the fish is quite simple, and the small size allows us to do high-throughput gnotobiotic work at costs that are orders of magnitude cheaper than what's possible in mice and other animals. However, these gnotobiotic methods were only recently developed, and this is an important point. The zebrafish as a model system for this type of work is a relatively young model. And so the community of scientists that are using it is a relatively small community, and many of the methods that are taken for granted in systems like mice are still active areas of research in the fish system. However, again, the gnotobiotic husbandry is quick and relatively inexpensive, but one of our growing edges, for example, is developing methods that allow us to grow germ-free fish to adult stages. This is something that we're able to do now, but it's something that requires further optimization. Another important thing is that during the development of the fish microbiome in the gut, it appears that anaerobes don't colonize until early adult stages. And this means that the early developmental stages, when fish are transparent and most amenable to imaging, it appears to be a niche that's colonized primarily, if not entirely, by aerobes. And so there's going to be some limitation for studying anaerobe biology in early stages of zebrafish development. The overall alpha diversity in major phyla in zebrafish are similar to those of humans, but when you get down into shallower taxonomic levels, there's not much overlap. But we anticipate that some of the chemical interactions and metabolism that these bacteria possess are going to be similar to what occur in humans. So work from our group and others over the last few years have been making contributions to the field with this model system. This is just sort of a highlight reel of some of the publications that have come out where we've been able to identify roles for the microbiota in different aspects of vertebrate physiology. We've been motivated to develop new genomic tools and genetic tools for otherwise intractable gut microbes. And work from other groups, including Karen Gilman's group, has been able to identify novel factors that regulate development of important processes like pancreatic beta cell number. So I'm going to tell you 2 short stories today that illustrate some of our recent work in using the fish to understand the depth of conservation in invertebrate biology as it relates to the gut microbiota and the mechanisms that mediate host microbiome interactions. So first we're going to ask, are intestinal epithelial cell physiologies conserved and are they regulated by conserved transcriptional programs? It's known that in mouse and in zebrafish, there are many of the same principal epithelial cell types, including stem cells, absorptive enterocytes, and secretory goblet cells, and endocrine cells. And we know that the overall physiology of the tissue is similar, but we don't know if that is underscored by a conserved transcriptional program. And then secondly, we want to ask if we can identify some of these transcriptional programs that mediate responses to microbiota. So we're going to be thinking about this through the lens of host transcriptional regulation. So genes typically consist of exons. You might think of them as having a transcriptional start site and a proximal promoter, but in fact most of the information controlling gene expression is encoded in cis-regulatory regions, which can occur very adjacent to, within, or in some cases great distances away from genes. And this is facilitated in part by packaging of DNA into nucleosome complexes. And the regions that are devoid of nucleosomes, this open or accessible chromatin, is where transcription factors can sit down, sometimes following histone modifications, and recruit coactivators and ultimately RNA pol-2 and promote gene transcription. And so I'm going to talk to you briefly about transcriptional patterns and also accessible chromatin patterns and how we can identify the transcription factors that sit down on this DNA and mediate responses to microbiota. So in one study, we took a comparative genomic approach and we asked how similar were transcriptional programs in IECs from 4 different vertebrate species, shown here, human, mouse, zebrafish, and stickleback, separated by over 400 million years of animal evolution. And we isolated IECs from their digestive tracts and performed RNA-seq. To make life a bit more manageable, we focused our analysis on one-to-one orthologs that are encoded in these genomes and asked how their expression patterns related. And what we saw was a general positive correlation between one-to-one orthologs. Each dot here is an ortholog. Here shown, for example, between the mouse ileum and the zebrafish intestine. And the genes that are most highly expressed up here in this corner are genes that we know to be important in intestinal biology, such as FABP6 and RBP2. If we take these transcriptional signatures and cluster them, these IEC data sets along with mouse data sets, we see that, compare those to several mouse tissues, we see that the transcriptional profiles of these IECs from different vertebrates cluster together distinct from other mouse tissues. And using this type of data, we're able to identify an operational set of 470 IEC signature genes. Again, these are one-to-one orthologs that are expressed generally in IECs. And this gives us a nice working set of genes to do a lot of different things with. One of the things we did, which I don't have time to tell you, is we were able to use this and other information to show that regional expression along the proximal distal axis of the intestine of many of these orthologs is conserved between fishes and mammals. And so the organization that we often think of in our own GI tracts of having a duodenum, jejunum, ileum, and colon, that program is ancestral to our common ancestor with the fishes. We also took these same tissues and looked at open chromatin using FAIR-seq and DNA-seq to try to understand the transcriptional regulatory factors that may be mediating this conserved pattern in gene expression. One result that I'll show you is here. What we did is to take those 470 IEC signature genes, used our open chromatin atlases that we have from these different species, identify these IEC signature genes that have open or accessible chromatin at their transcriptional start site, and then looked for enriched transcription factor binding motifs within those accessible chromatin sites across all 4 species. And in this list here, what you can see are the transcription factor binding motifs that were enriched in all 4 species. And And one of these in particular, which we'll come to in a minute, is HNF4 alpha. So this suggests then that there's probably a conserved transcription factor program mediating this conserved pattern of gene expression across 4 species. So one of the interesting questions that we had, and the one that really drove this study to begin with, is that now that we know what the conserved, or we're beginning to get a handle on what the conserved transcriptional program is in IECs, are microbes able to modify that program? Are they reaching in so deeply into the host biology that they're modifying this core IEC transcriptional program? So we had previously identified in zebrafish a particular enhancer which was suppressed by the microbiota. And so again, we had shown previously in 2012 that this enhancer is suppressed by the microbiota in zebrafish. And what we've reported recently is that this transcription factor, HNF4 alpha, which is one of the transcription factors that we think is involved in this core IEC program, activates this enhancer. And so we took a CRISPR-Cas9 mutagenesis approach and made mutants in HNF4 alpha, and we're able to show in a reporter system where this cis-regulatory element is driving GFP, that in wild-type animals, this reporter is very active, as we expect, and in mutants, it is suppressed, again, confirming that this transcription factor is a positive regulator of this enhancer. And what I don't have time to show you but I'll summarize, is that using these 2 genotypes and raising them under germ-free and colonized conditions, and then comparing the host genes that are regulated by microbiota and those that are regulated by HNF4, we're able to show that over 90% of the genes that are suppressed by the microbiota are positively regulated by HNF4 alpha. So again, microbes are suppressing the activity of this transcription factor, which is which regulates a variety of different genes across the genome. And this makes a prediction that the microbiota should be reducing HNF4 alpha DNA binding across the genome. So this is a point in this story where we needed to transition out of the zebrafish because currently the availability of chip-grade antibodies in the zebrafish is very limited. And so because we had been focusing our studies on processes which we believe to be conserved across vertebrates, we were able to make this transition into the mouse and ask these questions in mice. But to provide a little bit more context, we were also thinking that this was likely to have relevance for human inflammatory bowel disease. So both HNF4 alpha and microbiota have been separately implicated in IBD in the past. And for the sake of time, I'll just click through this, but human variants of HNF4 alpha have been linked by GWAS to both ulcerative colitis and Crohn's. IEC-specific knockout of HNF4 is susceptible to intestinal inflammation. Previous studies of DSS colitis had shown reduced HNF4 alpha DNA binding. And of course, we all know that microbiota have important roles in IBD pathogenesis. So ChIP-seq in germ-free and conventionalized mouse IECs showed in accordance with our predictions is that in germ-free animals, genome-wide, binding to the genome was much higher than in conventional counterparts. So again, the microbiota is suppressing the activity of HNF4. We then queried an existing data set of ChIP-seq data set for HNF4 alpha in DSS colitis. And this study had shown that upon DSS treatment, HNF4 binding genome-wide is reduced. And we took those same peaks, those 17,000 peaks that showed that result in DSS, and we saw that those same peaks were also suppressed by the microbiota in wild-type animals. In other studies that I don't have time to show you, we were able to show that human genes that are suppressed in ulcerative colitis and Crohn's are preferentially enriched for predicted microbiota-suppressed HNF4 alpha targets, again, suggesting some role in IBD. And in zebrafish HNF4 alpha mutants have an IBD-like signature, but only when they're colonized with a microbiota. So again, what these results tell us is that the intestinal epithelium in mammals and fish have a conserved transcriptional program, which appears to be regulated by a conserved set of transcription factors. And we've seen that the HNF4 alpha, this important and very ancient transcription factor, has important roles in IEC development physiology. It's suppressed by the microbiota per our results, and this is in accord with what's known about the suppressive impact of IBD on HNF4 alpha. It's important to note that these programs also likely have anti-inflammatory roles as well, which we're looking into. An important implication here is that we can use research findings from one animal model and translate them to others, and towards that build a translational pipeline towards human health applications. So these are points that I've already mentioned before. And what I think the critical challenge for the zebrafish model, at least, is we know how to build disease models and systems for studying processes in the fish. And we need to continue to find creative ways to develop more accurate and scalable models to help address the scale problem that the microbiome field is facing. And finally, I'll just point out what's probably obvious to most of us here, in that we can sort of think about the field of microbiome research as having a spectrum of human studies, which of course will have the greatest health relevance, to model systems, where you probably have the best chance of identifying mechanism. But similarly, you can study these things at the level of complex ecology with the full microbial community in the natural setting, and take it down to more simple systems such as gnotobiotics, where you're able to have a better chance at getting it function. And with that, I will close. Thanks.
Thank you, John. Our next speaker is Sharon Donovan from the University of Illinois, and she will talk to us about swine as a preclinical model for human gut microbiome research.
Okay, well, I'd like to thank Dwayne and the organizing committee for the invitation, and I will be talking today about the piglet model. So this is an overview. I'll first talk about some of the benefits of the pig as a preclinical model, what we know currently about the pig microbiome, and then some of the current obstacles. And I'd have to say I should win the prize for the cutest model. So there's a couple really nice reviews here. These are the advantages of the pig model, and some of the advantages: they're human size, they're omnivorous, so we can feed them human types of diets. There's various breeds, a relatively short gestation of 114 days. They have relatively large litter sizes, so we can get litters up to 15 and 16, so you can randomize them to different treatments, for example, different diets. I think these are some of the most important. There's high genome and protein sequence homologies with humans. Importantly, there's about an 80% resemblance with immune parameters versus less than 10% with mice. So I think as we're starting to look at host genome interactions and interactions with the immune system, the piglet is a very good model. And also many tools are available. With the sequencing of the pig genome, we have a lot more molecular tools. And so I'm a pediatric nutrition person, so I will have a bit of a pediatric focus on this talk, but I think that we don't really have a lot of good models for neonatal microbiome development. So they're easy to artificially rear, unlike mice where you have to do things like pop in a cup and put in catheters. Their intestinal physiology and anatomy is very similar brain development. A limitation is pigs, like cows and sheep and other species like that, have a poor or absent prenatal transfer of immunoglobulins, so they're completely dependent upon colostrum to get that passive immunity. So I'm just going to take you quickly through some of the benefits, I think, of using the pig model. So we can model mode of delivery very easily, vaginal versus cesarean delivery. We can model duration of gestation, so people interested in the preterm model. My lab does not do this, but there's a number of laboratories that do. You basically set up a little PICU to house your piglets. You can see the term for the pig is about 114 days versus up to 280 days. But the level of viability for the pig is much less. So a preterm pig is maybe 108 to 110 days, which is fairly close to gestation. Also, because of their bicornate, so depending on the location of that embryo in the womb, you can have within the same litter variations in birth weight, so large, appropriate, and small for gestational age. So you can have litter mates that are genetically similar that you can look at what's the effect of, for example, intrauterine growth retardation. My work, we can model the breastfed, the formula-fed, the parenterally fed neonates. I have a colleague at the University of Illinois who's very interested in using the piglet as a model for brain and cognitive development, so has developed a number of MRI methods. And so one of the advantages for the pig is that the timing of peak brain growth is more similar to the human than any other species, including monkey and rat. So that you have prenatal and then this peak around parturition. Also because they grow very quickly, so if we look at 50% brain size is 50% of adult, at 1 year of age in the human, we can achieve that in about 5.7 weeks in piglets, and 2 years, about 8.5 weeks. So the duration of the studies can be accelerated. So also, Dr. Dilger has developed a number of techniques to actually measure cognitive development. This little piggy has learned this is where his reward is going to be. So we've kind of moved away from this, but using things like eye blink conditioning where they see a light and then they get a puff of air in their eye, and we look at how long it takes for them to anticipate that puff of air, as well as the novel object recognition. And it's important to say both of these have been used across species in monkeys, rats, and are fully translatable to the human infant. So what do we know in terms of the microbiome? And this is a very nice recent review which has reviewed, again, you can see some of the species that we talked about today. And there's the relative advantages and disadvantages. So the pigs, as I mentioned, anatomical physiology, developmental stages are comparable to the human infant, generation time, microbiota, I'll show you at the phyla level, at least as the community structure is very similar. We can have gnotobiotic animals. And so one of the advantages, for example, in other species is the genetic tools, but this just came out this week in Science where I think the advent of CRISPR is going to allow us to do genetic modification of pigs quite easily. And then previously had developed transgenic pigs for overexpressing IGF-1 in milk, and those animals all had to be labeled as transgenic, even if they weren't, and all had to be incinerated. If you do genetic modification with CRISPR, you do not have to label that as a transgenic animal. So it reduces a lot of the hurdles. So just to kind of give you a broad view, I'm reviewing a couple studies in the published literature. This was recently just published, a meta-analysis. And so this group looked at 20 published studies of 16S ribosomal gene sequences. These pigs were 3 to 24 weeks of age. So typically, animals are weaned at 3 weeks of age in most common kind of commercial settings. And they had samples from 10 countries, although 5 came from sites in the US. And as we see with other studies, basically, these are the factors associated with community structure. So differences between studies, GI site, age, origin, which hypervariable region was sequenced, and the platform all can significantly affect what was reported. So I think this is consistent with what we know in humans and other species. So as we look at this, to orient you, so they took samples along the gastrointestinal tract as well as feces. So this is the upper GI, lower GI. These are the 3 most abundant phyla. So you have the Firmicutes, Bacteroides, and Proteobacteria. So if we look at the distal bowel and the colon, or the feces, you'll see that it's very similar to humans in terms of the distribution of the major phyla. And also, both for phyla as well as genera, you do see this gradient across the gastrointestinal tract. So in the gastric, in the stomach, mucosa, you'll even see Helicobacter. And then in the lower GI and feces, you see Prevotella, a little bit of Lactobacillus clostridium. So certainly at the phyla level and to some degree at the general level, there's consistency with the human. So basically in all the samples, they found Prevotella clostridium, Aloprevotella ruminococcus, and R69. These were found in 99% of the fecal samples. And then they also found a set that were shared in 90% of all the samples, regardless of the country of origin, diet, age, and breed. And so they were proposing this as a sort of a core microbiome in commercially reared swine. And I'll report this study, which is another recent study looking at the reference gene catalog. And again, they did metagenomics and found that obviously the metagenome was influenced by these factors that influence the 16S. So the first study was basically post-weaning. This is a study from David Mills' group at UC Davis. So they looked at pigs in the first 7 weeks. So these first bars through day 21 are when they were suckling on the sow. They're then very abruptly weaned, and then these are post-weaning. So you can see in this first 7 weeks, the microbiome, this is at the family level, was fairly stable. But then when they're weaned from the sow, they could see increases in Lactobacillus rhamnococcei and the Prevotellus. So, and again, from the previous slide, these were the ones that were more predominant in the older swine. They also did the metagenomics, and so the blue is the weaning and nursing, and the red is the weaned. So what they see after weaning is a large reduction in the proteins associated with lactose utilization and induction in the proteins associated with plant-described substrates, because most of the time they're weaned onto a corn and soy-based diet. So both, and then in the paper, I don't have time to present it, but they went back and attributed these different genes to different taxa. So this is some of the current obstacles. And so as we've heard from many other models, that the pig at a species level, even general level, is not the same as the human. Although, as I'll show you, the pig is more similar to the human than the mouse, and at the metagenomic level are also more similar than when we look at composition. So this was a very nice recent paper looking to develop a reference gene catalog for the pig. They did deep metagenomic sequencing on samples from about 287 pigs from France, Denmark, and China, numbers of breeds and a number of farms. They identified 7.7 million non-redundant genes and representing 719 metagenomic species. And I think what's interesting is if we look at the KEGG pathways, so 96% of those in the human catalog were present in the pig. So I think this is showing fairly good overlap between the two species in terms of the KEGG pathways. However, only 70% in the pig were present in the human. And you'll see this in terms of the next slide too. So I think there's good coverage of the human, but there's certainly species and as well as pathways that are in the pig that do not overlap with the human. I think there's a need to better understand that. So this is a figure from the paper. So these were the non-redundant genes. And so basically there was only 0.2% of the genes that were shared by the mouse, the human, and the pig. So a very small proportion. When they looked at what was shared between the human and the pig, it was about 12.62% versus only 1.3% when we compare the mouse and the human. So again, I think it's pointing to the fact that that the piglet is probably a better model in terms of some of these aspects than the mouse. So these are looking again at the KEGG functions. So these were present in 90% of the individuals. And they found a little over 3,000 KEGG functions that were shared by the mouse, the human, and the pig. This represented 69% of the pig functions, but 95 and 92% of the mouse. So I think this goes back to our reference databases that, you know, the pig I think is an excellent model, but there's less data available on defining some of these pathways. So again, I think it highlights the consistency of microbiota at the functional level, but suggests a significant set of pig-specific KEGG functions that are currently unknown. Okay. And this is some data from my laboratory where we have breastfed infants. So I think this is like also another limitation in terms of breastfed infants at 3 months of age and formula fed. At the phyla level, so you can see that's about 50% actinobacteria in both of these. Pigs, basically, 21-day-old pigs look like adult humans. There's very little actinobacteria and primarily Bacteroidetes and firmicutes. And again, we see this at the genera. This is all bifidobacteria in the human infant, and there's literally no bifidobacteria in the piglets. So this is a limitation. But, we do a lot of studies in the piglets where we will feed like a prebiotic or a probiotic, and I can shift that. So if I feed a piglet a prebiotic, this will change. If I feed an infant a prebiotic, it'll also change. But the translatability between these two has been a concern. So one of the limitations, I think I'm going to suggest we need a porcine microbiome project. I think they're a very important species, not only for production, but also as a preclinical biomedical model. And this is work that this is a review we recently done, but to get over the issue with the infant microbiome being different from the pig, we're developing germ-free and no-debiotic piglets. So these are nice isolators that are available from a company in Wisconsin, and we're able to generate pigs. And so I just have 2 more slides where we took piglets and associated them with human infant microbiota. Then they were either fed, and we gavaged them at these time points with about 10 to the 9th CFUs per mil. We either gave them formula, we gave them human milk fortified with a little extra protein, or I've had NIH funding to look at the functions of human milk oligosaccharides, which are very important in shaping the microbiome. So we looked at 2 of these oligosaccharides. This is targeted PCR, and just to point out, so that in the piglets fed human milk, we were able to maintain the bifidobacteria, but that was there in the donor stool, but not in the piglets getting formula or the 2FL, but the LN and T, one of the HMOs, maintained that. And there's very nice work at Davis showing that bifidobacteria utilize this HMO. So in conclusion, I think combined with the recently published pig genome, having a pig gene catalog is going to accelerate research that aims to deciphering these complex interactions. And as we've been saying all day, this integration of phenotypic, genomic, and metagenomic data will provide the key biological information. And in the beginning, I think that the piglet is a good model for us to model human clinical diseases as well as just normal development. So, thank you.
Thank you, Sharon. Our last talk is from Harris Wang from Columbia University, who will speak about the spatiotemporal metagenomics of the mammalian gut.
Okay, great. Thank you so much for giving me this opportunity to share with you what I think is a key knowledge and technological gap in the microbiome field. And so, today you've heard a lot about kind of spatial biogeography in the context of different locations within the body. And so then in this context of this discussion, I like to stress that what I mean by kind of space is really on the length scales of individual cells. And I think that's going to be really important in thinking about its relevance in ecology. So, natural ecological systems are highly structured, whether you're looking at a forest or the microbiome, and we know this from various imaging studies that the human-associated microbiota have very defined spatial structure, and this spatial structure is really important in mediating a variety of interactions, such as microbe interactions or host microbe interactions, as well as very important ecological interactions in terms of competition, cooperation, antagonism that really shape the stability and kind of the maintenance of what otherwise is a very robust community. These kind of local interactions are mediated by specific kind of processes, whether they be metabolic cross-feeding, such as between a variety of Bacteroides that exchange metabolites through degradation of kind of polysaccharides, et cetera. and they can be mediating a variety of both kind of commensal activities as well as kind of resist invasion of pathogens through production of a variety of bacteriocins and antimicrobial peptides. And so really kind of the key challenge, which I think is relevant in this field and related to all the model systems, is really that when we study microbiomes, we're essentially doing a one-dimensional analysis in the microbiome space. We're essentially taking fecal samples, we're blending them, putting it in a blender, and then we're looking at essentially the abundance distributions, generating what I really think is kind of single-dimensional abundance informations, which when we're trying to use a variety of kind of informatic tools to infer association information. And I think this is kind of one of the key bottlenecks in this area. And what I'd like to propose is kind of a new framework for studying this, and just share with you some of the work that we're trying to do in this space in developing approaches and techniques in this. And I call this spatial metagenomics. And the idea is that perhaps we can, instead of looking at the entire complexity of the system, essentially use high-throughput random plot sampling and statistical reconstruction to infer the overall properties of these larger structures. And this actually has a great amount of an analogy to ecology studies, where basically people go into a forest and they look at individual subplots of that forest to count the number of species and the interactions, to then build statistical inferences associated with kind of how that community is structured. And you can imagine doing this in the context of the microbiota by just essentially looking at these individual plots and inferring co-association information, which then you can infer these interactions. So we've been working on a technique which we call MapSeq, metagenomic plot sampling by sequencing, to essentially implement this strategy. And the basic premise is essentially we take a tissue, a fixed tissue of a microbiota, in this case a mouse intestine. We fix it in a fixative that essentially keeps the spatial information intact. We can then fractionate this into individual smaller particles, which retains the local information of the microbiota, which I think is really the thing that we want to capture. And then we can put this through essentially a droplet-based encapsulation system where we can encapsulate with individual barcoded beads that then could essentially amplify the specific metagenomic information associated with each of the cellular clusters. And then you can do emulsion PCR amplifications and then break up these emulsions and then perform high-throughput metagenomic sequencing. So this is just a kind of visualization of what that process looks like. You essentially embed this in the tissue. We can filter for specific sizes of these particles. And these are just cyber green stains for the DNA associated with each of these clusters. You can see that these are quite good clusters. When you put them through this fluidic system, you can also introduce methods to essentially break up the clusters once they're co-localized into individual particles. And these are just some distributions associated with this. I won't go into too much details about how we barcode these beads, but there's a variety of techniques to do that, really inspired by kind of the droplet-seq single-cell sequencing platforms. And this is just a visualization, kind of a movie showing the encapsulation using a pretty simple microfluidic chamber systems. So basically, you can encapsulate tens of thousands of these droplets in a few minutes. So, some simple kind of quality control shows that by essentially taking a pre-associated cluster of E. coli with kind of fecal microbiota, you can distinctly separate out these clusters into individuals. So here we can show that we can separate the E. coli from these other kind of fecal microbiota clusters, highlighting that essentially each cluster is can be uniquely identified. Kind of one nice thing is that you can basically also extract out kind of bulk level information just by taking the ensemble information of individual clusters, and they actually correlate very well with the bulk kind of droplet techniques, as well as kind of the technical replication of this study. So this really kind of helps us gain confidence that this technique has the ability to replicate at least abundance level information, but has this additional layer of information. And so this is kind of what the raw data looks like. So on the y-axis are just the OTUs that are identified across a variety of different individual clusters that we can sequence. And the heat colors correspond to the abundance levels of each of the OTUs for an individual cluster. And you can see that some of these clusters basically have a single OTU species, and then some have multiples. And so this is the type of information we like to be able to capture. This is just a density distribution showing the number of unique OTUs per particle, and as well as some curves showing how well we're sampling these communities and the types of interactions. So how do we start to analyze this type of information? So here is basically the different OTUs mapped back onto one another. We're basically looking at the frequency in which these OTUs are co-associated with one another relative to its total absolute abundance. So you would expect that kind of more abundant individuals would be found next to other individuals by chance. And so we normalize against that. And so based off of that normalization, you can build statistical frameworks to analyze microbiota that are essentially co-associated with one another at different length scales, especially here. So here, this is just a reconstruction of what that looks like across 2 different length scales, which we think this is going to be actually important just because it's not necessarily just who you're next to, but the types of influence that what your neighbors are able to introduce into your physiology that might be important relative for any individual microbiota. And so this is basically the same microbiome sample, but studied at 2 different length scales. And the numbers correspond to the specific OTUs, and the sizes correspond to their relative abundances. And the edges correspond to the statistically significant enriched interactions that are found. And you can see that basically at different length scales, you are potentially also seeing these different interactions. These interactions could also potentially be different across different geographic locations within a microbiome. Just for example, in the small intestine, we see different microbiota than in the large intestine, and their interactions are potentially different. Interestingly, for the same OTUs that are found in the small intestine, we are identifying potentially different types of interactions relative to the large intestine. For example, these OTUs 4 and 3 that are co-associated in the small intestines are not co-associated in the large intestine. We can also identify negative interactions, not just positive co-associations, which might tell you some things about kind of specific antagonism associated with these microbiomes. For example, here, this is a negative interaction between this lactobacilli and a Bacteroides species here. So you can start to look at this in the context of a variety of perturbations we might be kind of familiar with, seeing things like antibiotic treatment as well as dietary modulations. And I think we're really beginning to only scratch at the surface of starting to look at kind of the microbiome interactions really in the context of these perturbations once we can orient where they are relative to their neighbors. I think I'm hoping where this area goes really is just not just beyond identification of individual strains themselves, but kind of functional information. So, for example, not only can we start to think about the 16S and kind of species level information, but really we can start to then perform this type of spatial metagenomics on the whole metagenome, right? So not only understanding kind of the species, but the genes that are associated with them, as well as their transcriptional profile in the context of biogeography. So you would expect that co-localization with a cross-feeding partner may introduce additional transcriptomic changes that are really relevant only in that localized context, which is not necessarily going to be easy to deconvolute in a bulk study. And potentially, you could also link this to other immunohistochemistry approaches where, for example, you can tag the association of a microbiota to the localization of individual molecules or even the host tissue itself. And all of this, I think, really has this additional dimension of temporal profiling, which we really need to do at this length scale. So I think this really also is quite complementary to some of the more advanced imaging-based approaches for this. I think the key limitation for these imaging studies is really that we don't have the level of genomic resolution that we can get by doing deep sequencing and as well as the genomic and metatranscriptomic analyses. So with that, I'd just like to wrap up and say that I think mapping the microbiome and kind of spatial biogeography over time is really, really important in understanding mechanistic insights, right? So I think we get a lot of correlations, but kind of the mechanism really requires us to think about at the length scale that are relevant for the microbiota, which are, I think, on the micron scale. And this will help to infer ecology as well as function. The major spatial structuring in the gut seems to be only occurring oftentimes at the level of individual species, which is really hard to deconvolute using imaging methods. So I think these types of sequencing approaches are really important. And at the micron scale, individual taxa could be very heterogeneously distributed, which may be really important in the context of biodiversity. Certainly, a variety of different GI locations may be exhibiting different types of kind of association patterns that are relevant to the tissues and the types of kind of environmental cues that are mediating these interactions. And I think, additionally, these length scale interactions, studying these at different length scales as well as a variety of host variations is really important. And I think potentially maybe the spatial localization information could potentially also serve as an additional clinical biomarker. And we think this might potentially translate into kind of human analysis purely by just studying the fecal microbiota, because from our animal studies, it seems that the fecal microbiome spatial information seems to recapitulate that of the distal gut microbiota information. So with that, just like to thank the member in my labs and collaborators who have kind of helped do this work. And thank you for your attention.
Thank you, Harris. We can take questions now. Please identify yourself first.
Hi, Brandon Eicher. I liked the last talk. That was amazing. Thank you so much. I'm an ecologist, so I look for keystone species as a way to define ecological health. And I'm wondering if you think that, or any one of you would think that that might be an acceptable way to go about defining a healthy microbiome. I hear a lot of these stories from other scientists saying that we don't know what a healthy microbiome is, yet we can walk into a kelp forest and tell you, well, that's a healthy kelp forest because it has, sea otters, and the sea otters are keeping the sea urchins at bay, and therefore we have a resilient and resounding ecosystem. So I'd like to hear some thoughts on that.
So I think keystone species is really important in a lot of ecologies. I think it's oftentimes very difficult to identify them from these bulk studies because they are found in very low abundance. And so essentially, one of the motivations for thinking about kind of spatially orienting them is that you remove that abundance level information, and so that you can kind of drill down at low abundance individuals whose associations may be really important in that context. So certainly I think that's going to be really relevant in a variety of kind of studies here.
But I didn't want to steer too much to the spatial one, but that was intriguing to me as well. And have you looked at different diets, and in particular, will a liquid diet give more consistency than, say, a solid diet, especially with heterogeneous foods? Have you looked at anything like that?
Certainly we're kind of at the beginning of looking at this, and definitely we see kind of big differences between diets and high-fat versus high-fiber diets in terms of the association patterns. So I think that remains to be seen kind of what the relevance of understanding the ecological structure is. But yeah, that's a great suggestion there.
Hi, I'm Chris Lynch. I have a question I enjoyed all these talks. I have a question for Dr. Rawls about the zebrafish model. I wanted to know if some problems I've read about this model still exist. One is that there, a few years ago, there was a lot of papers coming out that they had no reference diet, and that a commercially available diet, some of the nutrients like iron, which can definitely affect the microbiome, were maybe 5 or 8-fold difference between the diets and way over the requirements. And then another thing was that when the zebrafish were kept at under normal laboratory conditions with a low-fat, commercial diet, that their adiposity ranged anywhere from 18 to 45 in males and females, and so in humans, after 35, you're eligible for bariatric surgery. So, and given that the obesity is linked to the microbiome, how do you, address that problem in the zebrafish?
Those are good questions. So the quest for a reference diet for the zebrafish that is adopted by hopefully a broad spectrum of the research community is still elusive. The zebrafish model really grew as a laboratory model with nutrition not being at the forefront. And the diets that are used commonly in most fish labs are adopted from other tropical fish species without specific nutritional requirements of the zebrafish being known or utilized in those decisions. So this is still a problem in the field. I mentioned how the community of researchers that are looking at host microbiome interactions in the zebrafish is relatively small within the larger zebrafish field. The same goes for nutrition. It's still a relatively small number of folks in the field that are paying close attention and thinking creatively about how to solve these sorts of problems. You're right. There are some studies that have suggested that some of the standard lab diets that are used for the fish produce rates of adiposity that would be considered obese by mammalian standards. In our work, looking at host microbiome interactions, we don't use those diets. We have some custom diets that we've utilized where the nutritional contents are more defined. And for our longer-term notobiotic work, we actually use axenic protozoa as the diet, which is more representative, we think, of what they eat in the wild. And so that opens up a whole other set of questions. But this is something that the field is still struggling with.
This is kind of an idea more than a question, but it relates to a question from the previous session about how to link bioinformatics tools that are looking at gene and strain level analysis to in vitro tools like the bioreactors that we heard about. So it's sort of just something I wanted to ask Robert for thoughts on. Connecting back to Howard's keynote this morning about the biological species concept and how much recombination there are between cells that we're calling the same species, I would echo what he said, that we see a lot of variation within what we would call, by operational definitions of species. Sometimes there's a lot of gene flow and sometimes there's not, and we've seen that he was looking at genomes. We've seen that in metagenomes with the tools that I talked about. So here's the idea/question. Is could we get some of those strains and test how much gene flow there is in your bioreactors, and how feasible is that? Like, what would be the limitations?
So that's a great question. It's actually something that we're doing, not from the level of gene flow, but we're trying to assemble more defined communities, more simplified, so that we can do deep sequencing and actually look at the effects on different environmental perturbations we can do on how the genomes change over time. And so that's one of the, I think, strengths of the bioreactors, is that you'll be able to sample 5 times a day, 10 times a day, depending on what kind of data that you want. And you can take that out. We've run them out for as long as 2 months. And so that's something that can be done. It's something that we're looking at with different perturbations such as diet and other things to actually do that. But we're working rather than with complex fecal communities, because the number of strains that are going to be in there are so great. We want to start with more, defined, more like 10 to 20 to start asking these questions. And one of the challenges we're having is to just figure out
who are going to be the guys that are going to assemble together, stay together, and be able to grow in the reactors. But yeah, that's definitely an application of the reactors.
So we had time for just one more.
I'm Lakshmi Goyal from Cell House and Microbe. Just to follow up from the previous sessions this morning, strain level diversity, strain level changes was talked about quite a lot. So it seems to me that some of the model systems where you have more minimal microbiota may be a good place to start addressing function of strain level changes. And in that regard, my question is to Angela. Angela, in Drosophila, if you look at wild Drosophila or from your lab versus another lab, how much strain level differences do you see, considering that Drosophila is, relatively speaking, low in complexity?
So strain diversity in terms of the bacteria, actually it's really important, and we've developed a method by which one can actually discriminate phenotypic effects and relate that to gene content and specific genes of the bacteria mediating the phenotypic trait of the host. I didn't talk about that in here, but I'd be very happy to discuss it in more detail later.
Great. Well, we'd like to thank our session 3 speakers again, and it's break time until 4:30.
Okay, for the people online we have many participants online. I think they can hear me online though. I'm just talking to the people online. We want to remind you that the people online that we set up a mechanism for you to participate in this, even if you weren't here. And so, if, some of the speakers, raise questions for you, or, you'd like something clarified in the questions session, or, beforehand, we set up a Gmail email account that you can send your questions to, and that Lita or somebody else is following. And it's etmicrobiome@gmail.com. That's a safe email account. Nothing will happen to you if you send your question in there. And then we also have, for the social media lovers, a Twitter account, #ETMicrobiome. So that's etmicrobiome@gmail.com and #ETMicrobiome. So this morning, we've in a number of the sessions, we've covered some different opportunities and some of the speakers have mentioned opportunities in microbiome research. In this next session, we're going to highlight some other general issues that haven't been discussed yet that impact human microbiome research and ideas on how to address them for the future of the field. And then afterwards, we're going to have a roundtable that will be moderated by Mike Reddy. So the first speaker in this session is Dr. Thaddeus Steppenbach. And Dr. Steppenbach is the Conan Professor of Pathology and Immunology, and he's the chief of the Division of the Laboratory in Genomic Medicine at WashU School of Medicine. And he's going to be talking to us about functional roles of the microbiome in animal model experimentation. Thanks.
Thanks, Chris, for that nice introduction. So we heard some really great talks in the last section about some interesting model systems. Some of them maybe are less explored than others. What I'm going to really focus on, I think, is a model system that's highly explored, which is the mouse system. Okay, so I think we all know. I think all of us that work with mice know that if you're working with a strain of mice where all the mice are genetically identical, we do see differences in phenotypes. And this typically is represented in cage effects that we see. So we all know this. We all have seen this. And this is just one example that we had discovered a few years ago in our facility. We're very interested in the function of IgA, the immunology of IgA in the intestine. And what we found is that in our wild-type mouse colony within our facility, that there were mice that had detectable fecal IgA and mice in other cages that didn't have fecal IgA. And this was independent of the IgA that was present in the serum. We saw this in our facility, and then we began collaborating with Skip Virgin, and he saw the same thing in his facility. So we both kind of went after this to try and figure out what was happening. And this is a phenotype that basically can be passed on both vertically and horizontally. So if a mom is IgA high, she gives rise to IgA high pups, and vice versa with IgA low. And this can be transferred then to horizontally as well. You can take IgA high and IgA low mice, put them in the same cage, and then all the mice will become IgA low. So this is, we went on to show that this was bacteria, some bacterial species that were driving this. But the real lesson here is that this is something that I think we all know we have to deal with in our mouse colonies, and that there is this transmissibility of the microbiome. Okay, so that's the easy part. The hard part is, of course, when we want to start looking at modeling host genetics in mouse model systems, particularly trying to model mutations that we see in human disease. And this is where this gets a little bit harder, and where people stop paying attention, I think, to the contributions of the microbiome. And what I'm going to go ahead and put my challenges slide up. And I think that a little bit of this is going to be preaching to the choir, but I think some of this is that this community has to go out and really expand beyond our boundaries and let people know that they need to be thinking about microbiome, particularly in animal model experiments. So the basic issue is that there really isn't a complete appreciation that the microbiome really contributes to phenotypes, not only in the intestine, not only in the lung, but really on a system-wide level. You can find examples in almost every organ system where the microbiome can affect that particular organ. And in many cases, I see this, and I'm sure many of you do, is you see studies that lack experimentation that lacks for controls for the microbiome. And these challenges really occur at many levels. This is at the level of senior scientists, junior scientists, people at the bench, but it also goes up to people that are administrators and even editors of journals. This needs to be taken seriously, I think, across many levels. How do you approach this? What's the best way to control for the microbiome? I think most of us would agree that the best way to do this is if you have a mouse that's mutant and growing separately from controls, and you see a phenotype, the thing that you really have to do is breed them for litter-mate controls. This, I think, is as many people have argued, including myself and Skip Virgin, we think that this is the gold standard. And so the key then is to do this breeding and then test to see under these conditions now are the mutants are not equal to controls. This would favor a host gene mechanism. Then you can go ahead and study your gene. If you do this, though, and the controls and mutants now have the same phenotype, this would really favor a microbiome trait dominance, and then you're trying to figure out what the microbe is that's doing this. There are caveats to this. It's unfortunately not, if you really think hard about this, it's not always totally black and white where this is going to happen. There are many examples now in the literature where you can have traits that are clearly influenced by a gene, but then that gene is also further influenced by specific microbes. And when you wind up into this land here in the middle, you need to be able to think about doing experiments that can then test for these combinatorial traits. And these can then take the form of a variety of different types of experimentation, including co-housing, including fecal transplants, including gnotobiotics and isobiotic mice, just to name a few. So do litter-mate controls matter? I think what's happening in the literature now, I think the answer is yes. There's an interesting paper that just came out in Immunity. It was looking at a gene called NLRP6. This is a nod-like sensor. I think you're going to hear more about this from Dana Philpott tomorrow. But the punchline here is that there were some papers published several years ago without litter-mate controls. Looked like a really interesting gene, very interesting phenotype in the gut of mice with injury, differences in microbiome. But when you then breed the mice and do litter-mate controls, there were no detectable significant differences in the microbiome, and there were no detectable differences in level of recovery from injury. So I think this is something that needs to be, I think it's beginning to be taken seriously, and I think there are other examples of this that have occurred and that are coming along, but this is just one that I wanted to highlight. So yes, litter-mate controls do matter. So what can we do to really facilitate reproducibility? I think there are a number of things that we can do, including reporting details of our experiments This is analogous to what went on with microarray experiments in the 90s when there were kind of crazy things that were happening all over the place, and then people began to reporting specific details of how they actually did their hybridizations, and this really helped clean up the field. I think we can do the same thing here. We can really report details of host genetics, experimental details of how you're controlling for the microbiome, husbandry details, which are really critical. Many of these can have a prominent effect on the microbiome, and then how you're actually doing your microbiome analysis. All these things really matter. And then one last thought here in this segment of the talk is that we often get we see a phenotype in a mouse and we get very excited when we figure it out, and we get very excited when we publish that paper. But I think that the time we should get really excited is when someone comes up to us in a meeting or publishes another paper that agrees with what we found. And those are the things that we should be striving to do, and have those confirmatory studies, I think, that are in the literature. They're underappreciated and I think undervalued, but I would propose that those confirmatory studies actually are just as valuable as the initial study that maybe found a phenotype with a particular gene or a particular microbe. So I think what this is incumbent upon us as a field is to share our mice and then really test this in different facilities. If we find the same thing, great. It really shows that it's robust under different conditions. If we don't, then it's actually an opportunity to figure out why is it different, and is there a component of the microbiome that's influencing the phenotype. So either way, I would say you can't lose, and we should really try and embrace this as a field. Okay, so what I'm going to do in the second half of the talk is just show you an example of how we're beginning to look for phenotypes that are reproducible, that we're interested in, how to approach this, and even how eventually to try and tie things that we know that are really variable in human, which is a very difficult system to work in, to try and model this in mice. So what we're interested in is this variation in humans in response to inflammation and infection. And I'm going to focus on one particular pathogen, influenza here. We know that this is a highly contagious virus, huge global burden, huge number a huge number of cases of morbidity and mortality every year. The interesting thing is that you can have different humans that are exposed to the same strain of flu, and there's vastly different responses to this, from just a mild upper respiratory infection to death. And so we've known this, but we don't really understand why this is the case. There's been a number of interesting studies that have been done in mice, both in antibiotic-treated mice and germ-free mice, where flu is given at a dose that kills 50% of the mice. And then when you give antibiotics to mice in that same facility, or use germ-free mice in that same facility, these mice are exquisitely sensitive to that same strain and dose of flu. So instead of killing 50% of the mice there, you kill nearly all of the mice. And so this has actually been replicated now multiple times in multiple different labs, published multiple times, and it's been suggestive that there's a role for the intestinal microbiome, or the microbiome in general, in actually mediating this. The question is what actually is what actually is the component in the microbiome, and then how do you actually get at this? So our first step was we had a hypothesis that perhaps type 1 interferon signaling was responsible for this. There might be variations in type 1 interferon signaling in patients, and maybe we could model this. So what we did is we picked on a molecule called the type 1 interferon receptor, IFNAR1, and looked at knockouts of this particular gene. This is really critical because it binds to type 1 interferons, which are generated through a variety of sensing mechanisms, and then there's an amplification of this process that's downstream of IFNAR. So when you do this, when you look at IFNAR knockout mice and do the same experiment with either control or antibiotics, now all of a sudden there's no differences in either the morbidity or the mortality with these mice. So we had the idea then that this was going to be dependent on this particular signaling pathway. So then how do you then go from having this idea about a signaling pathway to uncovering what the molecules are that are actually doing this? So this is our hypothesis. So we thought that there were specific microbial products that actually might be affecting the enteric microbiome to affect the levels of type 1 interferon signaling and therefore impact influenza pathogenesis. And so our hypothesis was that these metabolites produced by microbes in the intestine would be playing a role. So why metabolites? So this is one way. There are a number of ways that we've seen to probe for microbiome for function. These are nice because these are small molecules that are induced or produced by the microbiome. They're capable of rapid diffusion. They can interact very easily with cells in the gut. They can also then easily pass into the bloodstream and then affect distant tissues. So these are kind of nice bioactive molecules to think about. They also then are a little bit agnostic of the type of the strains and species of microbes that are present in the intestine. So what we did, so the experiment that we did was we did a screen where we took a pool of metabolites that had been shown by a group at RIKEN to be prominent in wild-type mice. And the nice thing is then these molecules, most of these we could then buy from a chemical company like Sigma, and then we then did a cellular-based assay. So we were interested in type 1 interference, so we obtained from Mike Holtzman a reporter line that essentially read out type 1 interferon signaling. We did this alone. We did these metabolites alone and in combination with essentially mimicking an infection with poly-IC or adding type 1 interferons. And then we had a simple luciferase assay to read this out. We got a number of hits with this particular assay. A number of these metabolites in this initial screen seemed to upregulate the activity of this reporter, which we thought was exciting. And then we did some secondary screens where we wanted to look for dose responsiveness, and then things that matched actually metabolites that actually were present in the serum of these mice, and found one metabolite that really piqued our interest, where we saw this dose responsiveness in combination with either type 1 interferon or poly-IC. And this metabolite was desminotyrosine. So this is an interesting metabolite. It's generated from dietary polyphenols or flavonoids. It's known to be produced by microbes in the intestine, and we found that it enhances interferon activity in vivo, which I'm not going to show. The big question is does desminotyrosine now protect from influenza? So the nice thing is we could deliver this in the drinking water of mice, and we could either give plus or minus DAT, and then prior to influenza, and then monitor the mice over 2 weeks. And to our delight, what we found in these experiments is when we delivered this single metabolite in the drinking water, we could protect from death from influenza. So this single metabolite produced, we think, by intestinal microbes was enough to protect. And then we could even do this in the antibiotic-treated mice, so we could treat for antibiotics. Remember, these mice are super sensitive to influenza, and then we could add DAT prior to influenza. And we saw also significant protection with this particular experiment, where, again, the majority of these mice were protected by desminotyrosine. We then looked into the literature, and there had been a lot of interest in flavonoids in the 90s, mostly because they're in red wine, and people love to drink red wine. So people had begun studying these and looking at the pharmacology of these and pharmacokinetics of these. And there had been a really nice screen of human microbes looking for the ability to degrade flavonoids into DAT. And one of these species was a Clostridial species, Clostridium orbicindens. So we got this strain. It was a human strain, but we were able to colonize mice pretty readily with it. And then we tested mice then that were then given antibiotics. Again, they would be super sensitive to flu, and then either colonized with Clostridium orbicindens or a number of control strains that couldn't degrade flavonoids, including Clostridium leptum and Enterococcus faecalis. And what we found, the key here is that now the mice that had received orbicindens, which is on the top, this is another Kaplan-Meier curve. These mice were protected from flu. Again, the correlation here is that desminotyrosine is a single metabolite that can protect, and then this is produced by a single microbe that could also protect. And this also is dependent on type 1 interferon. When we looked at if knockout mice, Clostridium orbicindens would colonize these mice but not protect from death from flu. So the summary here is that what we could do is take a really complex question, have a cellular screen and the right pool of metabolites, and then pull out something that was bioactive and seemed to have activity both in vitro and in vivo. So we think this is an interesting way to convert what we know about the microbiome into function. And I think that this is something that we're going to see now over and over again in this particular field. So I just want to say thanks to the people in my lab who did this study. Ashley Steed is a critical care pediatrician in my lab who really drove this. And Jared Keiko is a really talented immunologist in my lab who's starting his lab in Australia. And he's the one that conceived of the screen and did it. So with that, I'll say thanks.
And then we'll have questions afterwards. So our next speaker is Mildred Cho. She's the Associate Director of the Center for Biomedical Ethics and Professor of the Departments of Pediatrics and Medicine at Stanford University. For the last 10 years, she's been the PI of an NIH-funded Center of Excellence on ethical on genetics and ethics and legal and social implications of research. She's going to speak today to us on ethical issues in human microbiome research and beyond the usual suspects. Thanks.
Thanks, Chris, for inviting me, and Lita. So I'm going to talk to you a little bit about what I think are the critical gaps coming from the perspective of ethics in the progress of human microbiome research. And I'm going to focus a little bit more on the translational aspects and bringing the knowledge that we've all heard about today and we'll be hearing about over the next couple of days to be able to apply it to human health. So as you heard, when HMP1 started out and the field was just getting going, the initial focus was almost exclusively and still largely, there's a lot of focus on characterization at the genomic level. And that was the whole point of the human microbiome studies. It allowed you to characterize populations at the genomic level. And therefore, the associated ethical, legal, and social implications of that tended to focus on the collection and use of data and samples, naturally. And so the ethical issues that people have talked about over the last several years related to human microbiome research relates to things like returning results to humans, data sharing, ownership, protecting privacy, and so forth. And so these are all issues that relate to the data and the samples, and how do you get them, what do you tell people about them, and what do you do after you've collected them. However, as you've also heard quite a bit about, there's been a really rapid shift from the more observational studies to experimental research that involves interventions. There are hundreds of trials currently listed in clinicaltrials.gov that have the term microbiome or microbiota as of last week. And there are companies devoted to applying the results of microbiome research. Even if you look at standard business, mainstream business journals, they'll tell you which ones to invest in. So you probably can't read the letters on here, but this is just a chart from one of those business journals showing you private companies working in the microbiome space. And they have them arranged on a timeline here from early stage preclinical research all the way through phase 1, 2, and 3 clinical trials and marketing. So you can see there's already companies throughout this timeline, even at the later stages. So at some level, translation of microbiome research is going swimmingly. And in addition, it's in some ways easy to go right from some of the knowledge and the research directly to the consumer, partly because people are developing things that are aimed at nutritional interventions, and you can get this nutrition app based on your unique microbiome, as it says here. You just get the app, download it on your phone, order now. So that's just to say that these there's a wide variety of ways that knowledge from the microbiome research arena can be translated and brought to patients and consumers. And the probiotics industry was predicted to be valued at about $45 billion by 2018. So there's a big pull for these kinds of companies and technologies. So it seems that this is translation is moving along at breakneck speed, but it's also occurring in the broader context of the erosion of boundaries of traditional professionalized activities, such as science. I'm going to talk a lot more about that in a minute. But clinical applications of microbiome research occupy a gray zone between research and medicine, and between professional and what they call here citizen science. So I don't know if you can read that, but it says citizen scientist. Check out the uBiome Explorer to learn more about your microbiome. And as you can imagine, the microbiome lends itself to broad participation in research because it allows crowdsourcing of samples, ideas, analysis, and funding. And of course, many of you are probably familiar with the American GOT project led by Rob Knight. And so this is another example of a crowdsourced and crowd-funded research activity. In addition, there have been infrastructure that's been set up by patients and by non-professional communities to provide facilities, samples, screen samples, and data and networks of data for treatment and research purposes, such as this OpenBiome, which is basically a biobank, but it's for fecal samples, and they provide screening to screen out pathogens. And then people can purchase some of these samples that have been pre-screened, and it's a nonprofit organization set up by patients. So, as we've already talked about, the human microbiome may actually be a little different from other kinds of research in terms of thinking about how this is going to translate. What are the valid and viable translational pathways for human microbiome research? And part of that is because, as we've heard all day, the human microbiome defies categorization. It's hard to say sometimes who or what is the subject of human microbiome research. Is it a human? Is it the 30% or the 40% of a sample that is a human with all the bacteria on it? And is the human microbiome an organ, a pathogen, maybe a biomarker, a superorganism, or a network? And it can be many of these things. And while this is sort of interesting from a semantic and conceptual level, it's also very important from a practical perspective because categories are very important from a policy and regulatory and ethical perspective. And what makes it more complicated is human microbiome research is now also happening at a time when the boundaries between clinical medicine and research are increasingly blurred. And this leads to ambiguity about the ethical obligations of all of those involved. The subjects, the researchers, the clinicians, and everyone else who is involved, because those definitions and categories define what the ethical obligations are. If you're a doctor wearing a white coat and you stick a knife in somebody, that's probably okay. It's probably legal and you probably won't go to jail if you've done it in the context of a surgery. If you're walking down the street and you stick a knife into that same person, that's probably not okay, and there are different sets of legal obligations that go with these activities. So, and especially since research itself is defined as creating generalizable knowledge, in contrast to clinical care, which is very individually focused. When we are thinking about where we are in the context of personalized medicine, can that personalization be reconciled with the concept of generalizable knowledge? So human microbiome research is now just sort of blending with this other blurring of the boundaries, again, between medical care and research in a way that makes it very difficult to think about the ethical obligations associated with everybody's roles as well as how to regulate it. And so there are many examples of the blurring as well of the roles of professional scientists and non-professionals who are engaged in activities that look just like the things that professional scientists do, such as those sponsored like Patients Like Me that conduct research and have even led to FDA-approved products, as well as consumer-driven activities such as 23andMe, which results in the use of consumer or customer samples to create data and data publications, patents, and so forth. So there's now this need to define these terms such as citizen science or citizen scientist, and even what is a scientist. And also, if an activity is not intended to or cannot create generalizable knowledge, or if the process and the outcomes are not disseminated, that raises the question of whether we should even consider those activities science. So as an example, something that many of you are probably familiar with is fecal microbiota transplants. And there are many regulatory challenges that were posed by the activity of trying to transplant fecal matter into humans for clinical purposes. And part of that is because the FDA has categories for regulatory purposes such as drug. They had to add biologic some time ago, but even biologic drug didn't quite seem to fit with poo. And so they struggled with that, and at first they did decide to use the category of biologic drug to regulate FMT, but then partly because of the initial trials, some of which were done by individuals working on their own and not in the context of official clinical studies, seemed to be very effective. The FDA backed down and decided to use enforcement discretion, which basically means they're not regulating FMT in the way they do for other kinds of products. So because of the accessibility of the microbiome relative to other kinds of clinical interventions and the lack of high regulatory barriers, people are trying fecal microbiota transplants on their own, such as this person, Josiah Zahner, who agreed to have a reporter come along with him on his several-day journey of transplanting his friend's fecal matter into himself to help him with his intestinal problems. And so if you go on YouTube, you can find out how to do this yourself. I understand it takes a blender, a turkey baster, and some Ziploc bags. So I'm not going to show you a slide of that because dinner is upon us. But this is kind of the spirit now, is that is the do-it-yourself spirit. Everyone can just do it themselves because everyone can find a turkey baster and a blender and some Ziploc bags. You can even buy Josiah Zahner's do-it-yourself bacterial gene engineering CRISPR kit if you want to kind of add some excitement to your fecal microbiota transplant. So this is just the milieu in which we're thinking about ethical obligations in research. So in the absence of clear regulatory guidance, but also in the absence of clear frameworks on even how to think about things like self-experimentation, what are the ethical obligations of physicians, academics, or industry researchers with respect to something like FMT? And are there any ethical obligations of self-experimenters, or even more, perhaps more seriously, parents who try FMT on their children who have serious diseases? And a lot of this, I'll just remind you that if you're not doing any of these kinds of scientific activities in the context of, in a hospital, for example, or if you're not doing, if you're doing research but you're not federally funded, you have no restrictions in terms of having to get IRB approval, peer review, or requirements to publish or disseminate any of your work. So this is a completely open space in terms of thinking about ethical obligations and principles. So if you're working outside the realm of professional science and doing what some have called citizen science or participant organized science, or maybe it's just you working solo as a do-it-yourselfer, there's potential upsides and downsides to this in terms of the science itself. On the upside, potentially, you could be doing work that's more relevant to you, relevant to people that you know. It might be cheap, fast to do this. You might be able to recruit lots of people fairly easily in crowdsourced studies to get very large sample sizes, as people have seen with some of the studies using things like tracking devices, like this watch. And it might enable research not otherwise supported, say, by NIH. People have done a lot of Kickstarter projects, especially postdocs who are not able to get funding in other ways. So this can expand the scope of research and provide a diversity of expertise and perspectives that draw on many, many minds to increase the sources of creative thinking, and also potentially transparency, to the extent that there's a sort of culture of transparency in the DIY community. But there are, as you can imagine, also potential downsides to this, primarily in terms of the validity, ensuring validity, internal and external validity, especially if you're talking about these N of 1 type studies. There's also the great potential for self-selection bias. There are only certain kinds of people who are interested in DIY science or participating and who have the time to engage in these activities. So there's a big question about equitable access to participation and results, as well as the potential for therapeutic misconception, confusing clinical practice and effectiveness with the unknowns of research, and seriously, conflict of interest. Not the financial conflict of interest we typically talk about, but the more of the kind of conflict of interest we think about if you think about a desperate patient, for example, and their role in conducting research and analyzing data, as well as the lack of mechanisms of accountability. So we typically assess ethics of human subjects research according to weighing risks and benefits, autonomy, and justice. And we typically think about the integrity of research by thinking about whether data and materials are shared, whether it's published and disseminated, how rigorous is it reproducible, and so forth. But it's really unclear how to think about these principles and whether any of them apply in the situation of participant-organized science. Do they have any obligations to, even if they were to find something that was very effective, such as FMT for C. difficile infections, is there any obligation to disseminate that? Is there any obligation to in the spirit of duty to rescue, for helping other people with that kinds of information? Or also, are there any obligations to minimizing risks to people who are going to participate in that kind of activity? And does intent matter? Does it matter whether you're trying to cure your own child or just playing around in your kitchen? And so I'll just finish up by saying that this ambiguity of obligations actually does have real implications for being able to develop and assess microbiome-related interventions. And although we've heard a lot about successful clinical studies here today, sometimes these can go not so well, as described in this paper of a clinical study in which they recruited 150 individuals to receive FMT interventions, and at the end only had 3 people who completed the trial. And part of the way through, as they were eliminating people in this trial, they found that many of them, many of the potential participants basically lied about criteria that meant that they were eventually had to be eliminated from the study, or they were deceptive in other ways or broke protocol after being enrolled. So in the end, they only had data from 3 subjects. And part of this, I think, is an especially acute problem for study areas of research such as microbiome research, where there are sort of other avenues in which people can try to get at what they want or what they think they want. Either treatment or participation in studies that they control themselves. So I think the critical gap here from an ethics perspective is to clarify the ethical obligations of all participants in human microbiome research in order to facilitate reaping the benefits of this in a just way. Thanks.
Yeah, we have time for some questions, if anybody has any questions. And don't forget to you can send questions in online, too. Go ahead.
I'm deciding if I should ask the question about the beautiful interferon story or the ethics one. I'd like to ask about the second, and that's that I'm so glad that this topic is at this meeting. We often think of this as unnecessary because we presume ethicality to our activities, but you've pointed out the complexity of that. My question to you as an ethicist is that it seems culturally we're at a time when the belief or valuation of ethics is at somewhat of a nadir. And so what is the approach that those in the ethics community or in the broader community are thinking about to raise the conviction and the importance of ethics?
That's a good question. I was sort of thinking about that in terms of how one might try to increase motivation of people to think about ethics as they're participating in research. Also partly because of this idea that it's not just sort of people who have to take required courses, that get NIH funding in responsible research, but it's also potentially patients and physicians and other people who are not typically kind of in the fold of the traditional scientific communities that also need to be incented. And so I don't know what those incentives are, but I think thinking about ethics not so much in terms of regulation, but in terms of setting up systems so that they create incentives for people to think about ethics and think about what it means to do the right thing. Maybe something that might be more effective.
Hi, I agree that I wish I would get 2 questions, one for each of you, because those were fabulous talks. But I am going to ask you a question because I think it's a part of what you were talking about, Dr. Cho, which is, so how does it change it if research is being conducted on the group who are analyzing the data? Like if people use lab members, or if they use themselves, or if they use their children. Because that seemed to me kind of a blur that you were sort of talking about there in the context of clinical, but that also could be applicable to research studies.
Yeah, so this is sort of the conflict of interest I was referring to. That's one of the potential conflicts of interest. So, I know that we've had, there's typically a sense that physicians should not practice on their family members, for example, and that's partly because of the potential that you won't be as objective. And so I think that those same kinds of considerations apply to doing research on your colleagues or even on yourself. So you may be, either overestimating or underestimating risks or benefits or get too wedded to a hypothesis or so forth. And so I think with that, and so science has mechanisms to deal with that, such as peer review and, DSMBs and that sort of thing. So it may just make those things more acutely required. But I think when you're talking about desperate patients, I think that's just a bad setup from the get-go. So I think that, again, maybe the mechanisms of science that encourage collaboration and peer review and having other people get involved is one of the mechanisms that could be transferred outside the lab.
Hi, this is a question for Dr. Stappenbeck. these 83 or whatever, 80-odd metabolites, were they from flu-infected mice, or where did this list come from? These are just the most abundant?
I didn't. I kind of blew through that. So this was a study where the group did untargeted metabolomics. This was early days in this. This was a 2012 paper. Okay. So they took germ-free mice and then extra germ-free mice that were colonized with the normal, basically normal microbiome for mice, and they basically looked for metabolites then that were enriched in the presence of microbes. And so this was a starting point for us for this particular study.
I mean, it leaves the potential. There's many, many more of these out there as well. 83 is not a very large number, obviously. And of course, that's one way to do the experiment. But flu-infected versus non-infected could also be very intriguing, or something. Find metabolites that way and go that sort of route. So potentially, there's a whole number of these out there.
Yeah, exactly.
What are your thoughts?
I wouldn't say this is the only metabolite. So I think it explains the mouse story very nicely. But I think looking into human metabolomes will be quite interesting. And it wouldn't even surprise me if there are some metabolites that stimulate and some that inhibit. So I think it could be more of a combinatorial type of levels thing. So I don't think it's going to be so simple as it's a single bug, single metabolite. I don't believe that.
And in the spirit of citizen science, are you taking some of this when flu season rolls around? Or you?
I think you have to have the right bugs too. And so we only know one bug that can do this. So so the key is going to be to find the machinery within this microbe that can make desminotyrosine, and then see what other microbes actually have this. So it wouldn't surprise me if this is the only bug. So, but this is a really early phase for this work.
Hi, Dr. Cho, Lita Proctor. You didn't address this directly, but you sort of referred to it by mentioning this growth of companies, startups and otherwise. And it has to do with who owns the microbes. There's this notion that, we all have our own unique microbiome, right? So if you want to make a product or something out of it, can you cash out on that? And it came out early also in the field when people were going to these remote communities like the Brazilian Amerindians, collecting material. There's the entire issue of even trying to communicate with them about what you're doing and why it benefits society, them, and so on. And then there are other people who are actually trying to go out to remote communities to collect, archetypal microbiomes in order to discover these new molecules or new microbes. On the other hand, there's the other side of the story where the concept is microbes are everywhere and the environment selects, so there is no such thing as a personalized microbiome. But what's your community discussing around the issue of who owns the microbiome, and how do we deal with who should benefit from any products, interventions, so on?
Yeah, that's a great question. I think on the one hand, the fact that, for example, everybody's individual microbiome is, it's not something that you can standardize. It fluctuates in time and space. And so in terms of thinking about packaging human microbiomes as products, that's sort of a non-starter. And so on the one hand, that makes it hard to sort of envision sort of commercialization pathways that are viable, because you typically need to have something that's pretty standardized, that you can evaluate, that's replicable in different environments. So on the other hand, that's maybe a good thing, because then there can't be sort of monopoly ownership over it. It's hard to think about how to patent, somebody's microbiome, or even aspects of a mmicrobiome or a process that involves microbiomes, because of that. So it's so variable. So it's hard. It doesn't fit well into our sort of paradigms of intellectual property and so forth. But that kind of cuts both ways. On the one hand, that is sort of what it incents companies to move forward with developing products. On the other hand, the wide accessibility of it may be that, it can kind of throw it open to different communities to figure it out without having to deal with the ownership issue. But that does sort of raise these other questions of, if it's not standardizable, how can it be repeated?
Right. I mean, people are apparently going to airports now and sampling the sewage, pump sewage, because airports now sample all these populations from all over the world, and they go to the really large airports and they take the bilge, and that's how they try to isolate all kinds of exotic bugs. So I think you're right. The idea is no one owns this stuff because it's a waste product.
Right.
The reason I jumped up and started looking around.
Well, legally, waste is not owned. As soon as you throw something out or
the
flush
minute
it
it leaves your body, right?
Legally don't own it. So.
Right. The reason I jumped up and started looking around is I know Matt Henn is here. I'm sure he's here somewhere. So I think he told us, and I don't want to misrepresent the product, but I thought he told us that their series products were anaerobes, Clostridia and so on, isolated from human microbiome. So I wanted to hear from someone who had dealt with that, how they were dealing with the ethical issue around who owns those microbes in that product that they're now trying to market. I thought Matt was here. I saw him earlier. Okay, any other person from a company that wants to address that in general? Oh, here we go. Here's somebody. So anyway, I just want to hear
I'm not at a company, but I have worked in a company before. Normally, this is all regulated by the Convention of Biological Diversity, where all the countries who signed it signed away that the rights to all the genetic material within a country belongs to the country. And they have individual agencies which then give the rights to someone. However, several countries, it was North Korea, Iraq, and the US, they never signed this. So in the US, it means that you own the property, you own the genetic material and the rights for this material. So it's regulated individually on country per country. Interestingly, what you mentioned, the airport, I don't know how this would be regulated, because then suddenly you give your rights away by visiting another country. I don't know.
I had a quick addition to that from an example we see in agriculture where we're not really able to patent a microbe, but they're able to patent the processes to get to that product. So there may be some type of extravagant kind of process that brings a microbe to stability, and then you can put it into a product and make it shelf stable. You can sort of lock up that IP, if you will, if you're able to illustrate that for the patent agency, but you're probably not going to be able to own the microorganism.
But you could own combinations of microorganisms. There is intellectual property here. What you're going to have is you're going to have companies marketing their select combination to address your particular problem. If you have IBD, it'll be this combination of microbes that's best. If you have something else, it'll be that combination. And so there will be property that will be owned, and it will be likely combinations of composition of matter of different books.
Okay, one more question.
Do we have time for one more? Is that right?
Yeah, go ahead.
Great session. I'd like to return to some of the discussion on the mouse research. I think the point that you made about testing explicitly for facility effects is really important. And maybe it should even be thought of as a critical component of preclinical research, preclinical trials. That being said, I wonder if you have thoughts about how some infrastructure could potentially be established that allows investigators to explicitly test for facility effects, given that your average R01 isn't necessarily going to be able to cover maybe all of the mouse research you want to do in your own facility.
Yeah, that's a fantastic question, and I think it's. I think this requires this requires like all levels basically to recognize this. I think these are expensive experiments and they're non-trivial to do. And when they're done, they need to be recognized as they're non-trivial. So they need to be funded. And then when you get results, they need to be publishable in good journals. I actually think that we have these metrics that we all go by, like how many times our papers are cited. I think if you do a follow-up study like that, that's a confirmatory study, maybe you should get 2x points or something like that, or something like that, or something like that. I mean, it would just be something radical, but it would really say that those papers are important, and we really value those papers where we're trying to actually show a reproducibility. Otherwise, I think we're in a position where, if we get in a position where we can't reproduce our work, no one's going to believe us and no one's going to fund us, and then pharma's going to stop listening to us, and they're not going to care about what we say because it's just a big house of Babel or morass.
To add to that,
I'm going to let Lita decide, or do we have time for another question?
To continue that discussion, there's another example that, Eser Tolunay from Heart, Lung, and Blood Institute. Another example is the where you obtain the mice, vendor. Because there was an example where a postdoc couldn't reproduce the work of another postdoc. What it turned out to be, they had obtained the mice from another vendor. Until they equilibrated that mice in their facility, they couldn't get the same results. And it turned out it was the microbiota.
I think there are several nice examples of this where different vendors will show different results. I think Dan Littman's TH17 work is really a nice example of that, where it led to the discovery of a specific microbe that was stimulating this particular element of the immune system. And and again, I think when we find differences from different facilities or different vendors, I think we need to embrace it and actually should be okay. And then we, but I think it's an opportunity to figure out why this is happening, and I think there are examples in the literature for that. I think it's an opportunity, basically.
And you have been reporting a lot of best practices for reporting things, and one of the things I've noticed a lot of times that is not reported is what kind of diet that the animals are on, and to encourage people to use open source diets, or rather than rat chow, or purified diets they should be used, and what you used should be reported, because that could influence this reproducibility too.
Yeah, exactly. I think diet, water, husbandry conditions, even the amount of bedding that's in the cage is really critical. The temperature of the mice is really critical. We've been freezing in here all day, so our glucocorticoids are up, so it's going to alter our microbiome probably transiently. But all of these things really matter to husbandry conditions, and I think all of these things need to be reported because they could explain differences between facilities.
Okay, now they're going to stay up here, and the other speakers are going to come up. Is that right, Lita? You want to make them? You want to have them go down the front or up here? I don't know.
Okay, so I.
We need another microphone. Okay, grab, or maybe over there. Maybe on the other side, just so that we have one table and one mobile mic. Mike, the idea is that all the speakers are here, and if they have a question for a speaker, warm up.
Yes. So, hello everybody. It's nice to see people still in the room. Remember that people are still online, watching you in high definition. You all looked really good, by the way. I looked at it online. I'm a program director in NIGMS, the NSF of NIH, is how I personally think about it. So I've been charged with leading a roundtable discussion. The two problems with that is that, first of all, there isn't a roundtable. The second is by its very nature being at the end of a very long day, especially for those of you who traveled today also. This would have been so much easier for me if everybody had a beer and or a wine in their hand right this minute. So you can imagine that you can play along.
Don't run off. Greg?
I'd like to do just one piece of citizen science right now, if you don't mind. And that is to take a poll. So please raise your hand if, as a result of a talk today or chatting with somebody at a poster, you have initiated a new collaboration or you think that you will initiate a new collaboration. Great. That's just wonderful. That's just wonderful. Everybody online? There was another 7 hands that just went up. So that's very, very good. I honestly didn't expect that. That's wonderful. So most of you today did a very good job of addressing what we've asked you to address, which is a critical need or a gap. So on behalf of all of the organizers, I thank you personally and professionally for doing that. Hopefully this is going to be sustained for the next 2 days. Please consider, though, if it's appropriate to lead or put it much earlier in your talk, the slide with the critical need or gap, because there was a handful of talks where it was the last slide and it was up there for sometimes literally 10 seconds. Okay, so for those of you who are in the room or watching online who are going to talk the next 2 days, please consider that. All right, so what I'm going to do is now step back because this is your meeting, and I'd like to invite everybody who feels like asking a question to any of the speakers from today to please grab a mic and ask. If you can, try to do it in a big picture way, right? If you ask a very highly specific question of one particular speaker, it sort of defeats the purpose of the non-roundtable roundtable.
And there are 2 of us that are running mics. There are 2 standing mics, and the speakers have those mics that I just lifted from the table.
So maybe it's still in my mind because Thad just spoke in the last session. But the point that Thad makes about the role of the microbiome in phenotypes, this audience really appreciates it, I think, because they've seen it closely. What I have observed in my role as an editor is some of this you have to go out and evangelize to your colleagues in fields where the role of the microbiome is not appreciated, and it has a profound role, and it's sometimes not recognized, so especially mouse studies. So Thad, maybe you can take that.
This is the first time I've talked about this. So I've talked about this in many forums, and usually people get so angry with me, and it'll go on forever. And usually, and if there's alcohol, it's even worse. Because again, I think Lakshmi is exactly right. I think everyone in this room appreciates this. Everyone's seen it. If you work with any kind of model organism, you understand this. But we have to convince our colleagues and in other fields. I think it's really incumbent on us. I think I like the word ambassador. I think that's what we need to be.
Hi, I'm Tom Sharpton from Oregon State University, and I've got a question for, I think, maybe every speaker might have an opinion on this, hopefully. I think the history of science is littered with revolutionary findings that resulted from challenging assumptions. And the microbiome is a ridiculously complex system, as we've seen today through many discussions. And when we study the microbiome, we consequently make a lot of simplifying assumptions so that we can wrap our head around it and understand what's going on. I'd like to know what the speakers think is the most significant simplifying assumption we're currently making that we should try to embrace and destroy to improve our understanding of the microbiome.
But I think the community as a whole often associates association with causation, and I think we have to watch out for that. In the early days of microbiome research, that really seemed to show up very prominently. So I think we probably have to be a little careful about our language. Would help, I think, mitigate some of that. That would be probably the number one thing I keep seeing pop up, but welcome to hear other people's thoughts.
One of the other things which has come up a bit is the importance of getting beyond species or below species, because so many traits of bacteria do not map well onto species. And we all know, because of horizontal gene transfer, that we can have traits that are a lot of functional redundancy, and in addition to that, we can have a lot of heterogeneity that we can't detect taxonomically, and of course we also have phenotypic heterogeneity.
I'm Jen Fetweiss from VCU, but I'm actually just representing the ET Microbiome social media team, so I'm just going to read a couple of questions that have come in from Gmail and Twitter. The first one was from Julian Avila, and this was one that was tweeted out earlier. It was a specific question, but I think it could be generalized. The question was during the keynote for Howard Auckman's talk, why is dry erase B a good target for phylotags? And then there were some follow-up questions about what proportion of the human microbiome can't be classified, but I think we could think about that rather than just that specific method. What proportion are we missing using a number of the techniques that have been discussed today?
I won't speak to gyrase specifically, maybe Howard wants to, but I think there's quite a few proteins that are good taxonomic markers. The second question, though, I think is maybe more important about what we're missing, and I'll just recall a slide from my talk about what's, if we are interpreting our data in the context of what we already know from things we've sequenced before, the answer is most. We're missing most of it, and that varies depending on what environment we're studying. And just since we talked about ethics at the end today, just to mention that there are biases in what we know based on certain populations of people, in particular from North America and Europe, dominate what we know. And it's not clear how that's going to play out as we get genomes from more diverse groups of people. So I think that's really important from the perspective of understanding knowledge, and it's also really important ethically that we get genomic whole genomes from more diverse environments, including more diverse types of people. Yeah, good point. I totally agree with what Mike just said. Also, it's mostly bacteria. What about, there's not that many archaea. Well, the poor microbial eukaryotes are often missed and super important. Did you want to elaborate? No. Yeah, totally. All the viruses, especially the phage.
No.
Li-Ping?
Yeah, okay. I have comments on two things. One is this unclassifiable question. I think many people assume that you can only study those bacteria if you can classify them, if you can give them a name. But this is actually a misconception, because when you have a unique sequence, you know it represents some biological entity. No matter you cannot give a name or not, you can still work on it. You can still do statistics, you can do all kinds of characterizations. That's the purpose of sequencing, directly working on the sequence. So I don't think many people, they just work on the classify the sequences, and they discard the rest unclassified sequences. So that's probably, that's one comment. Another comment is we can now work on to string levels because we can assemble high-quality drafted genomes of prevalent gut bacteria now from the data set, particularly from interventional trials. And, but the question is still the sparsity question, because any single strain, if it's shared by more than 20% of the samples, would be already considered a prevalent bacteria. But you still have a lot of zeros in your data set in many of the individuals. And so I think we should introduce the concept of guild in ecology. So where you look at the associations or co-occurrence of individuals. And in one of our studies, we showed that you can actually identify about 10 to 20 guilds among nearly 200 strains. And then in each guild, the members actually increase or decrease together. But they can be from very different phyla, but they work together. So then if you get abundance at the guild level, then they can correlate with host disease phenotypes very nicely. But the individual strains cannot. So probably I think we probably could need to cluster bacteria based on what they do together and who does what with whom, rather than based on who they are, what we call them. So it's just a comment, and I like to hear your feedback. Thank you.
Yeah, so the only thing I want to say about, I agree with everything that you say, but I think the one comment I want to make is in continuation to what Mike said about this assumption that we used to make about association and causation. I think there is a similar assumption in microbiome that co-occurrence means interaction. And this is another assumption that I think we should validate before we take this into account. And in many cases, we know that co-occurrence has nothing to do with interaction, it has to do with external factors. And so while I think this idea of guild is something that is very powerful and we use it in our research, I do think that being able to identify the individual player functionally, and this is why we want the strain, it's not about naming them, it's about knowing what they do, is crucial for a lot of the functional research that we want to do.
Yes, so the beauty of getting the nearly complete high-quality drafted genome is because you are able to look at the functional genes. And in one case, about 30 strains associated with the urine TMAO concentration positively, but only in half of them actually encoded the two genes, converting choline into TMA. The others, they are just a mere association, but they don't actually have the functional gene, yeah.
Okay, Lita, you had a question back there, and I think there's one more afterwards.
Yeah, Justin Sonnenberg, Stanford University. We heard some beautiful talks today on longitudinal studies in humans, and certainly with so much variability and such complexity, applying multi-omics to people over time seems like a great way for discovery and actually learning things about this complex system. For those of us that are trying to design studies in the context of an R01 budget, as soon as you start sampling at a lot of time points and applying a lot of omics, your N goes down drastically. And so, and as Curtis said, there's no silver bullet for what sort of measurements necessarily that you want to make. And again, this is discovery, and so generating as much data as possible is wonderful. And so I'm wondering for the people that have been conducting these longitudinal studies, if there are any principles that you could pass on or that you're after that you could describe to us that might help inform the design of such studies, because this seems like it will be a major theme going forward.
I mean, in no particular order, and probably mostly things that you already know, but as general principles, think, as Owen said earlier, think a lot about baseline metadata in a consistent format. Recontacting is invaluable. Having samples in a format that allow you to come back and do targeted culture microbiological experiments in addition to your favorite omics is surprisingly difficult in large-scale human studies, but really worth it. And fewer subjects with deeper characterization, as Mike said earlier, can be very powerful. So those are my quick top-of-the-head comments in that vein. Mike, do you?
Yeah, I mean, I hope that the research we're doing will appear valuable to the ears that are listening, that they will launch large-scale things. I mean, I participate in lots of large-scale projects, many of you know, and they have a role, and it's very different from the role of small projects, individual investigator projects that I think are equally, if not more important as a whole. But I do think we need these large-scale projects. They are expensive, but they do reduce redundancy. I mean, I remember when I was a new assistant professor, someone in my lab spent a year sequencing a gene. That's how long it took then. And 2 other labs did the exact same thing. We were all funded by R01 research. There was a lot of built-in redundancy that was out there with people doing the exact same thing. And I think the advantage of these consortia is you can take on large projects. You can pull different kinds of data, different expertise. And every consortia I've been in, as long as people are, and they seem to be, they share ideas and things, that really does pull experts together to be much more efficient than I think R01s are normally. So I hope that will show a lot of value from some of these large-scale projects. And again, the relevant people, at least some of them I know, are in this room, and they'll see there'll be some extensions from these sorts of things. What are the next set of questions we can play? Multiomics assays. They are expensive. I don't think there's any way of getting around that. And it's true the price goes down, but the reality is we always go deeper, right? We always sequence a lane of HiSeq, no matter what we do. We get more reads, and then we see more. They don't seem to get as cheap as you. It's not perfectly cheaper when you get more depth. But anyway, you know what I'm saying. So I think it would be nice if we can continue these kinds of things. I know, speaking for myself and others I've listened to here, we are looking for our next phase. So that's self-interest speaking, I guess that.
And can I just address it also from a program perspective? The purpose of the HMP was to build those databases, those reference databases. And we've done things like gone in to see how many other people use the reference data to test a new hypothesis or build a new computational tool. And over the years, we found roughly half of people who are not funded by the HMP, who are using the HMP reference data, are testing new hypotheses. So it's kind of like free data. What it costs you is server time, but the experiment has sort of been done for you and maybe allows you to do some dry experiments before you commit to a set of assays or whatever. So that is actually the purpose of the HMP resource.
So I just have one thing to add to what Mike and others have said here. And it's basically that I think that prior planning is a really good idea, and I think maybe this is one place where the community could potentially help. When we started our project 6 or 8 years ago, we pretty much started it from scratch, from the seat of our pants, without a whole lot of input from other people who'd done large projects like this. I think that we could have learned a lot from other people who'd already done it, and probably there are people that could learn from us now. So I think as you're going into a big project like that, where you're going to do longitudinal studies, taking hundreds of samples from dozens of people, that it probably makes sense to make sure your IRB is written right, you've got everything, all your ducks in a row before you start, because if you have to change something in midstream, it's a real difficult challenge. So that's something the community might be able to contribute, is some kind of guidelines or help or boilerplate protocols for people to use.
I think we have time for one more question.
Rajiv Agarwal from Office of Research on Women's Health, NIHOD. So my question is, it's a very naive question, and I came late to the meeting, so maybe I missed this. So when we are talking about microbiome, are we specifically restricting our area only to bacteria? Because these days we are looking like bacteria, virus, fungi, even helminths and protozoans. So are we talking whole or just bacteria here?
I'm going to just respond in general. I don't do the work, but I've been following the field. You know, the bacteria, we know the most about the bacteria because we had handy-dandy biomarkers and all that. I think all along from the very beginning, we acknowledged and realized there were other major components of the microbiome that we were not including in a systematic way. We did do some viromic work, a little bit of microbial eukaryotic work, but everything that's microbial life goes into the microbiome. There are major holes. And tomorrow, right? Tomorrow is when we're going to hear from people who are working on phage and people who are working on fungi. So they'll champion the need to include those other microbial components.
So before I officially adjourn today, go ahead.
Was there another question from
There were
online?
two more that came in, but
Very
we can.
specific.
Do we have time?
Could we just squeeze one in?
Sure,
Because I
go
want
ahead.
the online audience to feel like they're included.
You're Lita Proctor, of course you can.
This was a very specific question to Greg Buck, but I think it might touch on some things that we haven't talked about today. So the question was, do you have plans to analyze the placental microbiome? And if so, were the births vaginal C-section? And how do you control for vaginal cross-contamination? So although this is a specific question, I think it touches on sample collection and controls, particularly in low biomass samples.
Right. So we have, as you know, Jen, and probably everybody else knows too, we have birth products from all over the neonates that were born. We haven't really analyzed those yet. We do have plans to do that. We don't have money to do it, but we do have plans to do it. The concern for looking, there have been a couple of publications on the placental microbiome and the microbiome of birth products, and it's controversial. I think that there's some report that there's a placental microbiome, and there's some people saying that it's maybe not a placental microbiome. The dogma is that the placenta should be a sterile environment. Of course, that some of those dogmas are falling. So I think you have to take this very carefully. So our plan is we would like to look at the birth products. I think in L&D we're well aware that that taking these birth products are not done under completely aseptic conditions, and one has to be very careful how do you interpret that. So basically we're looking probably at a low biomass issue. We can do controls. In all of our samples we have negative controls and positive controls. That doesn't really control for contamination of the particular organ that you're looking at, or the sample that you're looking at. So one of the ways that you can look for contamination is for example, if you're looking at a placenta or a birth product, you would expect the major contamination to come from the vaginal environment. It's not entirely true, but you would expect it to be coming from a body part of the woman. And since we have data on that, if you saw a microbiome profile that looked like the vaginal profile, you'd say, well, maybe that was a product of contamination. If it's very different, then you say, okay, is this real, or is it environmental, or does it come from the reagents that you're using? And I think you have to be very careful there, because if you're looking at these low biomass samples, you have to be very careful that you're not looking at contamination. So we have plans to look at it when we get around to it and have enough money to do it. We'll take these things into account.
Okay, so I just want to finish by saying the following. So in 2008, I left academia and my beloved phage T4, and I started to be a rotating program director at NSF, where I was for a 2-year stint. And I have nothing but good memories there, but the best thing that happened to me is that I met Lita Proctor at NSF. And I overlapped with her, and I was there at the moment, right around the moment that she got a clue that she was going to lead this when she left NSF. And I'll always remember that. And everybody knows how hard she works. She's a driving force on every level that a driving force could be defined. She's a wonderful person. I think her modesty was manifested when she introduced herself, when she asked Dr. Chow a question a little while ago. But the thing that I think is the most special about her, and that you probably don't know, is that she has this incredible ability to treat people better than they treat her. And I very often have seen her or heard her on our many phone calls and planning meetings about this very day and this very week, where she did exactly that. So I'm going to adjourn today by having us all thank Lita for being Lita.